I'm gonna do a clap test, Grove. Wanna frame me up? All right, you guys ready? Boy, Grove, it's Rich, it's loud in here. Jeez. All right, Grove, you framed up on me? No, I'm not. Okay, well, come on. Okay, you ready? Yes, sir. Okay, Ben, you ready? Ben? Here we go. 1, 2, 3, 4, 5, 6, and I'm not even clapping that loud. Let's do it again. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You good?
Please welcome to the stage Stat Senior Writer for Medicine and Editorial Director of Events, Matthew Herper.
Hey everybody, welcome to Stat Summit. I just met one of the biggest celebrities I've met in my 20 years of journalism. There is baby KJ backstage, so I just, do get your hopes up. It's great to see all of you here. I am Matt Herper. I'm the Senior Writer for Medicine, Editorial Director of Events here. And as I said, welcome. In journalism, we're taught to lead with the news. So I have some news for you guys. These are things that weren't on the agenda. First, at the CDC session later this morning, we will have former CDC Director Rochelle Walensky, who will be speaking about lots of things, including this weekend. This afternoon, we've added a session with Matt Might. If you don't know of Matt, he is an amazing researcher and expert in AI, who was inspired by his son, who was the first person diagnosed with a rare disease. Sadly, he's since passed, but Matt is inspirational, and he's going to be talking to our award-winning reporter, Casey Ross. So that's fantastic. And our executive editor and co-founder, Rick Burke, will be interviewing Joe Kennedy, but that was going to be today. It's going to be tomorrow. Some more big news. We have a new member of our events team. This is the biggest news for me. And if you followed Stat, you know him and probably love him. Damien, are you here? Damien's way in the back of the room hiding. Damien Gardet returns to Stat and to events, and he's there, and you can all accost him afterward. So this is an important place in journalism, Stat. It's our 10th anniversary. I wasn't here for the first 3 years. I didn't come when Stat started, and Rick Burke, who you'll hear from in a moment, will never let me forget that. But that means I had the ultimate case of FOMO for a long time. I had my nose pressed against the glass and could really see how important it was to have a place like this that does journalism about medicine, about biotechnology, about healthcare. In a concentrated way, but is not a trade and has national ambitions. And it wins a ton of awards. It was a bummer, actually, because I saw they wrote a story about my friend Adam, about how he was the scariest dude in biotech, and then they hired him. I thought, it won't be me. And then I got a call from Adam to call Rick. And I'm so happy to be here. All of you know this publication is indispensable. We felt it during the pandemic. It's true more now than ever before, and we appreciate your help. We appreciate your help that you're here. We appreciate your help that you subscribe. We appreciate your help that you share our stories. So here we go. We're going to start the day with the incredible story of baby KJ. After that, powerhouse panel, John Maraganore, Chris Viabacher, and Emma Walmsley. Then Stat's Daniel Payne will sit down with former CDCers Rochelle Walensky and Daniel Jernigan for an insider's look at an agency under attack, and before lunch we'll be joined by the internet's most influential physician, Dr. Mike. Lunch will be great. We'll come back for a fun demo on patient tracking that could change clinical research. Then Catherine McPhail, my right hand, will introduce you to a few of this year's Stats Wonderkins. This year there's a biotech focus. We'll have the CEO of Acacia here to talk about China's booming biotech industry, and we'll have the Matt Might session, and we'll end the day with 2 biotech CEOs that were impacted by the changes at the FDA. That's just today. I'm not even getting into tomorrow, but you do want to come back. We're the very model of a modern biomedical news source. We have an app. There's signage around the venue with a QR code. Download it. It's on screen. You really want this? There are going to be poll questions. Need the app to answer the poll questions. It's way more fun for us and you if you have it. I want to introduce you to Catherine. This is Catherine McPhail. Catherine, come up on stage. Catherine is a journalist on my team. We couldn't put this agenda together without her. She will be up here on stage throughout the next few days, but even more importantly, If you have a question, you should look for me, you should look for Catherine. You should also, there are two other reporters, the amazing Isa Cueto and the amazing Lizzie Lawrence, who will facilitate Q&A throughout the program. If you have a question, flag them down. So, a special thanks to the Stat Summit sponsors who have helped to make this event possible. Our presenting sponsor, Merck, our Stat Wonderkind sponsor, the University of Texas MD Anderson Cancer Center, our premium sponsors, Biogen, Genentech, Genmab, Medidata, and Vive Healthcare, and our supporting sponsors, the Chan Zuckerberg Biohub Network, Click Health, and Zeno Group. We are thrilled to have you all joined and encourage you all to share your thoughts on social media, please. Use the hashtag StatSummit on your favorite social platform. And with that, let's get started. Here to welcome you all to our 2025 flagship Stat Summit, please welcome Stat's co-founder and executive editor, Rick Burke.
Hi, everyone. Can you hear me? It's great to, I have a mic. So great to see all of you. Thanks for coming. I just want to build on what Matt said about his coming to Stat. Starting Stat in late 2014, I didn't know anything about the subject matter, and I asked everyone I could turn to, like, who is the best reporter in health and medicine? And I put lists together, and everyone said, Matt Herper, Matt Herper, Matt Herper. And I'd never met Matt. So I brought him to John and Linda Henry's house, and we tried to sort of woo him. And he said, you know, I'm really thinking about it. I'm going to even move to Boston. And then at the last minute, he pulled out, cold feet, and then switched a few more years. I said, I'm giving up on him. And a few years later, talking about the FOMO that was going on, we had an event in New York with myself, Damien, Adam Feuerstein, I think Aaron Mershon, our then Washington correspondent, and I could see Matt in the audience, like dying to be on the stage with us. I could just feel it, and he was like asking questions and everything. And then after that, Adam said he really wants to come to Stat, and he came, and the rest is history. This is our 7th flagship summit. I think it's sort of what everyone told me at the beginning about Matt. Not only is he an amazing reporter and knows the space, but there's no better events programmer in our space. He puts on, he's the architect of all that. We have all this. We have a great team, but he is the architect, and he puzzles through day after day, month after month, to bring the best programming you can imagine, as well as, and he's always said, the people in the audience that attend summits could easily be on stage. And that's the kind of group that we've attracted here today. So all of you are important, dazzling people that we're happy to have here today. I just want to say a little more about Stat. As Matt said, it's our 10th birthday, and how are we to ever know, you know, COVID, where we'd have Helen Branswell leading the way, leading the charge in coverage in the world about, you know, COVID is coming. We took on Purdue Pharma and won that suit over years of litigation, which led to all kinds of marketing information about the marketing of OxyContin that hadn't been out before. We took on IBM Watson for cancer rather, and showed that it wasn't sort of all that it was cracked up to be. We've taken on United Health, on and on. And of course, right now, our mission could not be more essential than ever in covering and writing trustworthy stories about what's going on in the sort of upending of science and health as we know it in this country. Our reporters are fair-minded, non-political, meticulous in what they do, and the great thing about Stat is every other outlet has a lot going on to cover with Trump and what's happening. But we have a single-minded focus and mission, which is health and medicine. And look at that press conference the other week with Trump talking about, advising you to take Tylenol and so forth. We wrote the lead story in most other publications that evening was Jimmy Kimmel comes back to TV. And that's fine, because that was a big story. But we don't have to worry about Jimmy Kimmel. We wrote 16 stories about that extraordinary press conference and what it meant for the health of Americans. And so what I'm saying here is we take our role very seriously. I don't think there's a collection of more authoritative, trustworthy, smart reporters covering these stories. I don't think there's a collection better anywhere in any news organization. And I'm just so proud of our team and what they do. And let me just ask you, how many of you regularly read Stat? And how many of you read Stat every day? Well, that's good. I think I want the numbers to go up to all of you reading at least once a day, because frankly, we have, you might not know this, but we have anywhere between 12 and 15 stories every weekday. We write a lot of stories, and sometimes you can miss out on them. And they're, so I just want to encourage you to know we are all over this. We are all over these stories on weekends, at night, everything. So I hope you take the advantage of that. And I really thank you all for coming. I think this is going to be a really blockbuster, great couple of days. So thank you very much.
From our presenting sponsor, please welcome to the stage Merck Associate Vice President of Neuroscience Discovery, Matt Kennedy.
Good morning, everyone. It's a pleasure to welcome you to the 2025 STAT Summit. I'm Matt Kennedy, and I lead Discovery Neuroscience Research at Merck right here in the Boston and Cambridge area. It's a real honor to be here among so many future leaders of the future of medicine today. And this year's theme, the next 10 years, invites us to look ahead with intention, while also reflecting on what has reshaped the landscape of science and medicine. The pace of progress over the last decade hasn't just been extraordinary, it's really been transformative. Technologies that to me as a scientist, once felt like distant possibilities, such as gene editing, mRNA therapies and platforms, and AI-powered diagnostics, are now transforming and shaping research, clinical trials, and treatment strategies. We have more targeted, individualized treatment plans and advanced therapies for cancer than ever before. Cystic fibrosis therapies have turned a once fatal disease now into a manageable condition. And across autoimmune disorders, rare diseases, infectious threats, and neurodegenerative diseases, where I focus, scientific advances have expanded our understanding of disease and opened up new possibilities for care. And through it all, we should all be very grateful that STAT has been there, documenting the science, elevating the voices behind it, and helping us make sense of the moments that have defined this era of innovation. With everyone gathered here at this summit, it's your ideas, your unwavering commitment that will shape the next decade of science and medicine. But as we look ahead, the question we should think of isn't just what's possible. It should also be what's necessary. What must we do to ensure that the next decade delivers not just on innovation, but true positive impact for patients? And among all these areas of medicine being reshaped by innovation, neuroscience stands out, not just for its complexity and the sheer scale of unmet medical need and possibility, but truly we view the brain still as the final frontier. We're still exploring how the brain functions, what causes serious neurological conditions. And we know that today 1 out of every 3 people will be diagnosed with a neurological disorder in their lifetime. Conditions like Alzheimer's disease, Parkinson's disease, sleep disorders, chronic pain are among the leading causes of disability and death worldwide. And this is only growing as our populations age. At Merck, we're focused on medical research aimed at investigating the complexities of these disorders. Our strategy in neuroscience is built on a foundation of deep understanding of human biology and a commitment to innovation. New imaging technologies, insights from human genetics, advances in cell biology, alongside some really amazing and transformative progress in the development of biomarkers, are helping us understand disease related in the changes in the brain and nervous system. These insights are enabling new therapies that have the potential now to truly modify disease, not just treat symptoms, something particularly impactful in the area of Alzheimer's disease where we're focused. And it's bringing us closer to our objective of really bringing the right therapeutic to the right patient at the right time. While our neuroscience pipeline continues to push boundaries, it's just one example of how our broader R&D strategy is evolving. Across our pipeline, we are delivering meaningful advances in medicine and vaccines by confronting some of the world's most challenging diseases with innovative science and differentiated solutions. We take pride in our role as stewards of scientifically rigorous and objective decision-making, guided by data, driven by purpose, and sustained by curiosity and optimism. Later today, my colleague, Dr. Aileen Pangan, will share some insights from our immunology research. It's another area where medicine and precision-centered innovation are really driving some change. It's a reminder that across all therapeutic areas, we're working towards the same goal: better outcomes for patients. And that's why this summit matters. It's a space for bold ideas, collaboration, and for asking the kinds of questions that will define the next decade. So over the next few days, I encourage you all to lean into the conversations you'll have. And most of all, think about what the next 10 years could look like for you. Because if the last decade has taught us anything, it's that the future of medicine is being written right now. There has never been a more exciting time to be working in the life sciences. And I've been doing this for about 25, 26 years, and I can tell you that without question, that is true. So thank you for all the work that you do and for being part of this journey with us. So let's get started.
Thank you for that, Matt. Here to kick off today's programming, please welcome to the stage Dr. Rebecca Ahrens-Niklas, Director of the Gene Therapy for Inherited Metabolic Disorders Frontier Program at Children's Hospital of Philadelphia, Dr. Kiran Musunuru, Barry J. Gertz Professor for Translational Research, Perelman School of Medicine at the University of Pennsylvania, Kyle Muldoon and Nicole Muldoon, the parents of baby KJ, and to moderate, Stat General Assignment Reporter Jason Mast.
5 weeks before KJ was supposed to be born, Nicole called me and said, we got to go to the hospital, like now. I don't know, 6 hours later, KJ was here. He was a beautiful little boy, you know.
Little preemie.
Yeah, in that moment, just seemed, everything was fine. He was born on Thursday, and then a doctor comes in Saturday morning and says, hey, listen, there are some things we saw that we didn't like. We were obviously very concerned, but we had no idea what we were in store for.
He was born at the adult hospital at the University of Pennsylvania, and a very astute doctor at Penn actually checked his blood ammonia level, which is a marker for some of the metabolic diseases that we think about, and it was very, very high.
We were sitting up there, and one of the doctors came to us and said, we think we know what's wrong. Your son is very sick, but the best place in the world for your child to be when he's very sick is next door. And I'll never forget that for the rest of my life.
We would do absolutely anything for our kids, so it wasn't like a woe is me. It's, okay, this happened to us. How are we going to support him?
So a urea cycle disorder is a type of metabolic disorder where you can't get rid or process parts of the protein that you take in your diet. This toxin, ammonia, builds up in your blood, and then eventually will build up in your brain. If that went on unchecked for a day to 2 days, the patient would be at very high risk of death.
You Google CPS1 deficiency and it's either fatality rate or liver transplant.
I bought a Jalen Hurts jersey, like when he was first born. It hit me like he's probably might not wear that jersey. You know what I mean? Like I just.
Thank you guys so much for being here. So that was about a year ago for that part of the video. Tell me a bit about what that was like for you. You get this diagnosis, and then they come to you and they say, we have this other therapy, this other thing that is out there. What was it like to digest that? What was it like to think through that? What were those moments like a year ago?
I mean, I think it's all, kind of a whirlwind, right? Like initially, he's born and he's really, really sick. They kind of find out rather quickly what they think to be wrong with him. And then, you just kind of as a parent, dive in and listen to everything that the doctors are telling you. And, people had asked us how did you learn all this stuff? And it's well, it's your kid's life. So you just kind of take your body, your mind just takes it all in. And so obviously we're hearing liver transplant is the only cure. And that, that's as he's kind of actively fighting for his life. So you're kind of collecting all this data and navigating, your way through every day. And my wife, Nicole, was still inpatient at Penn for a week or so after he was born. So as we get further through the process, he becomes stabilized, and then we make a decision okay, he's gonna stay inpatient until he gets a liver transplant. And then Dr. Rebecca walks in the room one day and kind of throws this crazy sci-fi futuristic technology at us. So yeah, I think it was a lot to digest at first. I personally had a very profound feeling when she came in. We had been praying, and we had a lot of people praying with us, and we were hoping for a good outcome in the situation. So Dr. Rebecca comes and she explains all this to us, and the thing that stood out to me the most was she never came off once she was trying to sell anything. Right, and then you realize as time goes on that this is a life's work type thing. This is a goal she's been, along with Dr. Kieran, working towards. And so at that point, when we realized this is legitimate, and that Dr. Rebecca was very, very clear about this could stop at any time for any reason. This could get shut down. This might not. She made sure we didn't get our hopes up at all, and I made sure to tell her that our hopes were on us. If this didn't work out, it was on us to make sure we navigated that as well. So I think at that point it was just kind of getting every ear, listening to every person, meeting with the different teams from the hospital, and kind of asking everybody what they thought. And at one point we had a meeting, and it was the liver team, the metabolic team, Dr. Rebecca, some other teams that were involved in KJ's treatment. And I said, if I brought him in with an ear infection, you guys would prescribe an antibiotic and we would go home. Somebody's got to tell me what we should do here. And I think after that meeting, we kind of walked out knowing that the gene editing was going to be the route that we took.
Yeah, and tell me a bit more about what exactly it was that you were offering or trying to do, because it's not as if there had ever been a gene editing treatment for his condition. It's not as if anyone had ever crafted a custom gene editing treatment for a newborn or for anyone before ever. What exactly were you guys doing? What was this treatment? How were you prepared? Yeah.
Yeah, so Kieran and I have been working for several years at this point, trying to figure out how you develop personalized editing therapies that we hope would translate into the clinic. But up to this point, it had entirely been done in a laboratory. And we had started with PKU, a more common rare disease, but our early work looked really promising. And as a metabolic physician or a rare disease physician that takes care of lots of babies with inherited metabolic diseases, I realized we needed to apply this to other disorders. And so we started working on urea cycle disorders because, as you could see from that video, these babies are really sick and they really need our help. And so we had been working through trying to streamline and optimize the time it took in the lab to develop a gene editing approach. And by the time KJ was born, we felt like we had the tools in place, and this is really work done by a lot of talented grad students, mentored by Kieran, and I help out a little, to try to move this work forward in a timely way. And we thought maybe we could actually try this with KJ. And
this is work you guys had to do because there's no, this is a very, very rare variant in a very, very rare disease.
Correct.
No company is working on this. You need to be able to make this on demand for a newborn who walks in, and you had been practicing this.
Correct.
And then KJ's born.
Yep.
You think you can make a gene editing treatment for it, and then you walk to the lab and you had it done in a second, or how did that?
I mean, the lab side of this, not to minimize it because it took a lot of work, and Kieran, feel free to jump in.
Yeah.
But I mean, that routine, we were able to very quickly get down to the point where we thought we had a solution in hand when he was 6 weeks old.
Yeah.
The gulf between an editing solution in the lab and a therapy that you're actually going to infuse into a child is massive. And I'll be honest, I don't think we fully appreciated how massive it was. I mean, we timed it out, but it's a lot. And so that was the more difficult part of all of this, really, obviously, is navigating that divide from the lab into a patient.
And if I could add to that, so as with most of these, I'm not going to call this a breakthrough. You can, render your own opinion as to whether it was a breakthrough. But all of these advances build on decades of work, right? So you needed to understand the human genome. That didn't really happen until the year 2000. And so if you think about what was happening in 2000, and I would never, in my wildest imagination, imagine 25 years past that this is where we are now. But you needed that information. You needed gene editing, right? And not just gene editing, but you needed something like CRISPR, which didn't emerge until 2013. And then even beyond that, you needed newer iterations of CRISPR. You needed the specific corrective editing technology, adenine base editing, and not to put her on the spot, but the inventor of adenine base editing is right there in the front row, Nicole Godoy, okay? So you need that. That was around 2017. And then you needed a delivery method. So that required the use of RNA. Some of the innovation behind that came from the University of Pennsylvania starting in 2010. You needed lipid nanoparticles, the soap bubbles that actually deliver the RNA cargo into the liver. So that emerged during the 2010s. You needed all of these pieces in place. But even beyond that, I think what we really needed was a proof of concept. And I don't think Becca and I would have felt at all comfortable going forward if my previous work as a cardiologist working on gene editing treatments to permanently turn off cholesterol genes in the liver and give a one-time therapy that would protect you for the rest of your life from heart disease. It was work I was doing in my academic lab starting in 2013 when CRISPR first came on the scene, developed over the next few years, then spun it out into a company called Verve Therapeutics that I co-founded with some of my colleagues. And in the work I did with Verve Therapeutics showed, hey, you can take this basic drug, lipid nanoparticles, RNA cargos, base editors, adenine base editors, and use that to turn off a cholesterol gene in the liver. And that is in clinical trials, and it is working dramatically well and safely. And so that was the very strong anchoring proof of concept that gave Becca and I the confidence to start moving ahead with the patients that Becca, as a physician scientist, as a pediatrician, takes care of, patients like KJ who have this devastating unmet medical need. So it was taking, in a way, a proven technology, not very proven, but proven enough that we felt comfortable that, hey, let's try to customize it for the situation.
So you have all that backstory, all those decades of science, these tools that let you change individual letters of DNA, these tools that let you get those tools into the liver, this proof of concept in humans, and then KJ's born, diagnosed with this very serious disease. Within 6 weeks, you have this thing you think works in the lab. To cut ahead, you get this consortium of researchers and companies around the world to come together to put this into something that can go into humans. And then in 6 months-ish, February, you have a drug ready to go into an IV, into KJ. Nicole, if I can ask, what was that day like for you?
I think all of the adults that were in the room were very aware of what was going to happen, what was at stake, very conscious of time and how much time was passing and how much time we had left on infusion. Whereas KJ, who's actually receiving the treatment, was so unfazed about everything. He slept most of the time. We wanted him asleep and comfortable, so at one point when he started to stir a little bit, I actually got in the hospital crib with him to keep him asleep while the infusion was happening. I would say for us, we were nervous, all of the emotions, but hopeful too.
Yeah.
KJ, it was just another day at CHOP for KJ. For everybody else in the room, he had no idea what we were experiencing. Anything to add?
Yeah, I was a nervous wreck. I talked the entire time. I cracked probably way too many jokes. But yeah, I think in all seriousness, we felt so comfortable with Dr. Rebecca and Dr. Kieran, like we trusted them so much that I think hopeful was probably, at least on my mind, like more in the forefront.
To add to that too, I think we built such a good relationship with Dr. Rebecca and Dr. Kieran that if we were unsure or had a question or, wanted to address anything, even up until that second, it was very open communication, open conversation, where we felt comfortable enough to ask questions or to question what was going to happen, what it would look like after. I want to know full picture, not just the tunnel vision of what we're experiencing, but what the adverse effects could be. And they had no issues answering those questions and making sure that, we had as much knowledge as possible and that was available to us to make sure that, everything would go smoothly.
So then he got 2 more doses, if I have correct. How is KJ doing today?
You can hear him in the back. If you see me laugh every once in a while, it's just because I hear him going, Dad, 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 Dad.
Why don't we actually just bring him back on stage? Yeah.
Ashley?
Everyone, this is baby KJ.
Hi. Hi.
So he's doing really well. He's meeting milestones. He's growing. His protein intake is, before he was very restricted, minimal to no protein. Now he has an allotment that continues to increase. He's meeting milestones. He's very active, if you can't tell.
He wants his microphone big.
Yeah, he
when he got it. Sorry.
Yeah. Who are all these people?
He also has the best temperament of all our children. He is incredibly happy. You would have no idea he lived in a hospital for 10 months. So we're just thrilled at where he's at.
Oh. Hi. See everyone?
You can tell he has 3 siblings and needs to say what he wants.
When did, so clearly he's doing very, very well. When did this seem, there was all this uncertainty for so long, when did this seem real to you? like, oh, this is working?
I mean, obviously, like, as we went through the 3 infusions and collected data and we get labs and tests and numbers, we saw some stuff that at least Dr. Rebecca told us was good. But I have this story. It was about three weeks ago. So we're from Philadelphia. We're huge Eagles fans. That might not sit well with this crowd, but sorry. Go Birds.
Go Birds.
When KJ was first born and taken to the ICU at CHOP, he was on dialysis, he was intubated. The Eagles preseason started, and I did not know if I was ever going to get to watch an Eagles game with my son. So we couldn't get it on the TV in the room at CHOP, so I took my phone and I sat next to his little crib that he had, and we watched it on my phone in like a very desperate moment, very like, hey, this might be all we get. So obviously time goes on and the story, you know, continues to write itself. But a couple Sundays ago, Eagles play the Rams. We're at home watching the game as a family, and KJ's sitting there with me, and the Rams are lining up to kick a field goal to win the game. And the Eagles blocked the field goal. And as they blocked the field goal, I kind of raised KJ up like we were there. And he was going to block the field goal. So the field goal gets blocked, the Eagles score. And I like get so excited, and he gets terrified. And he starts to cry, and you know, we're kind of, I'm like, oh my God, buddy, I'm so sorry, I'm so sorry. And then it hits me that, like, this is what we thought we would never get. And that's, you know, all the years of work that Dr. Kieran had talked about, all the hard work with everyone that you guys work with. And obviously, like, hope and perseverance from our family, like, got us to that point. And obviously, we're incredibly blessed and just so happy to have him here and have him squirming around on my lap.
And if you want to send baby KJ back as well, that's fine too. So rest, look out on Boston. That's cool too. Round of applause for baby KJ. So Becca, Kieran, that seems like a great testament to the power of modern gene editing and science and the work you and a bunch of other researchers have been doing for the last few years and for many, many years, as Kieran said. Are there more baby KJs coming? Are there more kids you're working on? What is the plan for the next kid who walks into CHOP with a serious disease?
Yeah, I mean, I think we're all very happy to see how well KJ's doing, and obviously I love this kid. But it's important to realize that this was a single patient expanded access experience, right? We need formal clinical trials to really evaluate robustly whether or not this actually works, whether it really shows efficacy, and whether it really is safe. And so our plan moving forward is to, as quickly as we possibly can, launch formal clinical trials. The other part of why we need clinical trials, and I think about this a lot as a rare disease physician that cares for a lot of kids with rare diseases, is that we need to be able to scale this. Yeah. And in our country, to be able to scale this, you have to have an approval so that that drug product can be reimbursed. And we actually can subsidize this to be able to help patients around the country. And so what Kieran and I are spending literally all of our time doing is preparing for a phase 1/2 trial that we hope to launch later this winter, and working with regulators who have been great partners in this to think about some creative and innovative ways to advance these therapies through the regulatory process. I don't know if you want to add more, Kieran.
Yeah, so when we started this, I think we very much wanted to do this in an equitable and fair way as possible, right? We didn't want a situation where somebody was lobbying for a particular patient to be treated, or a patient advocacy group was, saying, hey, you should really work on this. we felt that this opportunity should be available to anyone who presented themselves. And in our mind, that was anyone with a urea cycle disorder who happened to present at the Children's Hospital.
And to pause you there real quick, I mean, that has sort of been the case, or how these sort of custom therapies have gone over the last few years as they've become available. They're amazing, but they've been one-offs, generally speaking, for people who are either accepted into individual nonprofits or are able to raise the vast capital. And so you end up with a few patients as opposed to, say, thousands or millions or whatever it is.
Exactly. And so we felt there was something nice about the idea that a patient would be born within 48 hours, they'd be very, very sick, and then we'd be in a position to help, right? Spring into action and try to help them. It didn't matter who they were, where they came from, as long as they were within our catchment area at CHOP, we'd be in a position to try to help them. And when we were ready, which was last summer, KJ happened to be the first one up. He was born at exactly the right time, right place, at the Hospital of the University of Pennsylvania, and then quickly transferred to CHOP. And so we were able to do what we did, and it was a nice outcome, and I think we're all very pleased about that. But then what happened after that took us by surprise. So there was a great outpouring of positive feelings, but there was also some negative feelings as well. And Becca, maybe you can speak to that better than I can, because you actually are directly engaged.
It was a very exciting day. Obviously, people found hope in this in the rare disease community. But my patients, my colleagues' patients, so many other patients are saying, why not my child? Which I totally understand, right? I feel that way every time I try to allow a patient of mine or help facilitate a patient of mine joining a clinical trial that's full, right? It's like, why not my child at this time? There was a lot of things about CPS1 deficiency that made sense. The right place, the right time, but also the right disorder. CPS1 isn't amenable to AAV gene replacement therapy, for example, because the gene's just too big. So there's a lot of features as to why this made sense. But we need to scale this because we need to be able to help all of those other patients. We have gotten thousands and thousands of requests from individuals seeking help, understandably. A lot of them are from individuals with diseases that at this point in time the technology is just not ready for, so tons of neurodevelopmental disorders and things along those lines. And it's really hard, and we write back to every family, and speaking with them, you want to help everyone. But there is a population of patients that can be helped at this point in time. And so we're really trying to focus on trying to facilitate a rapid implementation of this approach for that population where the technology is there. We just need the infrastructure to be able to evaluate it and deliver it.
And that's sort of basically rare liver disorders caused by generally single base pair changes or single letter changes, because we have the machinery to make those changes, and you can get that machinery to the liver, which takes up lots of things that you put into the body. Yeah. How have those discussions gone so far with starting a trial? How are those plans? I mean, this is not a thing that, generally speaking, the FDA, if you want to start a trial, great, show us mouse, monkey, all this data, and have your drug product finalized and produced and not changed ever. You want to make basically custom treatments that you're changing constantly for individual patients, because there's 3 billion letters in our genomes and people have lots of different things that go wrong. How has the FDA been receptive to this? Are they on board? What's happening?
Yeah, I mean, so what we realized is overnight, we had thought our constituency were patients with urea cycle disorders who happened to be local to us at the Children's Hospital of Philadelphia. Overnight that changed, and we suddenly realized, wait a minute, our constituency is the entire world. And so we had to scale up. And so we've had very positive interactions with the FDA. They have shown the regulatory flexibility that we needed, right? So as you alluded, Jason, the traditional model is making that perfect optimal drug, spending years and years and years refining the technologies and literally pouring hundreds of millions of dollars moving towards approval. And that just isn't feasible for a variety of reasons in this kind of situation. We have to address a large amount of customization. We can't treat every single new entity we're giving to every new patient as its own drug, right? We really have to move towards a platform. And the other aspect of this that's been very interesting is that the FDA has made it clear to us that they don't actually want us to do more of these single patient expanded access INDs, these N of ones, these one-offs. And the patient community and really the physician community has told us they don't really want us to do that either. Because our constituencies in the entire world, the most equitable thing now is actually to have that open clinical trial where everyone, anywhere, can have a shot to be enrolled in the trial, right? So that's the first important aspect. The second important aspect is that only works if you can move to an approval quickly, efficiently, relatively inexpensively. If you do a string of one-offs, single patient expanded access INDs, compassionate use, whatever you want to term it, you're going to quickly run out of resources, and you're not going to actually help anyone beyond that, and you're not going to be within the structure of a formal clinical trial acquiring scientific knowledge. That's the point of a clinical trial, documenting everything about the efficacy and the safety, and getting that full understanding that the FDA, that other regulatory agencies will need to actually approve that therapy in the long run. Now, again, the traditional way of doing things, it spends years, you do many patients, you go through phase 1, 2, 3, et cetera. But we've come to a place, in no small part inspired by the example of KJ, where the FDA is showing us the flexibility. There's very productive interactions where we've come to an agreement, look, we want to have a platform, we want to cover 7 urea cycle disorders, we want to have a single, what we think of as an umbrella of umbrellas, clinical trial, in which we can recruit any patient anywhere who has one of 7 urea cycle disorders, so it's 7 different genes, any variant that's potentially amenable to adenine base editing, and be able to recruit them into a single trial, and with as few as 5 patients, be able to go from phase 1, 2, and then have a discussion with the FDA to go to phase 3, and then potentially with a few more patients, get to at least a conditional approval. And that is absolutely key, because that's when reimbursement becomes possible. Not to say that everything's great after that. There will be many more challenges after that. But if you can't get to that point, this whole thing falls apart, and we won't be able to help all the kids who need help.
Please.
Sure. Hi, Robert Green, Harvard Medical School. Congratulations on this amazing project and presentation. I just want to make the point that 2 weeks ago, the NIH announced a new initiative called Beacons, which will be sequencing newborn babies at large across the United States. So the potential to detect so many more babies who are at risk for rare conditions is now existing. And so your comment about platform and being able to manage this around the world equitably is kind of going to be on steroids now. This will reduce the diagnostic odyssey for many, many children who don't present immediately, but also redouble, I suppose, the intensity of interest in treatments for this.
As the physician that gets the calls from the newborn screening program, and a lot of my colleagues are working on the Beacon program, I'm so excited about this. I think it really is going to open up a world of possibilities. Right now, at a place like CHOP or a place like Boston Children's, these sick babies will get rapid genome sequencing to be able to make that diagnosis. So we had KJ's molecular diagnosis by the time he was 7 days old. But that's not true across the board. And so widespread newborn screening obviously is really important. So we're excited.
The one thing I should add, as a coda to all of this, is that we feel very strongly this needs to be scaled up. It needs to be accessible to everyone. That just doesn't mean, us at CHOP and bringing a lot of patients in and treating them, although that's probably the most efficient way to do it with respect to a small registrational clinical trial. But to really be able to deploy across the country, eventually across the world. And so in that spirit, interactions with the FDA tend to be very mysterious because it's usually companies that have proprietary interests and need to keep things confidential, trade secrets. You never see that information. And so Becca and I have committed to releasing all of our interactions with the FDA over the last few months related to the urea cycle disorders, related to another disease we're working on, PKU. It's going to be published on Halloween. No particular meaning behind that. It just happens to be October 31st in the American Journal of Human Genetics. There'll be a piece that we wrote that encapsulates, that summarizes all of our interactions. And some of the things that we have discovered the FDA is willing to work with us on is absolutely unbelievable, like things we wouldn't even imagine a couple years ago. That whole urea cycle disorder platform I mentioned, supported by a single rat toxicology study and no further animal work at all. Just absolutely astounding.
So not needing these monkey studies that are incredibly costly
or even rodent studies
something like this run into the millions of
all
dollars.
Cell-based work, because the technology lends itself, gene editing lends itself to that. So there'll be that perspective piece, and then linked to that will be the actual, partly redacted, but the actual documents, the briefing books.
From the FDA.
The written comments from the FDA. So we want to make that available to the community.
I've got 2 last things for y'all. One, can you say when you plan to start this trial? Or ask the FDA to start this trial?
This winter?
So this winter.
We are sprinting as fast as we possibly can.
Very exciting. And then for y'all, it sounds like by what Kieran and Lightbecker are saying is that, KJ has already had this massive impact, and his story has already had this massive impact on how a lot of doctors, regulators are thinking. When do you tell KJ about this role he had in the history of medicine?
Well, I think, we've kind of documented everything, pictures and kept articles and different things like that as this has all been going on. But I don't think it'll ever be too early. I think when he starts to ask questions, it'll definitely be a conversation where I'll have Dr. Becca and Dr. Kieran there with me to kind of navigate all of that. But we were able to age appropriately explain and then have our other 3 children describe or explain to others why KJ was inpatient for so long and why he was sick and all the different parts that went along with him living at the hospital. And we have a 14, 5, and 3-year-old, and they were able to do that really well. So I don't think it'll ever be that he's too young. I just think it'll be a conversation that I'll let him initiate first.
Thank you so much. Appreciate it.
So what'd you guys think? Okay, I think we have another great one here. It's always fun when you can get executives from some of the world's, or who've run some of the world's biggest drug companies of somewhat varying sizes on stage together. So I'm thrilled to welcome this group of leaders to talk about the next decade in the pharmaceutical industry. Please welcome to the stage John Maraganore, founding CEO of Alnylam Pharmaceuticals, Chris Viehbacher, CEO of Biogen, and Emma Walmsley, Chief Executive Officer at GSK. All right, so we did discuss what we were going to say on stage, but a lot's happened since then. But I'm curious, and I'm going to start with Emma. Emma, you're so far away.
Well, I didn't want to cozy up on the sofa too much, but
No, it's a bigger stage than I remembered.
Yeah.
But we've had a lot of news around pricing. In the U.S. and around politics and pricing and the threat of most favored nation and some companies starting to make deals. How do you think this continues to play out over the next, we said 10 years, but now we're probably talking next few weeks?
Well, I'm not going to make too many
I know you're not going to tell me about your last meeting with.
If we just step back, the US is the home of biopharma innovation in the world. Other countries are accelerating fast, and that needs to be watched. But I run a British company. 2% of our sales are in the UK. 52% of our sales are in the US. We're in 80 countries or so around the world, and this is the best country today to source and develop innovation, but it's also the best market if you're a patient for some to be able to access first in the world the most groundbreaking, the most exciting, game-changing innovation. And yeah, even though this is in many ways the world leader, we are still dealing with a healthcare system that is unfair and unsustainable. And therefore, and I think most of the industry, and Chris may add to this, would agree that it is incredibly important that we are able to bring this innovation at an affordable, accessible price, both for payers and, most importantly, for patients. So this is an access agenda. I think most people would agree that we would like to see all countries recognizing the value of innovation to bend the demand curve of healthcare far more. And I actually think it's a good thing to see industry engaging constructively with the administration. Of course, it's far more complex than headlines would suggest. And the critical thing is that whether it's individually as companies or as an industry, we find a way to bring more sustainably affordable innovation, both for the innovators and for patients, whilst allowing this country and the full ecosystem of academia and biotech and small, medium, and large pharma to continue to address the enormous amount of unmet need in the world. I mean, you've still got so many cancers without solutions. We still haven't, and I'm sure many of us are fighting for us to do that, cracked the huge burden on the system of neurodegeneration. Infectious diseases, even if it's a bit unfashionable at the moment, are still a leading cause of death. COPD is still a top ranking. So there is a lot more work to do, and I think we have to find a sustainable economic way to do that. Don't forget, drugs are 14% of the total cost of healthcare in America. Mm-hmm. And 90% of the volume of prescriptions in America are generics. They don't even exist if we don't innovate first. So, what we've got to do is find a way to see all countries, including this one, investing in keeping people out of hospital, keeping people well, and not seeing it just as, spending on the cost of sickness.
Chris, we've been talking about the gap, the industry in general, but you and I have been talking about the gap between European prices and US prices for longer than I actually care to remember. I remember having that conversation with you back at Sanofi. Do you think this actually shifts that? Are we in a place where Europe starts to pay more as the US pays less? Is that actually on the table now?
Well, I think we're certainly seeing some movement here. I think, just as Emma said, look, I think we as an industry would much prefer a system where there is more balance across countries in terms of shouldering the cost of providing an adequate return for the enormously risky and massively expensive activity of research and development. But it's been an intractable problem, as we would have discussed years ago, because essentially we are caught between governments. We have governments that independently determine what price they want to pay and essentially impose that upon us. And what people forget is it's not just a difference in price, say, between the US and other countries. There could be a massive price difference between, say, Germany and France. And so that's the system which we've lived, and it hasn't been balanced. We've had to figure out a way to make that work. And now we have a new administration that is doing a couple of things. First, it is calling out the fact that this isn't balanced and needs to change. And the president is now very much putting a sense of urgency in that in the discussions around trade and tariffs. And so suddenly, when you think about Europe as an example, they have lower prices, but the biggest source of exports for the European Union is biopharmaceuticals that are being exported to the US. And actually, I think the same is true of the UK and for Switzerland as well. So suddenly, we're in this now, well, there's the need to fund innovation, but with the tariffs and an encouragement of reshoring, there's a risk that a lot of jobs in manufacturing and research could be at risk. And so I think there is certainly a level of engagement with European leaders that we have not seen before. Now, whether that is actually going to lead to change, and then, in some ways, the difficulty, though, with all of this, and it's true for governments as it is for us, when we've been through this for so many years, unwinding this is not so easy. And that's where I think, actually, the Pfizer deal could be an opportunity for a way forward. MFN on its own is pretty catastrophic for the industry because we just can't unwind years of how we've had to deal with governments. But I think there are three objectives that people are looking for. The first objective is, on a go-forward basis, how do we get more of the balance? So as we launch new products, I think there is an expectation that we will have to have some, difficult decisions and discussions with other governments about the price. The second principle is that if we are successful at getting other countries to pay more, there should be a benefit for American patients, and that's part of that. But the third and most relevant to us is that this has to happen, this migration to a new approach has to be done in a way that does not put at risk the critical and life-saving investments that we are currently making research and development. Because if you had to go from A to B suddenly like that, again, that's catastrophic and we would lose so many new medicines. And that's where there is at least a promise. Now, there's a lot of complexity still. We're going to still need to see some PBM reform, for example. Yeah. That has dramatically changed the market in the last 15 years. And, as we know, none of this has fully played out. We have two companies that have seen agreements. Is that going to be enlarged or not?
We don't really know the full terms of those agreements at this point.
Pardon?
We don't really know the full terms of those agreements. No. So, John, John is founding a lot of companies. Just had a cool one, Coursera, that has, with Clive Meanwell in cardiovascular disease. As you're thinking about starting new companies, are you thinking about how pricing will be different in
very much
5-10 years, and how is it changing what decisions you're making?
Yeah, no, very much. Look, I think the evolution of the pricing story here that we're witnessing real time can in fact impact innovation quite significantly. Does it, for example, change the focus of innovation toward products that are not going to be Medicare-based products or in categories where government roles and price controls will be more impactful? I hope not, because I think that'd be a big mistake. But I do think it's on the mind of innovators in terms of where do you put the early research dollars and what are the indications you focus on, and where do you think about not only the unmet needs, which is always at the top of the list, but where can you get rewarded for all the risk capital that plays out. So I do think this can have an impact on the shape and framing and thinking around portfolio design and research investments, and that could be a mistake in some categories where you might have diseases that are ignored because of the headwinds that would be faced in the future on the pricing side. And that would be unfortunate, for sure.
Well, how does it change how we'd think of? We just saw a very interesting case on stage of a really amazing innovation that has a very difficult pricing issue already, N of 1. Does this shift things that are at that level of innovation where, you have experience with drugs like that? You all do with medicines that really have that kind of big impact. Does it change how you think about things where we get to bring kids on stage who wouldn't have lived before?
Well, go ahead, Emma.
I love it when I ask a question and everybody wants to.
Well, let me just jump in quickly on it, because I do think that challenge has existed well before the recent discussions and the recent changes in the pricing landscape, and just the cost of just the pricing model for curative therapies it has been challenging, to say the least. And in turn, the lack of commercial successes on curative therapies has, as we all witness, has impacted categories and modalities like gene therapy and gene editing from an investor interest perspective in return. So it is a cycle.
So I think probably like everybody in the room, when you see, and it's not just because I'm a mum, but when you see a baby like KJ come on stage and hear the story from his parents, it's hard not to be 100% on the side of every scientist who fights for that kind of extraordinary transformational treatment. So I think it's incredibly important that we protect these huge leaps forward in science, even if they're in a very personalized way of focus. That said, in my company, what we care about is the scale of our impact. And over the last 5 years, as we've doubled investment in R&D and hugely accelerated growth, we've reached 2 billion people around the world with our products through global health, through vaccination, and through scale disease solutions. And I think what's going to be important to keep, and that's, when you see what's happened in the obesity drug space, this is because there is a huge volume of impact on the demand curve for health systems when you can get step change results on something that impacts a large part of the population. So I actually think it's super important to look at diseases with very high burden. So steatotic liver disease, 5% of the world dealing with that. Hep B would be another one. Still, major burden in respiratory disease. And it's not because they're big diseases you can't have step changes. Look at HIV. We've gone from that being a death sentence to now very much within reach 6 monthly treatments and a horizon of getting to perhaps annual treatments for what becomes a chronic disease. So
maybe even cures for some people.
Yeah, we're working on that now. So I think, and you asked in 5 to 10 years, how do we think about early stage research? Honestly, I look at our industry and I think in 10 years' time, for sure, 5 years maybe to some levels, I think this is rightly going to be an industry that is completely disrupted because of technology. And in what we deliver, in the way we deliver it, in the way it's brought to patients, but also where in the world the great sources of innovation are going to come from. And I hope the US continues to lead that way. But in 10 years, when you consider the transformation of the Chinese biotech field, how will that look? I think the pricing question will look very different when we can do things faster for bigger step changes in outcomes because of what technology combined with biology can do over the decade ahead.
But, I think there's also opportunity here, Matt.
Exactly.
I've been in this. I was at GSK when we introduced the first medicine for AZT. And, something called managed care was coming along. And we had suddenly, generics coming along. And, for a long time, we were criticized for just having Me Too small molecules.
Yeah.
And, some of these changes have actually been an incentive to really double down on innovation. And that's certainly where I see it, and it's certainly what we're at Biogen now. We're at the complete opposite end of the scale, where Emma's company touches $2 billion. We touch less than 200,000. 0.1% of that. And
that's an amazing statistic given the scale in dollars of Biogen.
And this is a company that has been built on breakthrough medicine. And I think, having lived for 20 years in Europe, one of the things that you do see is that when you have a lot of affordability problems, it raises the bar on innovation. Yeah. Because if you really deliver a product that meets an unmet need in a way that no other product does, people will pay for that. And that's one of the reasons why today Biogen is not really that affected by the tariffs or the MFN. You can't be in rare diseases and start competing on price. It just that doesn't work. But where you can make a difference, as we just saw with this little boy, that is where you can go. And so we're actually doubling down on that. I think if you think about how many GLP-1s, has anybody ever looked? How many are out there in the marketplace? There's something like 45 of them, and then you got GLP-1s and GIPs. Do we really, as a society, need that? And should all of those people developing those really deserve a return on that investment? No, I don't think so. There are 7,000 rare diseases. We can treat about 700 or 800 of them. Take that money that you're putting behind another Me Too GLP-1 or another obesity drug, which is seemingly a market that is already well satisfied, and go do something worthwhile in society and invest in something that's more breakthrough.
But yes. Sure.
This is Monica Bertagnoli, the former director of the NIH.
Yeah, formerly Monica Bertagnoli, formerly at NIH, currently at the Harvard Kennedy School, and soon to be at the National Academy of Medicine.
Wonderful.
In between. It's fantastic. I benefited throughout my entire career by really wonderful collaborations between academia and the biomedical, industry, and certainly pharma. But I have a question for you. As we think about now really building a learning health system, we've had so much knowledge contributed by the pharmaceutical industry to our health. How do we develop the ways that we can partner with pharma to contribute knowledge that we need about care delivery, about global impact, about all the things that you've been talking about? Because it seems to me that we have the mechanisms to do that for the science, but we don't have the mechanisms and the approaches to do it for the broader public policy, where you're true collaborators rather than kind of the recipient of policy, it seems at times. So, interested in your thoughts on that.
Well, I'll just say one thing. First of all, I think a big part of that requires strengthening of public trust in the science that comes from industry and how it collaborates with the public sector and the academic community. There should be that trust there, and there's enormous amounts of value and knowledge that comes out of this industry that should be shared with the public from a public health perspective fundamentally, no doubt about it.
But I do think, if you think about it, Harvard University was founded before there was a federal government. MIT was founded long before the federal government got in the business of funding research and development. So I think there has certainly been a lot of dependence that has been created by the federal government, and that in some ways has created an opportunity for people to work in silos. But what we saw actually in the last financial crisis, if you went back to 2009 and 10, certainly I can speak for pharma and startup biotech, and we're in a period of that right now, where a lot of venture capital funding dries up, suddenly there is a lot of space for companies, for instance, to collaborate with startups, because we're getting approached by venture firms and who want to work together because some of their LPs are not so interested in investing in this space given all the macro risks and high interest rate environment that we have. And I think the same thing will be true. It has not always been easy to collaborate with universities for a whole host of reasons, but where there is a financial need, I think people will be drawn together. And I can certainly say, both Harvard and MIT are now got new initiatives. MIT's launched their HEALS initiative, and Alan Garber is now thinking about how do we as an entity at Harvard start to collaborate more with industry. And I think so, that's why I say whenever things are in motion, there is a lot of change, and people fundamentally don't like change because they like the predictability. But in that period, that is often where a lot of new opportunities can be created, and I think one of them is there.
Yeah, just to add, I fully agree with both comments. At GSK, we talk about our purpose being combining science, tech, and talent to get ahead of disease together. In this industry, more than any other industry, actually, collaboration is the solution. And we need to do that on a platform of trust, where industry, I didn't grow up in this industry, but it has astounded me, the difference between how it is seen by the general public because of all the Netflix movies and the reality of the scientists that work on the inside. And by the way, a legacy of behaviors in the industry that we have to transparently
I was going to say, there have been some significant fines paid by companies.
Zero issue on that. But if we are going to be able to move forward with everybody playing their part, then I think a trusted platform is critical. But we have two things in our hands now that could allow for some leaps forward. One is, exactly as been said, a moment when we are all seeing academia and biotech the world over, but the US particularly right now, coming towards industry, that we should lean into. But secondly, I think because you anchored the question also in the provision of healthcare. If we are able to open up the right kind of data collaborations for real-world evidence in systems of provision, this has been the forever promise of the NHS in the UK that we haven't completely unlocked yet. But a lot of work still to do. But if we can unlock that together, then we can be in a completely transformed world of both vast data of population health, right down to the individual for some of the more specific diseases we've been talked about. So I'm hopeful. Let's hope together that over this next decade we get better at unlocking data, including with the regulators in the US, and who knows what that will do to the model of clinical trials and the rest.
But Emma, in this country right now, we are also seeing a collapse in trust in the kind of science that you do. And
all science.
All science.
All science, unfortunately.
And disruption to a lot of, we were just talking about the NIH, but also the CDC, and some pressures on the FDA, which we're going to hear more about later today. You are one of the, or the company you run for a little longer, is one of the world's largest maker of vaccines. How are you thinking about vaccines in the US?
I take a long-term view. We take a long-term view. Vaccines is actually the smallest of our businesses, but it is, we are an important, and then as I said, in terms of our scale contribution, I think we vaccinate 40% of the world's children through our global health work as well. So
you also launched one of the most important vaccines of the past.
Several, yeah.
I'm thinking Shingrix.
Yeah, so Shingrix, the RSV vaccine, one of the biggest blockbuster launches in the last 20 years. I have to say we are cautious about the US right now, but the rest of the world vaccines is still doing very well. The reality is, tragically, history shows that when vaccination rates drop in any country in the world, infection rates rise. People die unnecessarily, often the under fives, and then vaccination rates come back up. And so I think it's very important to take a clear-sighted long-term view. We continue to invest in our long-term pipeline here. We walk towards the questions and the regulators. I was pleased. We did actually get to a new approval this year, so we have to separate the headlines from the reality, and a new further indication as well came through. But there is no doubt we are seeing a very politicized conversation, particularly on pediatric vaccines and COVID vaccines, which is not a business we're in. And we are seeing an erosion of public trust, which is dangerous. And we have to answer people's questions, not by shouting. As I've said before, we can't shout, trust us. We have to listen to what mothers are worried about, listen to what communities are worried about, and put the science forward, as we always have done, transparently, and answer people's questions. And I think over time we will see this settle.
So what about mRNA? It's an area,
what about it?
Well, it's an area you'd invested in.
Yeah.
It is also an area that here in the US has become highly politicized, and that the government has done wholesale research cuts. How does GSK think about that?
Well, we're still committed to mRNA. The timing of this conversation is helpful in the sense that we're not in the middle of massive phase 3 trials, so we are thoughtful about seeing how this settles down. But, I think our memories are a bit short. It wasn't long ago when the world was waiting urgently for solutions to, a pretty catastrophic global pandemic. And I don't think anyone can dispute that this is a technology that for respiratory infections can move the fastest in terms of addressing antigenic drift. And so it is definitely an important technology, as has been proven, for pandemics. And it's not like the world has seen the last pandemic. And obviously there are, and we're keeping an eye on this field too, there's very interesting science advancing in oncology as well with this platform. So I would say we take a long-term view, we stay invested, and we watch how the most current conversation plays out in this part of the world.
Okay, now we're going to do a bit of a lightning round. I want to go down the line with each of you, and I want in short answers, because we have about 3 minutes for each of you. What are you most worried about for the industry over the next 10 years? And what are you most optimistic about? John?
Well, I think in terms of worry, I do think we are seeing the emergence of a very new source of innovation in the world. And I actually think it's a good thing to have China be an innovator in the world, because I think we can use innovation from anywhere in the world. But I do think that we need to look at this as a Sputnik moment as a country. If we want to maintain our leadership in the US as the leading biomedical industry country, we need to change. We need to accelerate our abilities to get evidence faster. We need to think about the cost of our research and development activities and how to do it cheaper. We have to be competitive with what China's doing. I don't think we should erect barriers. I think we should find more carrots and ways of incenting the innovator to come forward. So I do think that is the biggest risk, but in it is also the biggest opportunity at the end of the day.
Can we do that and cut the NIH?
No, we can't. We need a strong NIH. We need to have the shining light country for medicine and science. And we cannot do those cuts simultaneously. Again, I also take the long-term view on this issue, but I do think that we need to address this current moment like we did with the space program and Sputnik. So that's the biggest risk, I would say. The biggest opportunity, I think, is what we saw on the stage a minute ago, this enormous innovation that's happening, never been greater. And there's so many prospects for science to ultimately cure and help so many patients out there at the end of the day. That's my
Chris?
2 cents.
The thing I'm most worried about are short-sighted people. Short-sighted people who are not investing in venture capital, short-sighted people who are not investing in our top universities, short-sighted people who are not investing in our next generation of scientists, short-sighted people who will pass policies or laws that would really cut our ability to continue to invest in innovation. What I'm most optimistic about is I think that there's still an awful lot of unmet need out there. And I think we're excited about our pipeline. I mean, lupus is a huge unmet need. Alzheimer's, we've started the battle. There's a number of new modalities coming along behind that. Rare diseases, we're decoding the immune system. I think things like gene therapy and cell therapy, they're still nascent. People forget how long it takes these things to bring. I mean, at last year's conference, we had Lotte Knudsen, who was talking about GLP-1s that she started working on in 1985. And, all of these technologies take time, but there's a huge unmet need. The science just gets better every year. And we have brilliant people in this industry. And I've been in this business a long time, seen a lot of cycles. This too shall pass, and I think we will come out the other side. But we are going to probably have to adapt to a set of new circumstances.
Emma, I'm going to flip this question on you, because we don't just want to hear about it in terms of the industry. We want to hear a little bit about what you're thinking about what you'll do after GSK.
That's cheeky. Listen, I am going to answer the first question. And I say this actually with a bit of a perspective after spending, it will be 16 years in the industry and 9 years as CEO of GSK when I stepped down at the end of the year. And I came into this industry through the consumer business, which I moved from China, by the way, where I was living and working, and I really agree that there is a helpful competitive threat there at the moment that everyone should be eyes wide open to. But I joined this industry and immediately fell in love with the phenomenal purpose, whilst recognizing it is about the hardest business to be in terms of needing courage and resilience and a risk appetite and a long-term view. But my goodness, it's worth it. And it matters. It matters to the mum sat on the stage, half an hour ago, to every family, to every community, to companies, to economies, to countries. And so it is worth fighting forward for the answer to every question in this industry, which is innovation. It's the biggest opportunity we have. And I think the decade ahead, and I applaud you all on for the successes, is going to see the most exciting and the most threatening disruption to that. But it's a moment to be full of optimism if the talent can harness the science and the technology to solve these big issues. Now, at GSK, we said the priority was R&D and innovation, and that's what we were going to spend our money on, and that's what we were going to put our focus on. And we've now got 15 assets of more than 2 billion potential revenue, all due to launch in the next few years. And that's how we went from being a just over 20 billion business to more than 30 now, with the prospects, and we've upgraded several times, of more than 40. So I'm super optimistic for this company as well as for this industry. What next? 17 years at L'Oreal around the world, 16 at GSK. I got another 15 of executive energy, so I'll let you know.
Oh, we all look forward to hearing that. We've got only about a minute left, and I have just one last question for Emma, which is just we were talking about the disruption at the FDA. There's a decision around leucovorin, drug usually used, that GSK has been brought in to apply based on the FDA's request. We know that some of the FDA staff are resistant to that decision. It's for folate processing disorder, which can cause some types of autism. There are some data. What is GSK's role in this moment with that kind of a request? I don't remember a lot of cases where FDA has asked a company to file data like this.
Yeah, I think on this one, it's not very much is the answer to the question. This is an administrative request. We have no commercial business with this asset. I think Chris, when he was working at GSK, took it off the market. I don't know. I won't say that's going to make me sound like I'm a lot younger than you. But anyway, many years ago. So we have no commercial interest. We have no scientific research. And we're not pursuing it. So it's just an administration at this stage. What we're focused on with the FDA is we've had 3 approvals this year. We want another 2, hopefully 1 this month in multiple myeloma, and a really exciting one for the world's first long-acting respiratory drug, hopefully in December. So that's what we're focused on. I think it's understandable that parents want some questions answered on some of these bigger things, but for us, that's not really something we're focused on.
So there's only one person who's allowed to ask a question now, Rick Burke.
The boss.
Well, hello.
No. Please, Rick.
I just have to ask, with the leucovorin, it just feels like meddling to the extent we've never seen in our lifetimes from the FDA in terms of their role in this. Is this extraordinary to you, or is this? It doesn't feel like just an administrative thing. It seems like
it's definitely just an administrative thing for us. Has it happened before? Not while I've been here.
And what is your take on what they're up to?
Nothing more than I've already said.
How about you guys? Any thoughts?
You want to comment, John?
I
see you shaking your head.
Well, I think I would hope that we can stay grounded in science. That's all. That's where I'll leave it. Yeah.
Do you feel it is?
Like I said, I would hope that we can stay grounded in science.
Happy birthday, by the way, for that.
Who told you that? How about for my birthday, can you answer the grounded science? When you say it's grounded in science, it's easy to read between the lines. You don't think it's grounded in science.
I just think it needs to stay grounded in science.
Thank you.
Would you say that you're worried that a lot of things are not being grounded in science right now, John?
It's all good.
All right. Well, thanks to all 3 of you for being here. Thanks for answering our questions. Don't forget your seat.
Oh, but never.
For this sponsored panel, please welcome to the stage Senior Vice President and United States General Manager at Genmab, Leslie Amendola, Vice Chair of Hospital Operations in the Department of Surgery at Memorial Sloan Kettering Cancer Center and Chair of the Foundation for Women's Cancer Board of Directors, Dr. Ginger J. Gardner, President and CEO of the Tiger Lily Foundation, Mayma Carmo, and to moderate, branded content editor at STAT, Jesse McQuarters.
Thank you.
Is she over here?
No.
So gynecologic cancers have long faced an imbalance in attention and resources, which leaves many patients without adequate options for prevention, diagnosis, and treatment. Leslie, I was really struck. We spoke this summer, and you told me, each year about 100,000 women in the U.S. are diagnosed with cancers of the reproductive organs, and roughly 30,000 die from them. Endometrial cancer, in particular, is rising at an unprecedented rate, with deaths increasing fastest among Black women. Today's conversation is about how we can bridge those gaps by rethinking the care continuum, ensuring innovation keeps pace with patient need, improving access and support, and strengthening collaboration among clinicians, advocates, policymakers, industry, and of course, patients. So, Dr. Gardner, just want to start with you. Can you give me a little bit of an overview from your perspective? What are the most urgent issues facing patients today in terms of both the treatment options and their day-to-day experience across prevention, diagnosis, and care?
Thank you, Jesse. We are here in October. This is Breast Cancer Awareness Month. And we truly feel in our heart the calling need for the 250,000 patients that will be diagnosed with a new breast cancer in this country this year. And yet I think it's also important that we recognize that women's cancer is actually a complex space. How many of us are aware that every 5 minutes in this country, another patient will be diagnosed with one of the 5 GYN cancers? That totals 115,000 new diagnoses a year and 1 million globally. So I'm a gynecologic oncologist, so conflict of interest for sure. I'll say it right straight out that GYN cancers are the here and now, and it is truly time that we shine a very bright light on the need to improve the future of women's cancers and include and prioritize the call to action for gynecologic cancers. I think to your question, what are the challenges and opportunities, is building collaboration and community to raise awareness and foster the future of research for women that face these diseases. So it's complex space, right? 5 GYN cancers. We've got, some of these are actually on the rise in this country. Ovarian cancer, uterine cancer, cervical cancer, vaginal vulvar cancer, and the rare tumors. That's a lot to think about. And from an awareness standpoint, we need to be thinking about the individual risk factors, tumor biology, symptom awareness, diagnostic tests, and treatment innovations that are patient-centric. Because when we do so collectively together, we can truly change the future for so many patients' lives. So I lead the Foundation for Women's Cancer, which is the foundation of our National Subspecialty Society, the Society of Gynecologic Oncology, the SGO. So we're a little bit of a rare breed as SGO members because this is our call to action, is gynecologic cancers. And as the Foundation for Women's Cancer, our goal is to unify and elevate all women's cancers with a special focus on gynecologic cancers. It's a really exciting time in our field. I would say it's a watershed moment in terms of some of the groundbreaking science. I mean, we're at a pivotal moment for gynecologic cancer treatment and discovery. And so I'm very excited about where we stand and what our opportunity holds with a community and collaboration together.
All right, and Mayma, you have built this community of cancer survivors.
Yes.
Tell me a little bit about their experiences. I know you've been so generous in sharing your own story of your cancer journey. What are some of the most significant barriers that women face, and what can we do to reduce them?
So I heard the words, "You had breast cancer" 20 years ago at 31 years old. And I almost didn't get to be here because I was told I was too young to have breast cancer, too young, and dismissed by doctors for almost a year. My tumor doubled in size, and I kept getting told, wait till you're 50, wait till you're 40 or 50. women are still hearing the same story today who are being dismissed by healthcare providers. One of my good friends, Shantae, found a tumor in her breast, a lump in her breast, and she's a nurse. Saw her doctor 3, 4, 5 times over the next 10 years, and her nipple began to bleed, and then was taken seriously. So the barriers to me are both systemic and societal. People don't think that women who look like me, who are younger, can get breast cancer. I'm still young, by the way, can get breast cancer. They think that, navigating cancer is hard. They think about things like fertility is an issue for me. Fertility, dating, lifestyle. And some people have issues with transportation, financial assistance. My husband left after my diagnosis, so I became a single mom after cancer and had to deal with bills and all the things getting to and fro. And also it's very scary. Imagine having told you have cancer, and you may lose your breasts, your fertility, your ovaries. Maybe you have to wear a colonoscopy bag, right? I mean, you have to think about all those things, not the cancer itself, but just the entire thing about just dealing with cancer. So people at Tiger Lily come to us for what I call, we are an anchor for a patient. When you're scared, what do you want? Someone to say, like, I got you, I'm gonna hold your hand, and you're safe. So they come to us, we have peer navigators that support them throughout their journey. They have community, they have literature, they have support. And having somebody you trust that can tell you, I've walked that walk before you. I get where you've been, I get you're scared. I get you don't want a bag on your hip for the rest of your life, or your vagina removed, or your anus, whatever the case might be. These are tough decisions to make, but I have been through those things, and I've survived, and you can too. So we built a community of what I called heart-centered. It's full of soul and compassion. People come to us for that place to breathe and become empowered. And when you're empowered, your whole life can change. When you can kind of control something about your diagnosis and journey, and own, maybe not your outcome, but how you will live your life at the time you're alive, that's really powerful, and we give them that at Tiger Lily Foundation.
Absolutely. And Leslie, you have this, sort of holistic view. This is very like ground level, and you're really like interacting with these women every single day. Leslie, tell me a little bit. What can industry do? How can industry facilitate great healthcare?
Yeah. First, thanks, Mema, for sharing your story. The numbers you heard both from Jesse as well as from Dr. Gardner are staggering. Over 100,000 women diagnosed in the United States every year, and 30,000, unfortunately, of them will lose their battle. That is unacceptable. And at the same time, there has been historically little investment in research. So it's an opportunity for all of us, and you heard from Dr. Gardner, to work together to change, primarily increasing research funding, so we can ensure that there's better treatments to patients across the journey, regardless of their stage of diagnosis. Building on that, we need to make sure there's access. Depending on where you live, and unfortunately your socioeconomic status sometimes determine that. So opening up access broadly, working together to raise awareness, ensuring if patients are detected early, and there's intervention early, there is widespread proof that outcomes are better. So a great opportunity once again, and then ultimately to wrap our support around that patient. So particularly at industry and at GenMab, we are very focused on gynecologic oncology patients. We already have a treatment that's available in cervical cancer that's in advanced stages, and an investigational therapy in ovarian and endometrial cancer. That is a critical step to make sure the research is advancing. But on top of that, the innovation doesn't matter unless there's access. So we are equally stressing and ensuring that can happen as well.
And trust.
And trust.
And trust.
Yes, for sure.
Absolutely, yeah. I'm really struck that it really is an entire patient journey. Everything in their life leads up to that. Leading up to the cancer diagnosis plays a role in how they react to what's happening to them, and generating that support system and that system that cares for them is so important. Sure. At GenMab, I know you really take the patient voice into account as you develop therapies and work. Tell me a little bit about that.
Yeah. So, really authentically listening to those lived experiences of patients is really important. And this is at the heart of everything that we do at GenMab, from research and development all the way through commercialization. Our patient advisory council is with us every single step of the way, really listening to them, learning and acting. And when we can't act based on insight, having that authentic, genuine experience. And I think to your point in your cue, which was great, we've built trust through those engagements of what we can, and oftentimes have found those better solutions as a result. So we were quite excited about what we're building and how we can really stay connected to the broader community.
Wonderful. And Dr. Gardner, I know you see, again, a huge part of the landscape and really work in research and also patient treatment. What can we do as a community, clinicians, industry advocates, policymakers, what can we do to move gynecologic cancer care forward? And what kind of breakthroughs on the horizon give you the most hope for improving outcomes for patients and their care partners?
Yeah, I think it's truly about collaboration and community. At the Foundation for Women's Cancer, we're making that a key priority. We were blanketing Freedom Plaza in Pennsylvania Avenue with the color purple, which is the color of all GYN cancers, so we need to come together as a community. That includes raising research funds for the pipeline of SGO members, gynecologic oncologists, and other investigators in furthering the research for these diseases. It includes raising awareness and access to patients. So as the Foundation for Women's Cancer, we provide free live patient education opportunities in multiple languages, in podcasts and online vehicles, because as new developments are happening in this space, which is really exciting right now, we need to be able to get that information from specialists in the field, from the SGO and the Foundation for Women's Cancer, to all communities. And what it's really groundbreaking, is that rather than it being one disease or another, we're seeing new classes of agents come to the field that have an impact across tumor types. So it's a complex set of diseases when you think about the 5 GYN cancers, each of which has multiple histologic subtypes, each one with its own underlying biology, and yet we're seeing underlying markers of activity that connect these diseases. And I'm talking about antibody-drug conjugates, I'm talking about PARP inhibitors, I'm talking about genetic drivers of disease, of which that field is continuing to expand. And so when we build unity through the Foundation for Women's Cancer and the SGO across our specialty as gynecologic oncologists, we can truly bring that center stage for the 1 million patients globally that are affected by gynecologic cancer in the coming year. And that's why I'm here today.
Right, and Mema, as you hear this, as you hear patients navigate the system, how does that resonate with you? What do you wish patients could tell their healthcare providers and industry and the world?
I would say that our voices matter, and for us, it's about building a living legacy. When I first was diagnosed, they told me I had on average 5 years for recurrence and I could die because triple-negative breast cancer had no targeted treatments. So when you're told that when you find your cancer and you push for diagnosis and you're blown off for a year, the tumor is now twice what it was before, then you have no treatment targeting a cancer subtype, you're told, you could die. What matters is how you live your life. And I think for me, getting patients to become what I call badass advocates and to know their risk factors, know how to act upon them, know what to do is really important. But I do think that, I made some notes, I'm having chemo brain. We must build for us and with us. Whatever is being built has to be with the patient. When I began the organization, Tiger Lily, I was a young woman, I was a single mom, I'm from West Africa, I'm an immigrant, and no one took me seriously because I was just a patient, just a little black girl. And I didn't know anything about oncology or partnerships or pipelines, but I had ideas that could transform patient-centered care. And with funding from people like GenMab and others, we now have built a national and global organization that's helping women all across the world navigate cancer care. So I say that every patient is a world in his or herself that could have ideas to transform healthcare. Listen to us, as you said, lean into us, learn from us, and let us lead. The things that my organization has done in the past 20 years has been transformational, but it's not just me. We get a patient who's scared. We say, come to us, I got you. I will take care of you. We're your anchor. We give them a navigator to help them navigate breast cancer treatment. Then they get support, peer support community, and they learn what they can do with their cancer diagnosis, and they become hopeful and empowered. So I think it's really important to build for and by us and to fund organizations like mine, because especially with the administration that we're in right now, we're losing funding, and this work needs money and time, and it needs the dollar bill as well. Community's everything, though, and our partnerships really matter to me, and that gives me hope every day.
And I always feel that as a patient, you know yourself. You truly know, when something is a little
Is it
off
wrong?
or- yeah.
I mean, when I found the tumor, I got a mammogram and it came back clean because they couldn't detect the lump in my dense breast body. So be open to policy change, systemic change, technology change, because I had to push all that time for a biopsy against all the odds. Then I had cancer. If I hadn't pushed, I would have been dead. I wouldn't be here today. And so it's really important that people hear patients for who they are as individual. We're all different, we're unique. Hear their voice, listen to their bodies, and take them seriously, because we're each a unique person with a story to share and impact to make in this world. I always think, what if I had died? I think of the work that I've done, people that I've been able to help. I've created policy change, clinical trial change. We're in the communities across the country that are high disparity communities. But what if I was dead? I wouldn't have made the impact. And my daughter, who's calling me now, when I told her I'm on a panel, she's now 22 years old, and I get to see her be a young woman. I get to see her for her to live, right? And other people should have that privilege of being with their families for that length of time and more. That's my gift to other people, to give them that time and that family and the joys of learning how to ride a bike and tie your shoes and meet their first boyfriend and go to college and graduate and go to their wedding, and that's what we all want for our patients. Right?
And see their grandkids, and you see their grandkids, and
Yes, exactly.
I'd just like to follow up on that. Such a great point, Mema. And for gynecologic cancer, some of the times the symptoms masquerade as a general benign gynecologic condition. So again, it's that patient education, patient awareness. So to raise a bright light about these diseases with the innovation that we are seeing, surgical innovation, technology, AI is certainly the buzzword in almost every other field, and it's true here as well. Immunotherapy. I mean, there are so many aspects of elements that we're looking at across diseases that apply to the 5 GYN cancers. So breaking down those barriers like Mema's talking about, increasing that access and awareness about symptoms, genetics, and early recognition, and available new treatment options is a game changer. So congrats to you, Mema.
Oh, thank you. And people that look like you as well. My best friend, Kat, who passed away, Kat was a yogi, and she had cervical cancer. And she felt ashamed because they asked her, the first question was, what did you do? Were you promised? Like, were you having sex? Were you having unprotected sex? Like, they thought they made it seem like it was her fault. So she hid the problem. She would just bleed for months and years and just think it was just cleansing the things she'd done. We all have been young and done things, but it was never her fault. And so when she came with a choice of either I take everything out and live, or I have a life without a baby or my body parts. Even I have scars, but she would have had worse scars. She made a life-altering choice that she wanted to change at some point, but she couldn't change it. She passed away. And so beyond the things we talked about, there is the importance of seeing somebody who looks like you, who says, I have the scars, I have a bag, I have these things, but you know what? I'm alive, living my best life as well.
Yeah. Thank you for sharing that. Thank you.
Yeah. And Dr. Gardner, in that clinical situation, as you are talking with patients, how can you see the best practices for supporting them through that journey?
I spend more time in my clinic talking to my patient than thinking about the disease itself. I mean, after a few years of doing this work, I know these diseases, and I'm interacting on what's happening in the field and working. My first research grant was sponsored by the Foundation for Women's Cancer back in the day. But I spend my time listening to my patients because each of their journeys is unique to them and special to them. Who do they have to rely on, or who do they not have in their life? What are their concerns and what are their hopes? Because when cancer shows up on the scene, we need to understand the context of what's going on. So from the Foundation for Women's Cancer, our live events, we call them forum. It's not a didactic, there's a lot of content, believe me, but it's a conversation because we need to hear from our patients so that we're designing the right research, designing the next trial, designing the best solution that meets our patient where they are. And so building collaboration community on behalf of Society of Gynecologic Oncology and the Foundation for Women's Cancer, that's really what we're here to serve, and to unify the community around these diseases and the patients that need this help so much.
All right, Mema, closing message. Let you
Oh,
blast
wow,
that.
in 45 seconds. I'm just thankful to be alive today, to be honest, and to serve people that I care about. I was once a younger, scared girl, and now I'm a national leader. That's a privilege I have that other people don't have. So my gift is to use my time, my breath, and my heartbeats to give to other women, to be on these panels, have these partners, meet women who are serving others and make a difference in the world, and transform cancer care as I know it by asking questions like, why not? Why can't we do that? What about this? And just making epic shit happen.
All right, thank you very much.
Thank you.
Pleasure.
Yeah.
Thank you.
What a great session. Next, here to discuss recent changes, it's a little euphemistic, at the CDC. Please welcome to the stage Dr. Daniel Jernigan, former director of the National Center for Emerging and Zoonotic Infectious Diseases at the CDC, and Dr. Rochelle Walensky, professor of medicine at Harvard Medical School and the 19th director of the Centers for Disease Control and Prevention. And to moderate, Stat Washington correspondent Daniel Payne. I have a mic. Look for me if you have questions.
Thank you both so much for being here.
Sure. Thanks for having us.
It seems like we can't go a day without some big CDC headline, and certainly we've had some yesterday, over the weekend. Talk to me about, when you're talking with your colleagues in the last few days, what have you heard about the most recent round of layoffs, and what do you make of it?
I can start. Yes, I was at CDC for 31 years. And resigned from the director of the National Center for Emerging and Zoonotic Infectious Diseases. And so do keep in touch with a number of the folks in that infectious disease center. And so I think one way to describe it is just chaos. You've got folks that are trying to do their job. They have been working in deep science for a long time. And suddenly you get an email that says that your services are no longer needed, or you're non-essential. And then the next day you get another email that says, never mind, we made a mistake. So about 1,300 total people received emails, about 700 received never mind emails, but there's still 600 that are there. And what we can see so far is that the people that are not there, the ones that have continued to be riffed or have a reduction in force, are those that do the real work that makes the science happen. So they may have reversed it so that scientists could come back, but the science can't really happen if you don't have the infrastructure to do it, the grants, the communications, the infrastructure, the data analysis and things like that. So right now, while people were able to come back, the science that needs to happen in the engagement with state health departments is still at risk.
And maybe I'll just add, a lot of the information that I'm getting from news and from friends, but we can have a conversation about what this is going to ultimately do to the infrastructure of public health. I think that's critically important. But as you think about the uncertainty of trying to manage your work life, when one day you have a job, one day you don't, one day you have email access, one day you don't, and how you're supposed to sort of function and do the good work of public health in that context, that Dan and I are trying to get our information from crowdsourcing. We don't even know whether this job exists or doesn't exist, whether this person is in it or not in it. And that is actually not a way to sort of have an effective public health system.
Absolutely.
Yeah, and I think those cuts were on top of what already had been cut, so probably around 3,000 total people.
And I want to ask too, another major moment for the CDC this year was, of course, the shooting. Dr. Jernigan, you were in the building. And you wrote to Congress in a letter, you said, over the next 6 hours, while we waited for police to clear the building and give the all clear, I visited staff throughout. In talking to them, many were shaken, but most were resilient. What were those conversations like?
Yeah, so that was in a note that I had sent as a part of the hearing to Senators Cassidy and Sanders, just describing what happened there. It's interesting because the coverage of it was not very complete. I think most people don't realize that 500 bullets were actually shot at CDC. 180 of them actually hit the building, and several of them actually went through the building into the areas where people would have been. And essentially every one of those bullets had somebody's name on it. It wasn't just a random act. This person was really trying to do something. And so in talking with folks as we were going through the building, we were all stuck there for about 6 to 8 hours. Talking with them and hearing about the experience, most were really pretty resilient. A number of folks later on, I think we recognized were having some trouble. We have a number of vets that work at CDC Veterans. And they communicated how the sound of that repetitive shooting really brought back memories. One person that worked for me indicated how she was having some post-traumatic stress disorder because of her memories of what was happening in a helicopter many years ago. So I think people were definitely affected by it. But in general, these are folks that are willing to work in biosafety level 4 labs with those whole moon suits on, working with dangerous viruses. They're willing to sleep in the forest in a tent during Ebola outbreaks. So they're able to make it through. But for them, when they found out that this was actually fomented by somebody who was getting rhetoric that was anti-vaccine and anti-CDC, they were not prepared for that. It just says that if I go to work, it's less safe than me going into the field in the Democratic Republic of Congo.
Absolutely. And Dr.
Yeah, I mean, maybe I'll just add on to that and say that when I arrived at CDC in January of 2020, I had been told that the security team had previously said to the agency, remove all of your parking stickers from your car for fear of vandalism. So this is an agency that has been feeling a lot of trauma for the last 4 years. In 2023, there was a mass shooting in Atlanta, and the only fatality was a CDC employee, Amy St. Pierre, who had been in harm's way. So the agency itself has felt a lot of this trauma before this event. Dan, I don't know that you mentioned that there was a daycare that those bullets had to pass, a staff daycare that people were actually picking up their children in. So I do think that all of that, those layers, not just this one, but on the heels of all these others, really does have an impact when, quite honestly, the work of the subject matter experts and people at the CDC has for large part been completely quiet, protecting people quietly without heroism, without wanting credit, and just kind of doing the good work of public health.
And one hero that day was Officer Rose, who lost his life protecting the agency by trying to stop the shooter.
Of course. And given all the headlines, not only the shooting and fears of security concerns and layoffs and changes to how science is done and who's advising the CDC, do you fear or do you think that the days of the agency being the leader for public health in the world and in the United States are over?
One of my greatest times while I was CDC director was traveling the world, because I think one of the things you learn as you travel the world, CDC has offices in 60 different countries, and you realize how revered that work in science is around the world. We are considered the gold standard. Many countries come to us and say, how do we set up something like the CDC? So this branding problem, I think we have, is mostly a domestic problem, candidly. I do think that there are still extraordinary subject matter experts who are at the CDC who will soon one day be able to rebuild into a better, I would say, it'll be different. It has to be different because it has necessarily been cut and traumatized, right? So I do believe that a rebuild is possible. That said, I will also say I don't think we know where the nadir is. And so we have seen government agencies completely washed away. And so we have to really understand where the nadir is and what we need to build back from.
And I'll come back to you, Dr. Jeremy. I want to hear your thoughts on this. But I want to follow up on this because you actually told one of our reporters last month, I think, the vaccine information on the CDC is not necessarily that of the subject matter experts or the CDC. There's a lot of other great information on the CDC website that I don't believe has been tainted yet. How do you make sense of which is which? And do you think that that is going to catch up with our global reputation?
Yeah, I mean, when I started at CDC in January of 2021, among the things that I asked my principal deputy then, Ann Schuckett, to do was to comb the CDC website for things that had not been vetted by our own subject matter experts. And many things needed to be taken down, several things needed to be taken down because they were placed under a CDC banner without the subject matter experts. I think it's getting harder and harder to tell. Again, those things are happening, which ones they are. I will say that I've been a consumer of the CDC website since I did my infectious disease fellowship. So, and I do think that it is probably the case, I don't know for sure, I'm not there, that there's still extraordinary amounts of very helpful information for practitioners in all sorts of disciplines on the CDC website that hasn't been touched. I think the things that are making the news that we know are real challenges, especially in the vaccine space, are going to be increasingly harder to trust. And I don't know how one deciphers sort of where that line when that line is drawn or where that line is drawn. You know, so I think it's going to be harder and harder.
Yeah, I think just in terms of the global issue, the 80 years at CDC and so many other places that are similar to CDC, they named CDC. And they do that because they trust the science, they trust the processes that we do, they follow the ACIP recommendations, they follow a whole lot that we do. If you take away the scientific process that's being used at CDC, if you're not using evidence to make decisions, then you're going to start eroding that trust. So other countries will not have that trust, but equally, state health departments in the United States are dependent on CDC for funding, but also the recommendations that we make, they're able to then say, well, this is what CDC says, this is what we'll do. If they can't trust what's on the CDC website, then we're having a significant impact on what happens at the local level where people actually engage with public health. And that is something that we don't want to see, but I'm afraid that we're heading in that direction now.
And I used to say, like, we knew we were starting to regain trust in the agency when we could report good science, we could report on an investigation, and the word CDC didn't really matter. What happened is it had the brand of the CDC logo. CDC wasn't the news. We were reporting out the science, and that's really what we want. And increasingly, since, you know, the layoffs and rifts and all sorts of things, CDC has become the news again, and that is not a place CDC generally works, operates, or should be. We should not be the news. We should just be reporting the outbreaks in scientific investigation so the public can know and trust them.
When you're talking to just ordinary members of the public, would you tell them that they should or can trust the CDC today?
I'll try that one.
How about it?
In general, there's a lot of information on the web, and you tried to decrease some of that because there was so much.
Yeah.
But there is a lot. A lot of that is scientifically based, it's evidence-driven, et cetera. Foodborne outbreaks, things like that. I think the area that I'm most concerned about is in vaccines, vaccine recommendations, and vaccine safety information. And so that's one where I think if you go to that website, I don't know if I could trust it or not, because you can't really get a sense about, is this something that is an agenda-driven ideology, or is this something that was achieved through working groups and a process for evidence to recommendations? So if you can't do that, then what can you do? And I think there are some outside groups that I think are going to be absolutely necessary now. It's almost a good housekeeping seal of approval on those recommendations. And so some of those you can talk with about the Vaccine Integrity Project, but the professional societies, AAP, AMA, IDSA, AAFP, those are groups that really need to step up and start making some assessments of those things, but also groups that are getting to the data, evaluating the actual evidence, and then telling us, we've looked at it, normally the CDC would be doing that, but here is the evidence evaluation, this is where we think about these recommendations.
And I think that this is sort of a moving target. So among the things Dan referred to was, when I started at CDC, I think we had 240,000 web pages. And so it was very hard to sort of pull that all together and make sure it was in a coherent space. But, you know, as Dan said, vaccine information may be a challenge. But if I were traveling to Chad and wanted to know what travel vaccines to get or what prophylaxis, that I don't imagine has yet been touched. I don't know for sure. If I wanted to know maternal mortality rates across the country, that space probably hasn't been touched. And yet, I do know that there have been layoffs around the spaces of people doing those surveys. So how long will that integrity be in place? So I think that this is going to be a moving target.
We've talked a lot about vaccines for obvious reasons, but I wonder, as people who understand the agency at a really deep level, what's something we're not talking about that people should be paying attention to that could be changing?
I think that the vaccines have sort of been the oxygen sucker of the news, if you will. There are a lot of places, public health works well when you don't know that it's working. And we are starting to see areas where, for example, here in Massachusetts 2 years ago, we had 2 cases of a very resistant gonorrhea. Gonorrhea rates are on the rise. I think they've fallen slightly this year, but we have the highest rates of gonorrhea that we've had probably in 15 years in this country, similar with syphilis. And in these rifts that happened in April, the resistance testing lab for sexually transmitted infections was shut down, as was the disease investigation area of sexually transmitted infections. Syphilis is another one. There have already been 3 fatal cases of neonatal syphilis this year in New York City alone. Syphilis cases are really high right now in this country. And again, we have lost some of this infrastructure to deal with sexually transmitted infections. HIV, you've probably heard in some of the news that our HIV testing resources, our HIV surveillance resources have been cut. There is currently an HIV outbreak that is being investigated in Maine. They've asked for CDC's help, and CDC doesn't have the bandwidth to help. So I think that we are going to increasingly see, from an eroding of lots of different layers of infrastructure, these things pop up in many, many different spaces.
Yeah, I think just in terms of what other things other than vaccines, I think 3 in particular: communications, awareness, and response. And so just from a communication standpoint, as you see from the most recent RIFs and from previous reductions in force, the communications staff have been removed. And there was some sense that, well, AI can take care of that kind of thing, or we'll use the communications groups at HHS. But you really need to have very quick communications. So if you have a foodborne outbreak and you want people to know, don't eat the lettuce, you need to be able to say that quickly and with recommendations that people can follow, because people have to adhere to something if they're going to prevent the disease. However, if you're not saying that, and you have to go through HHS and wait 2 weeks to get a response that says, yes, it's okay to say it this way, that is not good public health, and people are not better for it. From an awareness standpoint, CDC is bread and butter is surveillance or monitoring of infectious diseases. And right now, we know that for flu viruses are collected from all over the globe. I was in the flu division for 15 years. We worked with WHO and others to get those viruses to come to CDC, characterize them, and figure out which ones do we put in the vaccine to protect people. Right now, 60% less of those viruses are coming into CDC because people are just not participating. They don't know exactly what's going to be done with the information. And so we have less visibility on what's happening with flu, but that same approach is happening with other pathogens globally and in the United States. I think as funding goes down, states will not be able to do the kind of work they needed. The last thing is just on readiness, from a pandemic readiness standpoint. And working in flu, that was something that we always cared about a lot. And when we have a big response for whatever the complex infectious disease issue is, we need people that can work on that response. And so those are deployers. Those are people that come from all over the agency to work on that particular problem. We've gotten rid of chronic diseases. We've gotten rid of birth defects. So we don't have the people that can respond and fill in for those big responses. We don't have the birth defects people. So there's a thing called Orapuche, which is a new virus emerging, does the same thing as Zika with microcephaly. If that takes off, we don't have the people that can provide that expertise like we did before. And frankly, everything is connected. And if you start cutting parts of the overall system, we're just not prepared to respond as quickly or as the way we need in order to have the most way of preventing disease.
Daniel, we have a couple audience questions.
Please.
Hi, I just want to thank you both for your thoughtful answers and for joining us today. I just want to follow up, if I may, on Daniel's question about would you trust the CDC website? I mean, you gave really thoughtful, detailed answers, but my takeaway was, well, don't really trust them on the vaccines, but perhaps on the international travel. There's a lot on that site, so maybe go through, perhaps certain things. But if you're just untrained in health and medicine, what I'm hearing is sort of a lot of sort of parsing, but to the average person, like, isn't it a danger to even look at that? I mean, what I'm hearing from you, maybe it's okay, but they have to know, like, can you give us more direct advice for a person on the street who doesn't know anything about what to believe or not? Should they be taking advice from the CDC site or not?
I think just an answer that we often give is talk to your doctor. And so if your doctor then goes to the CDC website, it's kind of the same problem, but there may be a way that doctor can actually make some better sense that the individual cannot make. In addition, if we do have surrogates, if we have others that are providing that information, if physicians have access to that, then I think that would help. So we have to make sure that the physicians that are caring for those patients can answer the questions that those patients may have. But for things that aren't urgent, you can go to the website, and how do you know? There is kind of a move now to say that people should be making up their own minds, that they should be able to do their own research because all that information is available now. But I don't think that that's a responsible way for moving forward. I think we have to have good information that's vetted, that people can trust. And if we can't do that now, we have to find some alternative.
There's another question over here.
Oh, yeah.
The real challenge in my mind is CDC is such a wealth of information. I can say for vaccines, we can go to the other places and we can triangulate and say, you should go to your hospital website or go to the American Academy of Pediatrics or AMA or whatever it is. But if I said to you, well, what vaccines are you going to use to go to Chad? Those are not, obviously, you don't have another source for that. So I think that that's actually the real challenge to say, does your local hospital tell you what travel vaccines to take? And so I think that becomes a real challenge because there's such a wealth of information that is captured on the CDC website that may not be captured uniquely in other places. So I do think consumers are going to have to do a little bit more careful vetting of where they're getting that information and trying to understand what is there. And I think that is, in fact, the real challenge of the moment, if not the intentional challenge of the moment.
Thank you for letting me ask a question. My name is Jonathan Rosen. I'm a family physician. So it seems over the past half decade, there's been a fairly nefarious attempt to undermine the credibility of the CDC and to use that lack of credibility to attack it. And I just wonder what your organizations that you personally are involved in, the general professional organizations, ought to be doing to counteract that in a very organized way, which doesn't seem to be the case. Right now there's a Stand Up for Science rally at I think at the NIH on Friday. We have our grassroots movement of Indivisible going on. But our professional organizations seem lacking in the response.
Maybe I'll take that and talk about the Vaccine Integrity Project. So back in April and May, I started having conversations with Mike Osterholm at University of Minnesota and the Center for Infectious Disease Research and Policy, CIDRAP. And we sort of said, how will we know when we know we are entering an administration that may or may not be friendly to vaccines? How will we know when something needs to be stood up? And so we started having conversations in a very nonpartisan way with people across the aisle, listening to state and local health officials and insurance companies and the medical associations and the hospitals to really just get a sense of what that would look like and what the triggers would be. And among those triggers was, in fact, the firing of all of the ACIP uniformly, which had never happened before. Among the things that we have done, and work is currently underway, is to try and, usually we have an advisory committee on immunization practices that reviews all of the literature for what is happening in respiratory viruses so that they can make recommendations for the fall season. We did not see that comprehensive review of the literature in this year's ACIP meeting. In fact, there wasn't really a big discussion about even the COVID vaccines in the ACIP meeting with a presentation of data. So a team of scientists has gotten together to review the literature from the last ACIP meeting that flu, RSV, and influenza had been reviewed. That included reviewing 17,000 abstracts and critically reviewing 1,400 papers and then extracting data from about 500 papers. If you want to see those 500 papers, they're all on the Vaccine Integrity Project website. You can sort of say, what are the papers in the last year and a half that looked at COVID and pregnant women, or RSV and PEDS? And it will produce all of on that website, it will produce all of the PubMed citations that have been published in the last year and a half since that was reviewed. That is transparency. That is, here are the data that are out there. Here are the published data. They're for all of you to see. We are working on synthesizing those data for a manuscript, so you will have them, so you can see them in synthesized form. But we wanted to sort of say, here's the science. Part of the reason to do this was because we knew the societies were going to have to step up to the plate to produce these recommendations. And as they were doing so, we thought maybe everybody is not going to have the bandwidth to do this massive review, each of them all at once. And so we'll help with providing this massive review for state and local health officials, for societies, for hospitals as they're thinking about these policies. And that was really our intent, and that work is underway. We had a webinar a couple weeks ago to sort of present the early data from that. That webinar was moderated by Caitlin Genelita and of your local epidemiologist, and Mark Siegel of Fox News, and Eric Rubin of the New England Journal of Medicine. And we really wanted to transparently show all the data. And I think that is among the efforts that are underway. There are many others that I'm less involved with, but that's the intent.
Can I?
One more quick question. I just want to give it, it's fast, of course.
Hi, I'm Tim Leshin with the Association of Schools and Programs of Public Health. I kind of disagree with the earlier comment, but I wanted to ask about the fact that it seems like all the staff for the Center for Forecasting and Data Analytics are gone, and the whole CDC DC office. And can you talk about why that is so bad for our country?
So maybe I'll start with the DC office and say CDC is appropriated and authorized from works from Congress and the Senate, right? So we can't do our work without that fluid communication. CDC is also in Atlanta, right? So we need a presence in Washington to have those communications. So the loss of the CDC office, I think, is going to be really, the agency, I think, will really feel that. We need to be able to, or CDC needs to be able to communicate a new outbreak to there's an outbreak in Maine of HIV. Let's have our experts talk to the congressional staff in HIV in Maine so that we can sort of let folks know what's happening on the ground. All that sort of fluid communication happens through our CDC Washington office. A member wants a briefing on A, B, or C. That's how that all happens. In terms of Center for Forecasting and Analytics, that one is tough. That's a center that I started when I was at CDC to really sort of understand and use new and novel methods to understand and project and look at surveillance and happening now and what can we think about for the future. Extraordinary experts that were hired to do that work. And as we think about the future of surveillance, the future of predicting outbreaks, merging data from open table and TSA and surveillance to sort of say, where's the next outbreak going to be? That was the new novelty that our society today demands, and we will now lose.
Can I circle? We have just a little bit of time left, but I want to try to squeeze in two more questions quickly, which one is, a lot has been made of the officials that we see today at HHS sort of rose in prominence during the pandemic because they were COVID contrarians, a lot of people have called them. If the public health mainstream, whatever that is, comes back to power in the coming years, should they take a different approach in the future? Is what happened through the pandemic somewhat responsible for where we are today?
Yeah, if you go back to 1918 with the flu pandemic, again, my background in flu, there was a lot of disruption that happened after that. A lot of people really did not agree with it. There were effects on the communities. It's all written down. There's no way we can expect to have a significant infectious disease outbreak and go through it unscathed. We at CDC we are responsible for making all the decisions and the recommendations, et cetera. We know that when we do that, there's risk of failure, there's risk of us taking on negative aspects because of that. But that's our job. And so the fact that there were things that didn't work, we figure out how to fix those things. So there are things that we do that are extremely important. We have to continue doing them despite whether or not we're going to have a bad outcome. I do think going forward, though, we're going to have to figure out ways to protect the scientific process at CDC. We cannot have a choose your own adventure approach to the ACIP. We have to have evidence to recommendation processes, and we should demand that any recommendation that comes out actually they demonstrate how it is and what scientific process is being used to do that. So there are some things that we'll have to do better, but it will take a while, I think, because people are leaving, some of the funds will be gone, and I think the trust, which they were concerned about before, is going to be in a very worse position later.
Yeah, I think if you dialed back the clock to 2019 and you asked the American public what CDC is, most people would know, right? And CDC sort of became the spotlight during the pandemic, and it became a source for people to go to. Now, CDC subject matter experts and CDC guidance had not necessarily ever been written for the American public previously, because the American public were not the consumers of it. They were the public health officials and maybe superintendents of schools or other things, right? So now all of a sudden you have, during the pandemic, everybody being a consumer, knowing what the CDC is, coming to the website, being a consumer of guidances that were previously intended for a different audience. Among the work that we did just as I was leaving the CDC, and I know a lot of this work continued after I left, was to sort of say, like, let's hold a mirror up and say, in 2023, post-pandemic, what are some of the challenges that we had as an agency, and where are the areas that we need to do better? We needed to streamline our website. That was among the things. We needed to be able to talk to the American people and have our guidelines talk to the American people. We needed to be able to say, it's not like do this and not that. It's do this if you can, do this if you can, do the next thing, do this. You know, we had to have guidances that were easy to easy to implement wherever you were around the country, and in Navajo Nation and Manhattan and Hawaii and Alaska, and these are all very different communities. So I think the agency learned a lot during the pandemic. There was a lot of work ongoing to try and step up to what is happening in 2023 compared to sort of what they were used to in 2019. Sadly, that work has, certainly stopped and been diminished.
In 30 seconds or less, you're in a room full of industry leaders, pharma, hospitals, physicians. Are they doing enough to stand up for public health right now? What should they be doing?
I will say that when people say, it's going to be a little more, when people say to me, should we trust public health? My answer to them is generally, did you brush your teeth with the tap water this morning? Because if you did, you trust public health in some way, right? And to say, I know we're having a real challenge with vaccines and what to believe and what to read, but there's a lot of public health that continues to work. And I think we will start to see how it doesn't work as we see outbreaks here and there. We have another measles outbreak now ongoing in South Carolina. So we will start to see where that trust is, where public health has been eroded. I do think that when people used to say to me, like, how are you going to improve the trust in CDC and public health? CDC does not have the bandwidth to improve the trust. No single individual has the bandwidth to improve the trust. And everybody sort of has to work together to say, that we need this public health. We need this infrastructure. We need people doing this great work. They're heroes doing this great work, quiet heroes doing this great work. And so I would love sort of more people singing that from that songbook.
Have they done enough?
I would love for them to do more.
Last word.
Yeah, agree. I think there is a lot more to be done. If you do public health well, you never see it. That's a thing that we talk about all the time, but it's true. Public health does not do a good job of communicating all of the things that you're not getting. But right now, though, more than ever, I think we do need people to be talking to their congressmen, to Senator Cassidy, Senator Sanders, all of those people that may have some capability to make change, to put in new authorizations and things like that. Also working with the professional societies to make statements. Right now we have multiple different professional societies that are saying that the secretary should be removed. We have never been in that place before. And so this is a time where we are in a different place. We have to act differently. We want to talk about science, that's great, but we have to talk about how we make science happen. And if we can't do that in this current environment, we should be saying that out loud.
Thank you both so much.
Can I just say thank you to STAT for being like one of my go-to resources in health news and for like incredible work at STAT News. So thank you all.
Excellent.
Thanks. So much.
All right. I think that was Stat Summit's first ever surprise guest. So thank you again to Rochelle Walensky and Dan Jernigan for that important discussion. We are at our first coffee break. Right upstairs on the mezzanine, you will find snacks, drinks, and our sponsor booths. Please go check them out. We will resume programming at 11:30. Enjoy.
Please take your seats. Our presentation is about to begin. Thank you. Please take your seats. Our presentation is about to begin. Thank you.
I see folks filtering in. We would like to ask you to come back from coffee, fill your cup, and come please take a seat. We have a very exciting evening of programming coming up. It's still the morning. I obviously need a little more coffee to get going. Please grab a seat. You can even fill it at the front. I hope everyone takes a picture of the beautiful view today. All right. I think with that, we are going to get started with the next part of our day.
From our supporting sponsor, please welcome to the stage Teresa Bertrand, Head of Strategy, US at Zeno Group.
Good morning. I'm Teresa Bertrand. I'm the U.S. Head of Strategy at Zeno Group, a global integrated communications agency that helps brands solve business challenges. Next slide. And when we look to the future of health and wellness, the question isn't whether science will keep innovating. The question is whether connection can catch up. And, go back a little. And that connection, so the decade's next greatest risk is, as we see it, not a failure of science, but a failure of connection. Connection between innovation and the people it's meant to serve. Connection between providers and their communities. Connection between advocacy groups and families across a lifetime of care. There's a lot of talk about patient trust in healthcare, which is incredibly important. But we also need to consider the trust between companies and clinicians, the trust that truly drives and guides everyday decisions. We see this in the data. Zeno's new brand love and demand study focused on health and wellness and clinicians found something striking, that nearly 9 in 10 doctors, nurse practitioners, and physician assistants keep prescribing treatments that they first started using during training. And that's not inertia or habit, it's loyalty. Loyalty that is born from early trust and confidence. And once that connection happens, it can last for decades. And that connection, it's also measurable. So we see here that emotion really drives those decisions that the clinicians are making. Almost 8 in 10 clinicians said that the personal connections that they have with a product influence what they prescribe. And 2/3 or more feel the same way about the companies behind it. So in other words, it is more than what a treatment does, but also who stands behind it, how they show up, and whether their story feels human and true. And of course, that is based on a few simple but very powerful qualities: reputation, transparency, and experience. Those qualities really shape perception as well as behavior. And of course, before I move on, emotion does not replace the science. Science is, of course, hugely important. Clinical outcomes are the top influencing motivation for most clinicians. But emotion is an amplifier of data because it drives belief, and that belief drives change. So companies who lead with empathy, clarity, and patient-centered purpose turn innovation into impact. Because even when you understand the science, what moves you to action is feeling understood by it. And that decision isn't made in a silo. It's not made in isolation. There is trust that travels through people. So clinicians tell us that peer networks, their peers, is a huge influence on where their sources are coming from and where they're spreading influence when it comes to what they prescribe. And that far outpaces conferences, advertisements, and reps. It also lives in patient conversations. 70% of doctors and 80% of nurse practitioners say that patient requests drive action. They take action on patient requests, and that most of those patient requests are clinically appropriate. So if we want our science to scale, we need to connect with the ecosystems of trust that surround all of those decisions. And of course, connection looks very different across generations. So when we look at clinicians from Gen Z through boomers, connection doesn't show up the same. So for Gen Z and millennials, those younger clinicians, they are peer-powered and purpose-led. They put their trust and they power companies that prove transparency, access, and ethical pricing, which came up a lot. As one clinician, a younger clinician, told us, sometimes companies don't understand just how many people can't afford their medications. For Gen X clinicians, they are reputation-led pragmatists, and they have a lot of trust in and loyalty in brands, but they're ready to walk if values misalign. And boomers, they rely on experience and consistency. Boomers will give the benefit of the doubt to companies who show up again and again. But across all generations, 3 things really stand out, and that's the trust, the transparency, and the humanity. So as we look ahead to the next decade, let's make this our challenge, to build brands and companies that don't just promise better outcomes, but create better connections, to invest in relationships with the same rigor that we put into research, because the future of medicine will rely not only on what we discover, but on how deeply we connect. Thank you.
All right, everyone, this next part is very fun because we get to learn some insights from all of you. So if you have attended past Stat Summits, you might remember there were some poll questions throughout the event. So I'm going to talk you through how this will work and get into our first question. Please, first, we need you to take out your phone and open the Bizzabo app. If you don't have the app downloaded yet, you can use the QR codes on the screen to get it from the App Store and quickly set up a profile. In the app, on the event homepage, you will go and click the 3 dots at the bottom right-hand corner of the screen where it says More. And then from there, you will select Polls. And from there, poll question number 1. You should see this question that will be up on the slide in a moment, also populate on your screen. And for those of you who are turning in virtually on your laptops or the browser version of Bizzabo, you should see the link to participate in the poll appear in the chat window to the right of the live stream. So our first question of the day is the following. How important is it to you that your company's reputation helps influence the prescribing behavior of healthcare providers? Not important at all, somewhat important, important, or very important. So if everyone could take a moment and think about it. You can select what works best for you. That is, how important is it to you that your company's reputation helps influence the prescribing behavior of healthcare providers? Not important at all, somewhat important, important, or very important. I'm going to open the app myself and vote. And after I vote, I'll be able to take a look at the results. It looks like we have, at the moment, very important in the lead. So a lot of people are saying that this is their priority. And then the second option is important. So for some people, it might not be the top thing, but still something that they want their company to be thinking about. Oh, very important is now really taking the lead. So I think we have our answer. Thank you all very much for voting, and please keep that app downloaded so that we can do this a few more times throughout the day. And now, on to our next session, here to discuss the frontier in immunotherapy revolution. Please welcome to the stage Dr. Miriam Murad, the chair of the Department of Immunology and Immunotherapy and director of Precision Immunology Institute at the Icahn School of Medicine at Mount Sinai, and to moderate, Stats cancer reporter Angus Chen.
Okay, well, Miriam, thanks so much for taking time to be on the stage with us today. You've spent really your entire career as a scientist studying this one cell macrophages. And I think I hear from people that your vision for macrophages is that they are so important to the future of immunotherapy. So I'd love to hear that from you in this kind of big picture sense. What is the great potential of this cell for patients?
Yeah, so maybe I can define macrophage for a minute. So it's a very interesting cell type because they are the most conserved immune cell type across organisms. So from a small organism to large animals, this is the one commonality in terms of the immune system is that cell. And their unique role is to constantly clear damage, okay, and detect threat. So there is two big lineage of macrophage. Well, one anecdote, one anecdote, is that the scientist who discovered them, called Elie Mechnikov, he was looking for a principle of life. He wanted to know what drives, what is life, like, and what is essential for life. So he was looking for commonality of biological principle across organisms, and this is how he discovered these cells, right? So very important cell type. Now, he discovered these blood-derived macrophages. There are now two lineages, one lineage that we discovered in 2010, my laboratory, where those macrophages are populating all organs of your body and are constantly protecting those organs by clearing damaged cells. And there is another lineage, which is the lineage that Mechnikov discovered, that are blood-derived macrophages, inflammatory macrophages, that goes where the injury is. So each time you have an injury from a myocardial infarction, an infection, rheumatoid arthritis, or cancer, there is an accumulation of these inflammatory macrophages. And in cancer, for example, I'm an oncologist. By training in cancer, these macrophages sometimes exceed tumor cells. Right? So if they are so present in site of injury, we can potentially manipulate them to help us get rid of that injury. And to me, this is really the mission of my laboratory.
Yeah, I mean, you talk about the incredible therapeutic potential of these macrophages. There being these two distinct populations, but they have so many different functions that you can use in cancer. But you also mentioned all these other diseases.
Yeah.
So the potential is very broad.
Yeah, it's exactly right. So because they have very unique machinery to detect threat, and the unique machinery to also clear damage, they can adapt to any type of lesions, right? So they go, for example, in mass upon myocardial infarction, trying to help, but sometimes they harm, okay? But you can use them because they go exactly where the lesion is. In cancer, it's the same thing. They accumulate there, trying to help. So they are going to try to repair the tissue because they see these cells proliferating, and in trying to repair the tissue, they promote tumor growth. Okay, but the fact that they can detect where where the damage is extremely important to us, and this is what we are trying to exploit: is use them as a conduit to really to treat the damage. There is a big focus now in the field on targeted therapies, right? How can you? And I think the future of medicine is exactly that: is how can you bring medicine to the site of injury and maximize your therapeutic window at the site? Well, macrophages do that. They go to the site, and if you can just follow their cues, you can develop extraordinary targeted therapies that are going to really focus on getting rid of the damage without affecting, the body. Yeah. And then you can maximize, therapeutics locally.
So, I think I heard someone say the other day that they felt that the juice has been squeezed out of immuno-oncology, sort of referring to this string of failures in immunotherapy when it comes to a lot of T-cell targets, but also some macrophage targets. And the question for you, I think, is, what are you seeing in the field now that gives you optimism that this cell can be used soon? And I guess, really, why now do you feel like is the time to push forward on this biology?
No, absolutely. So we have seen this extraordinary success of checkpoint blockade, right? Which are these molecules that can, by blocking that molecule that reduce the effector function of lymphocyte, by blocking them, you can reactivate this effector function. So these checkpoints are really are targeting that branch of the immune system, which is called the adaptive branch, right? So it's a branch that is educated by cells, such as macrophages, to go and kill. we also know that patients resist these checkpoints. So my really big focus of my lab has been to identify why we resist to checkpoint blockade. And we've seen that one of the mechanisms of resistance are those macrophages that are still trying to dampen effector T cell function. So, and I think now even the T cell chauvinists totally accept that this is really the frontier we need to focus on. So those macrophages are always trying to reduce inflammation, right? They are trying to repair. And so you can put all the checkpoint blockade if they decide that they need to shut down that effector T cell function, they can do it. They can do it because they have the resources to do it. So many of us have looked at how are they shutting down effector T cell function, and we are finding programs that are very efficient at making T cells not effective. So we've talked, well, I talked to you about that program on IL-4. There is a repair program that macrophages use very well to repair. This is always induced in cancer. We are trying to dampen that program, and there are other ones. You know, in CAR T cell therapy, for example, you know, Carl June now says everywhere that the reason why the CAR T cells don't accumulate in solid tumor is because you have these lines of macrophage stromal interaction that really block T cell entry. So we think that this is a compartment that can be so useful to target in combination with checkpoint and in combination with CAR T cells.
You recently actually found a way to potentially make that happen, allow sort of shift the balance of the immune system to allow for our existing immunotherapies, blockbuster drugs like Keytruda, to work better in patients where it had stopped working, using Regeneron's Dupixent.
Yes.
So I guess part of the question, I think, is where do you think the science is with macrophages, and do you think that we are really going to see more of these targets and maybe us actually starting to enter the clinic with some of these targets?
No, I do. I think we are very close. So just to talk about Dupixent, it's an interesting story. So Dupixent is a molecule that Regeneron made to treat type 2 disease, so eczema mostly, and now it's been approved for severe asthma. And that type 2 immunity, we know, distracts from what we call the type 1, which is that type of response that enables the T cells to eliminate cells, so whether they are virally infected cells or tumor cells. So what we were, and that type 2 response can give rise to allergy, but is also a very good way to promote this repair function of macrophages. So we repurposed this drug, and now are saying that if you combine Dupixent with checkpoint blockade, we were rescuing response to checkpoint blockade in patients that have started to resist checkpoint therapy. So that is putting us on a path of saying, okay, how can we reprogram macrophages, so that they dampen their repair program and shift them toward more immunogenic. So tell them the cancer is fooling you here. You still have to eliminate, stop repairing, and they know how to do that. I always give this example of COVID. During COVID, and I've studied the lung of COVID patients for a long time. The patients were not dying. The patients were dying of inflammatory macrophages. They were so activated. They were activating those T cells, and this is why patients died. We never saw, a virus or trace of a virus in these advanced COVID lung patients. So the virus was gone, but these inflammatory macrophages were really killing the tissue, that lung organs. And this is what we want to do in cancer: is put them in that state, that immunogenic state, and it exists because they know how to do that. And it's up to us now to really teach them how to get out of that repair program and go. So Dupixent is one, but we have now identified many other mechanisms that help us, switch from a repair to a pro-tumor, to the non-tumorigenic, or what we call an immunogenic program. But then there are other strategies that other different groups are doing. So a strategy, for example, that a company I advise for, but I like very much, called Joanne Bio, where they are using bispecific antibodies, where they will have one arm of an antibody that recognizes the tumor antigen and the other one that will bind to a receptor on macrophage that is an immunogenic receptor. So macrophage, when they see a bug, they eat in an immunogenic manner. They eat that bug and they become immunogenic. They want, an inflammatory response to that bug. So this is how, an interesting strategy is to bring the tumor antigen to the immunogenic receptor that is usually utilized to uptake a microbe, and that we know leads to that immunogenic program, but cancer are usually eaten by different type of receptor that put them in that repair program. So that's very interesting. It's going to in the clinic soon, and that particular program was bought by Sanofi, so it's going to be developed quite, I hope, significantly, and there are other strategies.
Yeah.
That is exciting.
So there's things entering the clinic for oncology. It's really interesting to hear about how targeting macrophages in this way really shifts the immune state of the whole immune system, kind of not just affecting one cell, but sort of bringing other things along with it. And I want, we started out this conversation talking about how it can be used in many disease states. I think one of the focuses of your lab now is looking at autoimmune and inflammatory conditions and macrophages and aging, so it's trying to find ways to promote healthy aging.
Yeah, so one of the framework is to look at macrophage across different diseases, right? Because this is how you learn so much. So cancer, they're in this repair program. In inflammatory disease now, they are too immunogenic, very similar to what we'd observe in COVID. So when you look at macrophage across state, then you reveal, potential targets, right, and pathways. So it is clear, and now I think the field is very aligned on the fact that macrophage is a big driver of inflammatory disease. For example, inflammatory bowel disease, multiple sclerosis, fibrosis are all driven by this inflammatory macrophage. These macrophages that are going to activate also fibroblasts and contribute to fibrosis. So inflammatory macrophage, this big inflammatory macrophage lineage, is a big focus of ours to treat cancer and inflammatory diseases. But there is another lineage which my lab discovered, which are those protective macrophages, which we call the tissue-resident one. These are macrophages that are present in all our body, from the brain to any muscle, and they are localized in very specific niches. So, for example, they line vessels, they line the neurons, they are close to the stem cells of muscle stem cells, and they are constantly making sure that those structures that are very important in every organ are functioning properly. And we are realizing that this pool of resident macrophage, which self-renew in tissue, declines with age. So a big focus of the lab now is to restore that pool of macrophage in all organs. And we are seeing some very interesting things. So when you restore them, by identifying why they are declining, and often feeding them with metabolite, we see rejuvenation of these organs because we see them clearing all the damage that accumulate. So, there is, I think this is a very interesting time for macrophage biologists or myeloid biologists in general, is really to focus on this cell type across diseases, from cancer to inflammatory disease to aging.
Yeah, I think one of the things that really struck me about these tissue-resident macrophages is how they kind of are gardeners for our body. They help to maintain the health of our body.
Yeah.
And so as we age, if we lose that, you try to find a way to bring it back.
That's right.
Are there targets now that you're looking at? Are there things that are actually approaching the clinic for this purpose?
Yeah, so it's interesting, this gradient. So they are the gradient, but they can harm us also, right? So I've discussed how in cancer, they come to help and then they harm. So there are those that harm, right, in inflammatory disease and cancer, but they are the protective one. And this is what has been a little bit difficult to, explain, but I'm hoping you will understand the complex. It's complex, right? So biology is complex. It's okay. you know, to embrace complexity also. But you know, thanks to advances in technology, we now can see very clearly these two lineages and their ability to have many different roles. So the guardian of tissue health is a very important one. You know, and in fact, I just came out of an aging meeting where we were, you know, one talk after the other was describing how these resident macrophages were reduced in the aged muscle, in the aged heart, or dysfunctional in the aged brain. The aged brain, we know that it's a disease of, that cognitive decline with age is a disease of macrophage because of all the work on Alzheimer's disease. You know, all the susceptibility genes of Alzheimer are in fact genes expressed by macrophage of the brain. If your macrophage in the brain cannot handle clearance of, and it's all associated with lipid handling. When you clear cells, what you have to clear is the lipid in the cells, because cells are full of lipid in their plasma membrane. And we know that if you don't handle lipid very well, you are likely going to age poorly. And cognitive decline is so visible that this is something that we notice the most. But you can extrapolate that cognitive decline with muscle decline, with heart decline, except that you don't measure it as easily. So we are all realizing that that pool of protective macrophage is reduced or is dysfunctional and cannot handle you know, clearance as well. Now with age, you damage, you know, many cells are much more damaged. So you need a vacuum cleaner. It's much more efficient. So and the fact that they are declining while damaged cells are increasing, I think is the reasons why they contribute so dramatically to tissue damage with age. And the hallmark of aging is really that this decline of that accumulation of damage that's not clear.
Yeah. I think the macrophages of the brain that you were talking to, people might have heard of them called microglia, is that right?
Yes,
yeah.
It's exactly the same cells.
Same cells.
You know, every organ has a name. You know, we call macrophage of the liver Kupffer cells and macrophage of the brain microglia, but they're all macrophages.
Yeah.
Yeah.
So with our last few minutes, you know, one of the things that you've been very outspoken about, especially this year, is
politics.
Trying to defend, yeah, talking about science and sort of the future of science.
Yeah.
When you think about this, are you worried that this incredible vision you have for immunotherapy and the role of macrophages in immunotherapy in the next 10 years is under threat? And is there something that you try to encourage the scientific community to do?
Well, what I would say is that we are at an extraordinary time of science and medicine. You know, because there's these extraordinary advances in technology with single-cell technology, which enables us to really understand lesions at the cellular and molecular level. You know, I look at a lesion now, and I remember when I started in the field 25 years ago, you know, we had like 5 markers. I barely understood what was going on in that lesion. Now I look at it and I just understand, right? So we feel so close to being able to reprogram, you know, those cells and repair. And at the same time, where everything is happening, we hear that we are going to pull off, you know, like a big source of funding for us, which is the NIH funding, right? NIH for us is very important. You know, we also benefit from other source of funding, from philanthropy in this country, because our donors are so generous, and industry, but that anchor is the NIH. So why? You know, I just don't understand why would we, as a society, just reduce funding when we are so close and when we are so able, you know, to discover and to treat. You know, I see cancer, well, I don't see patients anymore, but I do a lot of clinical trials and see families where, you know, when you have cancer and you know that maybe there is potential therapy there, but the reasons why you are not going to be able to test it, because now it's all about permutation. You know, when I was talking about combination, you just have to try them. You have to try all these combination treatments, you know, in models and in patients, and this is extremely costly for us. And the reasons why we are slow now is just because of funding. So, you know, it just strikes me that as a society we are not more vocal about accepting that our military budget will prevent, you know, threats that you know, are potentially there, but not so close, right? And we accept that and we at the same time don't fight when we, you know, our tax, you know, money is not funding life, basically. So I'm saddened by it, and I think we have to be vocal about it, you know, in a very bipartisan manner. This place has been dominating the research scene, right? Many of us, you hear my accent, you know, I come from here. You know, I came here as a kid because I thought this is where I wanted to do science, right? This is where the mind is, this is where the optimism is, this is where the ambition is. And we can't let that go, so we are here and we're going to fight it. I hope you'll fight with us. You know, it's, we fight for patients, we fight for science, and we have to fight for progress.
Yes. Right, thank you.
For this sponsored conversation, please welcome to the stage Vice President and Therapeutic Area Head of Immunology Clinical Research at Merck, Dr. Eileen Pangan, and to moderate, STAT Senior Writer for Medicine and Editorial Director of Events, Matthew Herper.
Hi, everybody. And welcome, Dr. Pangan.
Thank you.
Thank you for being here with us for this sponsored conversation. I've covered Merck a long time. I've gotten to see Merck shift its focus many times over the years, and we're here to talk about a new area, which is immunology. We were talking backstage, and you said one of the reasons you came to Merck was the company's commitment to this space. And there was a really big example that meant a lot to you personally, which was the $11 billion 2023 purchase of Prometheus Biosciences. Why did that deal actually mean a lot to you in doing your work at Merck?
Mm-hmm. You're right, Matt. I came to Merck in 2022 because I believed the fact that Merck was committed in building a new immunology therapeutic area. And I was very fortunate enough to be given this opportunity to lead the clinical space. But more importantly, Merck's focus on patient needs really enabled them to move to the immunology therapeutic area. The acquisition of Prometheus in 2023 absolutely signaled the long-term commitment of the company in immunology. So what my team is doing right now is really building on that investment by expanding and advancing our global clinical trials, and also continuing and heightening the efforts on precision medicine that Prometheus started when they were still a company out in San Diego. In the end, what we want to do is hopefully address the remaining unmet needs for patients with immune-mediated inflammatory diseases, but especially inflammatory bowel disease. You know, Matt, these patients with IBD, they usually get the diagnosis in early adulthood, and it can truly impact a patient's life, impairs their quality of life, limits their ability to function, and for some of these patients, when available therapies don't work, they sometimes progress to surgery.
And it's right when their lives are starting off, right when they're starting their professional lives.
Right. And this is really why Merck is so focused on IBD as well. Merck believes in addressing patients' needs and understanding where they are in their patient journey and hopefully getting new therapies to them.
Well, let's zoom out, because in order to decide where you're going to go, you need to know what the landscape looks like.
Yes.
What does the immunology landscape, particularly in IBD, look like? Where are the gaps in care that you think you might be able to address?
Mm-hmm. You know, I've been in this industry for over 2 decades, and we've come a long way, right? Our field in immunology has come a long way since I was still a prescribing rheumatologist to today, where a lot of these diseases, especially IBD, do have available standard of care. But when you look at these patients and what they experience when they go to the clinic, number one, the decision of what treatment to go to is still trial and error. A physician doesn't have a way to understand this patient needs this drug, this other patient needs this drug. And if they do get into remission, the second gap, Matt, is can that remission be long-term? Like if I'm a patient, I suddenly feel so good after I got the right treatment, I want to know that I can be on this treatment for a long time and not have to think about my disease. So I think those are 2 gaps that I'm hoping
further understanding of the multiple pathways involved in the diseases in immunology like IBD could help us address those needs.
So what makes Merck special here? What gives you an edge in addressing some of those needs and filling those gaps?
We are carefully and strategically thinking, how do we really elevate standard of care? Everybody talks about we want to elevate standard of care. First of all, understanding what that means and the goals that we have for our patients and prescribers, and then having a multi-pronged approach. Number one, of course, we're looking at novel targets, right? The more we understand about the diseases, we identify novel pathways, but we're also looking at combinations of targets. We fully understand after watching all the drug development that sometimes you might need more than one drug or more than one target. But the third strategy in that approach is how can we bring precision medicine to immunology? Because in the end, being able to identify which patients should get which drugs could also elevate standard of care. We're doing that at Merck, and we're actually embedding precision medicine in every stage of drug development, from novel target identification to patient stratification. And then lastly, but I believe more importantly, Merck believes in patient equity. That means our early discovery efforts are also looking at oral therapies with biologic-like efficacy so that many more people around the world can have access to Merck's innovation in immunology.
Talk a little bit about the advantage of having an oral there.
Yeah.
Why does that bring equity?
For me, number one, as you know, patients need to have preference. They have a preference and they need to have a choice. Some people don't want to inject themselves. Some people don't have time to be in the clinic to get an IV infusion because they're working, they're taking care of their kids. Having an oral therapy that delivers similar efficacy and safety is a good option for patients. But also think about patients who live in areas where there's no refrigeration. Like, how can they be on a biologic that requires refrigeration? So hopefully an oral therapy can also address that need. So I think it's not just about bringing better efficacy and safer drugs, but also thinking about making it accessible and convenient for patients whenever there is a need to. Because believe me, a lot of patients sometimes prefer an injection than having to remember a daily pill. It's giving patients options, we believe, is an important work that we at Merck are trying to also address.
Sitting where you are, it's kind of like you're on a mountaintop. You can see a little bit further than the rest of us. So give us a sense of, in the next 2 to 3 years, where do you think this field is going? How are things going to be different in 2028 compared to now for patients with these diseases, both in terms of what's going to be coming down the pike and what will actually be there?
Mm-hmm. I'm really excited about all the work, not just what we are doing at Merck, but what everybody in the field of immunology is doing. I'm really excited about novel therapies and combinations because I believe it's going to be a collective effort to change the narrative for these immunology patients. My hope is that we see novel targets that are bringing a different type and level of efficacy and safety for these patients, combination therapies that are hopefully breaking the ceiling. And Matt, my dream is, hopefully, we will be able to bring a predictive biomarker to take the guesswork of which patient should get which therapy. And I know a lot of efforts are going into that, and we at Merck are obviously trying to invest in that field as well.
Let's talk a little bit more, because you've mentioned it twice, this precision medicine idea, and it's been a big deal in cancer, it's been a big deal in a lot of, in some other diseases. I mean, in some ways you can argue cardiology was always precision medicine. But how do you see this, how do you actually find that biomarker, and how does it change what you do in developing a new medicine?
Mm-hmm. I know it's been a really hot buzzword recently about precision medicine, but our field has been trying to do this for a long time. Mm-hmm. And it's not easy, it's not simple. Otherwise we would have something. But I think when we see drugs that are being developed, the kind of efficacy that we're seeing, it is important for us to look at precision medicine as another way to do this. There's a lot of tools and technology right now that you can use to funnel the work towards precision medicine. There's AI and machine learning, looking at all of the biobanks in the registries to help inform your predictive biomarker. So that kind of work is happening at Merck right now because we believe this is one of our strategies to hopefully elevate standard of care.
So, and what does this mean for you? You were a practicing rheumatologist. You've been working in this field for a long time.
Yeah.
You talked about the commitment.
Yeah.
Talk a little bit about what this means for you personally to be doing this work.
Well, I will tell you, I still remember when I was still practicing, it was a long time ago. But talking to a patient, I always felt good about making the right diagnosis. Like us physicians, we love the fact that we can tell a patient what they have. But when they ask me, well, what will get rid of it? Or like, what will treat my disease? And I tell them, I have this 2 or 3 medicine, and they will ask me, what's the right one for me? And it's hard to say, well, let's try this. I think it's going to work, but we don't know if it doesn't work. Let's try this. That conversation becomes a little bit less confident and less certain for the patient. So I think what's important as I continue my work in industry is to be able to give some level of certainty when a physician and a patient has this conversation. Of course, there's never going to be 100%, right? But at least you have a little bit more knowledge, being more personalized in how we approach the treatment of our patients is something that I believe is really worth the time and investment in doing, Matt.
All right. Well, thank you so much for being here, and thank you so much for all those thoughtful remarks.
Thank you. Okay.
And now, for our next session, we're really excited to welcome the Internet's most influential physician. Please welcome to the Stats Summit stage Dr. Mike Varshavsky, board-certified family medicine physician and influencer, and to moderate, Stats host of the Status Report, Alex Hogan. And as they are making their way up to the stage, roll it.
It all started in med school. I'd heard that it's impossible to have a social life and still be a successful student. I set out to prove that wrong. What I didn't set out to do was completely change the world by posting a selfie with my dog. But that's what happened.
He's been named the sexiest doctor by People magazine. The hot doctor. What?
The term going viral can mean so many things.
As a lifelong nerd, this was awkward territory for me, but I wasn't mad at it. Thousands of Instagram followers turned to millions. New opportunities came. I got national press attention. I got invited for media appearances. I was able to raise money for charity. Yeah, it was superficial attention, but with my foot in the door, I thought surely the TV producers would let me talk about subjects that are actually important, right? Breakthrough research, preventive care.
I'm feeling a little faint. Can you catch me?
Oh my God. Not so much. I was done asking for permission. Despite over $300,000 of medical school debt to my name, I hired a small team to help me start a YouTube channel. I decided that every moment I wasn't in the hospital would be dedicated to brainstorming, scripting, filming, and posting videos. I'm not gonna lie, it was a bumpy start.
So hey everybody, I'm Dr. Mike Varshavsky. Hey everybody, I'm Dr. Mike Varshavsky.
We pushed through, and eventually I became a bit more natural on camera, more myself. We hit some of the YouTube staples with an educational twist: morning routine, night routine, day in the life, what I eat in a day, responding to comments. Our viewership was okay, a modest, enthusiastic audience. We certainly didn't take off like a rocket. In fact, a year in, our partners considered the venture a failure. They fired us, but we never gave up or signed a DNR. Now, over 1,000 videos later, our viewership has created opportunities I never saw coming. We've done videos with leading experts, top athletes, the sitting vice president, went on a tour with a live show, made some new friends and some enemies, started a podcast, built an international team, and we've raised hundreds of thousands of dollars for charity, all without ever giving up my job actually practicing medicine. More importantly, I've reached more of you in a time where misinformation is spiraling out of control. We now have billions of views experts accurately breaking down an endless amount of medical topics. I get messages all the time about the content bringing families together or about lives being saved because of me banging on about
Chest compressions, chest compressions, chest compressions.
Together, we've built something bigger than I ever imagined possible. And here's the thing, we're just getting started. Stay curious, stay informed, and as always, stay happy and healthy.
That got me fired up. I thought we'd get pyrotechnics, but they're like fire codes. And but,
honestly, the editors do a great job in making our production, which is me facing a camera, seem huge. So I thank them for that, and obviously appreciate everyone's attention today. So,
Dr. Mike, thank you. So let's just get started. You know, people have been increasingly turning to content creators like yourself for their information. You've been successful at making sure trustworthy science-based information cuts through the noise on social media as anyone. How do you do it?
I think I realized that there was a gap, and that gap was we were doing great research. There were great doctors that were taking care of patients, but the messages weren't landing in part of the healthcare system that most people deemed not part of the healthcare system. You know, being a family medicine doctor, I'm trained to be there for my patients, whether it's in the ER, inpatient setting, nursing home, we even do home visits. But then I realized we weren't there on social media. And because we weren't there, there was a lack of evidence-based voices online. It created this gap that was filled by grifters, by snake oils people, people who were selling all sorts of miracle cures. And we weren't there to combat that. I actually wrote about that in 2017, before the pandemic, warning people that this is going to be an issue. 3 years later, we're facing the pandemic, not enough doctors to fact check the immense amount of misinformation and disinformation being thrown at people. And as a result, people were confused, people were making bad decisions, and the CDC, the FDA, all these large agencies were playing catch up because they had no presence on these platforms. And honestly, the most watched videos on the #1 video website in the world, which is YouTube, were mine. And that's sad. Because it should be brilliant researchers, it should be brilliant scientists, but because they didn't invest in those tools, in learning about the platforms, in learning how to communicate to millions, huge missed opportunity.
And to that point, you know, the public seems to be increasingly skeptical about our health institutions. You know, are there lessons that can be learned from successful content creators like yourself, particularly when it comes to authenticity and trust?
Yeah, I think we've lost trust in the healthcare system for a myriad of reasons. One of the big ones being our craving for certainty and feeling that we needed to be certain in order to win over authenticity. If we go back to 2021 when we're making recommendations about the COVID booster as an example, instead of putting out a very honest message of here are the high-risk groups who would most benefit, here's who may not need the vaccine. We said everyone should get it. And the audience felt that they were being tricked. They were being misled because doctors were trying to resort back to this paternalistic approach of, we'll tell you what's good for you, just listen to us, you are not the expert, we are. And by taking that approach, long term, we've lost trust. People don't know what to believe. They start following all sorts of echo chambers on social media, and as a result, fall victim to misinformation. Doctors are finding it more difficult to talk to their patients in exam rooms, on television. And now, when we want to counter something that's coming out from even our current administration, we don't have any tools to do it. You know, 20 years ago, if there was a bit of misinformation, it was easy to do a sit-down interview with the leading news station or 60 Minutes and counter that narrative. Now, where do they go? I wanted to be the place for them to be able to come on my platform, have that conversation, or for me to be that voice to be able to fact check that.
And speaking of COVID, you know, you had already had your YouTube channel for a few years when the pandemic hit. Do you think like the pandemic fundamentally changed or kind of broke how the public kind of processes health information?
I think it threw gasoline on the fire because people were already using social media to answer their health questions. If we're being honest, like show of hands, first time you have a symptom or first time you get a diagnosis, is the phone the first thing you look at? I mean, that's natural. And that was happening in 2017. It's why I started YouTube. And when the pandemic came, and there was so few answers even if those answers were incomplete, coming from the legit agencies, people craved those answers and they got them. But they got them from those grifters that I mentioned earlier, and as a result, those people were misled. Those grifters ended up getting massive platforms. They learned how to make use of social media and how to use confrontation, all of these skill sets that marketers have to influence people, while we all raised our noses in the healthcare community and said, we're above this. Good scientists, good doctors do research or they treat patients. They don't spend time looking good on camera or getting pretty lights. But that was a missed opportunity because a big part of healthcare is making sure you're closing that loop and making sure your message lands.
I'd like to talk about your process a bit. You're pretty prolific on YouTube. You put out a lot of videos. How do you come up with your video ideas, what you're going to cover?
This is the biggest struggle because we're going on year 8 of making content, and we've been publishing 2 videos a week at a consistent rate that entire time. We don't want to sensationalize content. We want to always keep medical integrity at the top of our moral compass. But the reality is it's hard. You have to make content about your personal life. You have to bring your dogs into it, your hobbies. And some people might laugh at that or say, perhaps it's unprofessional for a physician to do these things. And I sort of get it because when I first started, I got a lot of side eye from physicians who said, what do you mean you're going to start a social media platform for healthcare? There was a lot of doctors at the time who were making content, perhaps in traditional media, with subjects like, how does your zodiac sign impact your heart health? That was a legit segment on ABC. And they viewed me with this skeptical lens, and I understood why. But I said, there's a way to get a lot of people's attention here and then redirect that attention to something valuable. So when I make, even to this day, content laughing at medical memes or reacting to TikToks, people say, but Dr. Mike, we're in a healthcare crisis. You can influence so many people. Why are you choosing to make comedic content right now? Well, guess what? When that comedic content reaches viewership of 5, 10, 15 million, that video is now in that user's watch history, which means when I make my next video discussing the press conference linking Tylenol to autism, my video by the algorithm will be served to that person. Had I not made that silly video, they would be completely unaware of the fact that there was misinformation being shared with them at the highest levels. So there's a tremendous amount of strategy that goes into figuring out cadence, style of video, accuracy of content, but at the same time, studying what do these experts, quote unquote experts, do so well to trick people into watching their content. They make content relatable. They remove scientific lingo. They use really good quality thumbnails, titling that's captivating. So we learn and study what everyone else is doing well, but we bring it to go to bat for evidence-based medicine.
And I'm glad you brought up the Tylenol video because it was great. And you put that out pretty quickly after the press conference.
I wish it was quicker, to be honest.
Well, it was pretty quick. And we actually have a clip from that, if we don't mind showing that real quick. Just a quick 1-minute clip.
But let's get to the science. Throughout this press conference, President Trump repeatedly says,
don't take Tylenol. There's no downside. Don't take it. You'll be uncomfortable. It won't be as easy, maybe, but don't take it. If you're pregnant, don't take Tylenol. And don't give it to the baby after the baby is born.
Well, as a board-certified, actively practicing physician who's been in practice over 10 years, he's flat out wrong. In healthcare, every decision has consequences. Taking or not taking a medication both carry sets of benefits and harms. That's because medications, therapy, surgeries all have biological, psychological, and even social implications and trade-offs. And what's even more interesting is that those benefits and harms will be drastically different depending on which individual we're discussing. All this is to show that I'm not making an appeal to authority argument here, saying that Trump isn't a doctor, so he must be wrong. I'm actually saying he's wrong because he's wrong. If he was a doctor, he would know that not treating a fever, especially early in pregnancy, can carry serious risks.
And I think that's interesting. So you just saw that video, you saw the press conference, you said, I have to make a video on this. It's right now, this is urgent to counter this. And then, but I think it can be just as important to cover things that are more evergreen. There's some topics that just people just don't necessarily know about. A great example of which is CPR, a very fundamental medical technique. And I saw a clip on the Diary of a CEO podcast where you explained how CPR works. And it was so interesting because the comments on Reddit, it was on Reddit, and a lot of comments were like, I thought I knew what CPR is, I guess I didn't, or I completely reoriented how I thought it worked. And it made me think like, oh, sometimes you think like, oh, everyone knows these concepts, but honestly, like, it can be just as important to remind people about these things. And can we actually show that clip real quick?
So the purpose of CPR is if you find someone who's unconscious, not breathing, is pulseless, that means their heart's not beating. That means clinically they're dead. So anything you do here can only potentially help, can't hurt, right? Because the person's already dead. So by doing proper CPR, what you're doing is you're compressing the chest compressions, to squeeze the heart, which has some blood in it, to circulate the blood throughout the body that still has some residual oxygen in it to deliver oxygen to the vital organs. So you're not actually saving someone's life by doing CPR, you're buying them time. So the purpose of doing CPR is to allow time for first responders to arrive to then give advanced cardiac life support.
Oh, okay.
And that's why the first step of doing CPR has nothing to do with the person and has everything to do with calling for help.
So I guess what I would like to am getting at is that how do you balance those two things? Like when do you decide, oh, this is a newsy topic that needs to be covered, and sometimes like, oh, I have an idea, like maybe we need to discuss something that's more evergreen.
Yeah, it's very tricky because what I don't want to do, given my large platform now, is give air to misinformation, because the reality is, even if I make a video like the one you showed here with Tylenol and autism, I don't want to have 98% of people agree with the video or find it very valuable, find 2% of people who actually disagree with me, and give that misinformation more airtime and get more people to buy into that theory. So when we try and fact check, we weigh whether or not this theory, this video, this press conference has gotten enough attention that is now important to fact check because the benefits now outweigh the harms. I think about it like any other medical intervention in that regard. Because what I see a lot of my younger colleagues who are getting onto social media is they find some obscure, ridiculous statement by someone regarding healthcare, and they say, oh, this is easy for me to dunk on to make content. But now, they're not actually educating anyone because no one believed in that to begin with. But as a result, they're giving that person who's already spreading misinformation more airtime so they look more familiar. And that's a big part of the psychology of influence, just seeing someone's face over and over. In watching this press conference, I also thought about historically what has happened. I remember when President Trump, during his first term, went out and discussed bleach as a potential treatment for COVID-19. And I always felt like that the outcry that media did at that moment was going to come back to bite us. Much in the same way, the way we use our vocabulary these days, we reach for the top in trying to describe words. There's a comedian, I forgot who it was, who said this quite funny, that if your friend says, oh, get a straw for this, it'll be easier to drink, and you say, you're a genius, you now lose the ability to call someone a genius when they actually are a genius. So I look at the situation with the bleach incident where it really should have just been a simple fact check, no emotion, no, we don't inject bleach, we use it on household surfaces, that's it. But instead we created this endless press cycle, endless uproar, that when we need to apply that fact check now, it's less meaningful. Because people say the press is always outraged at everything President Trump says, why am I even going to bother to listen to this? So when we make content, we always try and be patient. We actually don't abide by the typical rule where it's be first. We try to be right. We try to be patient. When we first covered COVID-19, it was 3 weeks after everyone else was covering it because we wanted to bring valuable info, not just to scare people. And same thing with this press conference. I would have loved to make that video a day earlier, right after, but I needed to see how the message was landing with people. I needed to see what the pushback was, how people felt about the pushback, to allow me to make a better quality video that would ultimately influence people who both agree and disagree with President Trump's message.
Looks like we have an audience question.
Sure. Hi, my name is Chris Worsham. I'm a physician and a health policy researcher at Harvard. So one of the speakers earlier today, who was formerly at the CDC, said that good public health is often in the background and unnoticed. And I don't agree with that. I actually think you're part of the public health system, and I imagine you might agree with that. You can tell me if you don't. TV, The Pit, is part of the public health system because people learn things. My research group had a study showing that a single joke in the Barbie movie led to people searching for information about gynecologists. So my question for you is, from your vantage point, how do we change that perception of what public health is, how public health works, how we change health behaviors compared to that way of thinking?
I understand your point because I agree that the pendulum has shifted too much within the last 10 years to being behind the scenes for public health. And as a result, it feels like that's the wrong approach. But I don't think we should write off the work that's done behind the scenes for public health in totality. It's like if I have a patient who has hypertension, where on multiple visits I find that they have an elevated blood pressure, I would approach them with lifestyle changes. But at the same time, if it's high enough, perhaps medications are appropriate. I would never say that lifestyle changes are worthless because medications are so much better. It just really needs to apply differently to each individual. And if you think about it as a whole, both need to be used. So I think behind the scenes work in speaking to legislators, very important. Making sure that the people that are working behind the scenes for the president's administration are also getting influenced. But then the public has such a great untapped influence. The examples you mentioned are great. I think about the movie Sideways and how it tanked Merlot sales because of that one line in the movie. So there's a tremendous untapped resource here of linking healthcare with culture that I feel like my channel is just scratching at the surface. And everyone likes to give compliments, and I'm very appreciative of it, the fact that we've done so well. But I think there's a way to do this even better, to do this at scale. I'm one person, a young doctor. Let's get the brilliant minds at Harvard in on this. Let's get the brilliant physicians trained up to be able to have conversations that are difficult and not to be afraid to step outside of the lab, the medical office, the ER, because the more of that we have, the more that they're viewed as people, not experts all the time, I think the more effective they'd be in having their message land.
And we'll get one more from you.
Hi, I'm Sarah Todd. I'm a reporter at STAT. I was wondering if you could talk a little bit about what kind of pushback you've gotten from Trump supporters when you do that video on his response to Tylenol. Do you face threats? Just if you can talk a little bit about that kind of reaction.
Honestly, minimal. It happens, you get negative comments, but honestly, when I make a video grooming my dog, I get negative comments. So the reality is you're always going to have some subsection of the internet dislike you. What's interesting about algorithms is that they've gotten really good at showing your content to people who will enjoy it. So when something goes viral, that means the algorithm has deemed it wide appeal. And when that happens, that's where you actually get the biggest influx of negativity. Back in the day when YouTube had a trending page, you'd know you made the trending page because of the influx of negative comments. Because now your content is being shown to people who don't like you, who disagree with you. And I think that we need to be less afraid of that because A, it doesn't happen as often as we think. And B, when it does happen, there might be a kernel of truth there that we can use to get better. You know, even the most ridiculous comments, I think, carry a small nugget of truth that we can learn from, if for nothing else, to become strategically more effective as communicators.
And that actually leads perfectly into my next question, which is last week, Congresswoman Alexandria Ocasio-Cortez said that Americans are being algorithmically polarized, sorry, that's a mouthful, by social media. What do you think about that? Do you think it's affecting how people access information about health?
For sure. I mean, this is happening on social media, but it even happens within our AIs. You look at ChatGPT, you ask the same question. It depends whose chat you're using. Your answer will be drastically different. What I think is happening, that's at least the most under our control, because I like to think about all these situations as practically as possible. Perhaps that's the family medicine doctor in me. is, are they being algorithmically polarized because the algorithm is bad, or because one group of people has learned to use the algorithm to their advantage so well, and we're falling behind? I think that's a big part of it. I don't think that there are enough people who understand what the algorithm is, how social media works. I look at a recent example where Secretary Kennedy essentially defamed the entire medical profession and went on a podcast and said all doctors are just in it for the profit, and they're aiming to keep their patients sick. I mean, something that would immediately lose someone's position at any corporate job in America, at least I like to think so. But he says this, and the AMA, which is the group that supposedly stands up for the physicians in America, issues a strongly worded tweet that reaches 5,000 people on their X. Or they get the president of the AMA to sit in front of a webcam with terrible audio, the screen reflecting in the glasses with no lighting, to also make a response video reading a script from a screen. It just shows, like if you show those to people side by side, Secretary Kennedy sitting relaxed, great atmosphere, perfect video content, in comparison to this really robotic speech, and you don't even know what the substance of what they're saying is, you trust Secretary Kennedy over President Mukamala. So I think we need to understand that it's not about putting on fancy outfits. It's about being presentable, realizing that fancy cameras are no longer just for the top tier studios in Hollywood. You can get an amazing set for a couple $1,000. And I know how much the AMA fees are every year, and I know they can afford that.
So it's that point. Like, how do you think medical institutions themselves should be thinking differently about reaching people more directly versus with social media?
I think they need to study the algorithms. I think they need to understand what works on each platform. They have to hire individuals who are users of those platforms. One of my biggest strengths and why I think we have so much success is because I spend a lot of time scrolling. Perhaps these days it's doom scrolling, but I scroll a lot, not because I love social media and it's so engaging to me. I watch it as an educated consumer. I see what works. I see what's getting people's attention. I see how people's behavior shifts between likes and comments and how it works distribution-wise on each platform. Understanding that when a YouTube video is made, someone actually has to click and choose to watch it, versus on TikTok, it's passively fed to them. How does that impact someone's behavior? How should that impact how I present my information? So you have to think about all of this as a starting point. And from there, you will gradually become more adept at making content go viral, having it land to more and more people, because user-generated content succeeds from people enjoying it, liking it, and while that is annoying for us in the healthcare space, because no one really wants to sit through a science lecture, there is a way to make science enjoyable and make it not seem boring. And I think we have to invest in that.
And as so many people nowadays get their not videos, it's often not sitting through a 10, 15-minute video. Sometimes it's a TikTok or a YouTube short. Those can be 60, 90 seconds. But often medical topics that are important, like you cannot get through in that short amount of time. So I was kind of curious to how you think about accuracy versus brevity on these platforms.
It's tough. Some content cannot be put into a short. Like you want to, you have to, if you really want to, you would have to create some sort of piece of content that encourages people so much to go watch the lengthy video. But you can't really talk about it in depth. At the same time, with that Tylenol video, the video that I posted on YouTube was about 12 minutes in length. And in order to put that same video, or I refilmed a few sections of it, for short form content, I had to be really careful. What information am I going to choose to omit? What information is very valuable? What information is going to make people keep watching for the first few seconds, which will tell the algorithm to show this to more and more people? Because ultimately, if after the first second people swipe away, it doesn't matter how effective my messaging is, because no one's going to see it. That is one of the craziest things that I find about social media. I can make the most amazing YouTube video in the world, but if I make a terrible thumbnail or a terrible title that doesn't perhaps match what's in the video, no one's going to see it even though I have 14 million subscribers. And when I say no one, it might get a few 10,000 views here and there because of the following. But big picture, if you want to reach millions of people and go past your subscribers, past what your initial set of audience that you want to reach, you have to think about how engaging, how relatable, how entertaining your medical and science content is. And that's very unusual for us. We're not trained in that way in the healthcare sphere.
Yeah. Is there any other audience questions? Alex?
Hi, real quickly. I'm Alexander. I manage engagement at STAT. I'm just curious, since you're watching other creators, who do you think is getting it right and who do you think is not doing a great job?
Well, I would go back in time and say before this current administration, the CDC and FDA weren't getting it right. How many times would I scroll past the CDC post with 3 likes on it? It's literally the equivalent of not doing it. Like, save time, save the money, go do something else, because if 3 people are seeing it, you're just not doing anything valuable. And I think one of the messages that my professors really hammered into me in my education was whenever you do anything in healthcare, reassess, 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 because good intentions can have bad outcomes. And in a scenario like that, when the CDC was spending money to reach people and not getting anything back for it, the bad outcome is that you're losing money, having no influence, and you're allowing misinformation to keep spreading. So I think bad examples would be the big health agencies. Good examples, I look at creators who are not even in the health space. You look at someone like Jimmy Donaldson, Mr. Beast. Perhaps your kids know him. He makes viral content. He understands what it takes to get people to watch a video, to click on it, and then spend an enormous amount of time watching it. So I look at what these creators who are out of the medical space are doing and trying to figure out how do we crowbar some scientific education into these topics so that people are saying, wow, I normally watch Mr. Beast, but I could totally watch a Dr. Mike video because I'm getting all that entertainment plus a little bit of knowledge. And I think parents in the room would probably be happy to see that happen as well.
So what do you think the future looks like for people who value providing accurate and authoritative health and science communication?
It's so hard to say. I think that the future is bright. I was at Harvard last week speaking to students, and I saw that they had a class specifically made for this, meaning dense scientific communication, understanding how to make it relatable, learning what works, studying, watching back footage. One of their assignments before I came up was to watch a podcast that I did with a Dr. Elizabeth Potter, who recently challenged United Healthcare, and write what worked, what didn't work about the interview. That is how you learn. And before in my healthcare education, there was never any emphasis put on that. There was no value placed on the doctors that were communicating best with their patients. It was always who got the best scores, who was the bright student in the class. But more and more what I realized was it was the best communicators that knew the information solid, but the best communicators were the ones that were having the most clinical success. And I feel like now that situation is basically on steroids, where the best communicators are not just great in the clinic, but they're also very valuable in the social media landscape. So I hope these future bright minds of Harvard and other institutions can lead the way in this regard.
And we're almost at time, but there's a lot of people in this room that have, whether they're in academia or they're research institutions or industry, that have an interest in communicating accurate, authoritative health and science information to the public. And if you could just leave people here with one last takeaway about like what you think how to be most effective at that, what would that be?
I think the message there is think human first. And that message has really resonated with me and done wonders for my career, whether I was treating a patient in my exam room or I was thinking about what content I should make or how I should open a video. I think about what a human would do, not what the science shows or what the data shows. What does the human want to hear? And a lot of times, especially when you're having a debate with people who disagree with you, I recently sat surrounded by 20 anti-vaxxers on a big platform to debate vaccines for 3 hours. What I learned very clearly from these encounters is that these people have had interactions with healthcare that were terrible. They've seen bad things happen, that if you just lived their life and were exposed only to their anecdotes, you'd also be vaccine skeptical. So what I realized is a lot of content, a lot of messaging needs to be thought through a human-first lens, which could mean validating people's beliefs, validating people's misbeliefs, saying that it's okay to misbelieve something and allow them space to be wrong. Allow space for yourself to be wrong, which means taking chances. These days, with short-form content, it's easier than ever to start making content, of wide variety of content, and just seeing what works. Allow the algorithm to figure out what works and what is most successful, and guide you in that process.
Dr. Mike, thanks so much.
Thank you.
Please welcome to the stage CEO of Boston Globe Media, Linda Henry.
Hi, everyone. Thank you for being here today. This has been terrific. What a great gathering and what amazing, insightful conversations we've been having. So thank you all for being here in Boston at the epicenter of life sciences innovation. This year marks a decade of STAT. It was launched 10 years ago. John, my husband, myself, and Rick Burke, the executive editor, got together with this incredible dream that we could bring together the smartest and most capable life sciences journalists from around the country here to tell the incredible story of what's going on in your industry in a way that is smart, accessible, and actually moves the needle. And what you've been seeing on stage here today are so many of these incredible journalists, and they've had such an impact. So I just want to give a quick thank you and applause to the incredible STAT team that's been here together. And over the past decade, it's amazing the impact that STAT's had, which is reverberated throughout the world of health and medicine. So in addition to really being the right place at the right time to really reflect the real news of what was going on with a global pandemic, we also have been there for the science-shaking directives of a new president, the emergence of a transformative new class of obesity drugs, the rapid adoption of AI in healthcare. We've investigated the needless deaths caused by the scourges of opioids and hepatitis C, uncovered questionable business practices of corporate behemoths, and revealed how a scientific cabal may have delayed progress in the search for Alzheimer's treatments. This is just some of what STAT has been really leading the way on. And with this incredible, unrelenting news cycle that we're all living in, STAT's mission could not be more important. When it comes to health and medicine, the changes that are happening in DC are more relevant to our daily lives than ever before. It's affecting the prices of the drugs that we take, the research that we're able to do, the potential of medical innovation to change what we think is possible, and of course, the very direction of healthcare companies, other institutions, and the country itself. STAT covers it all with the intelligence and perspective that our readers need, and these subscribers are responding in record numbers. STAT really continues to push the boundaries of its journalism with an ever-popular weekly podcast called The Read Out Loud, a newer First Opinion podcast, which is inspired by STAT's longstanding opinion column of the same name. STAT has led the way on initiatives that foster community with programs like the STAT Wonderkins, STAT Madness, which continue to recognize the unheralded heroes in cutting-edge research in science and medicine. Last night, we actually had the Status 50 list, which is 50 influential people who are shaping the future of health and life sciences. And after lunch today, you'll hear from a few members of this year's class of Wonderkins, which was just announced today. And I love this program so much because it's really telling the story of the future and where we're going in the industry. And if I can just brag for one more minute, the success of STAT's world-class journalism can be seen in the incredible recognition that this journalism is getting. So we had 2 Pulitzer Prize finalists, countless awards that include just last week, we got a Gerald Loeb Award for Lev Feature's series on how the U.S. has stumbled in fighting the opioid epidemic. STAT won the National Magazine Award for General Excellence, multiple Edward R. Murrow Awards, George Polk Awards, In our world, these are the Oscars and the Emmys, showing the world-class journalism that STAT is being recognized for each and every year. Which brings me to all of you. We could not do this without you. So thank you for being here and being part of this. If you are a STAT subscriber already, thank you. If you're newer to STAT, I encourage you to subscribe, bring your organization along. You'll get an incredible amount of emails, subscriber-only newsletters, like Adam Furstein's really popular and insightful biotech scorecard. Gatherings like this with influential voices across the life sciences community are what shape the future of medicine's biggest breakthroughs. We are thrilled to have you here with us today, and I encourage you to keep being part of other STAT events. They do an incredible job here. We have a Hallmark Summits in New York, San Francisco, or at industry events across the country. STAT's mission is to give you indispensable insights and exclusive stories on the technologies, the personalities, the power brokers, and political forces driving massive changes in the science industry and a revolution in human health, and also to be a catalytic convener for the industry, to bring you together so you can have these conversations and the inspiration and insight to share. These are the stories and the connections that matter to all of us. So thank you so much for being part of our first decade of STAT, and we can't wait to see what we do together in our next decade. Thank you for having me.
I'm just here to say lunch. Thank you, Linda. This is how this works. Right outside this room, in both the Harborside and Cityside rooms, there are buffets set up for you to enjoy. These are the same rooms where those of you who are here for breakfast enjoyed breakfast. Head through the hallway to access either room. Lunch is great. Programming will resume here in about 80 minutes at 2:10 p.m. You want to be back here. Thank you so much to all of you for being here.
Please take your seats. Our presentation will begin in 5 minutes. Thank you. Please take your seats. Our presentation is about to begin. Thank you. Please take your seats. Our presentation is about to begin. Thank you.
People could find their seats. If there are people out there that you want to drag in, go grab them. We're going to start real soon. All right, welcome back from lunch, everyone. Was it good?
Yeah? For our next session, we have an awesome demo of Wi-Fi-enabled tech to track patients. Please welcome to the stage Dina Katabi, the Thon and Nicole Pham Professor of Electrical Engineering and Computer Science at MIT, and to moderate, Stat Health Tech correspondent Katie Palmer.
Hi, everyone. Hope you enjoyed lunch. Dina, thanks so much for joining us. We're super excited to see your technology in action. I think we have enough people here. I first wanted to throw it to everyone here and just see a show of hands. Who's wearing a wearable of any sort right now? We're wearing a watch, at least half it looks like. I've got my watch on. So Dina, your technology is essentially, the idea is to grab some of the same kinds of health metrics and potentially some additional ones, but instead of having to wear physical hardware to pull that information from thin air.
Yes.
How?
So basically, our bodies are full of water. So when wireless signal, like Wi-Fi, for example, hits our body, it bounces off. But it bounces with all of our, or not all, but a variety of our physiological metrics. And from that, with AI, we can analyze the wireless signal in the environment and to get a variety of physiological metrics from thin air. No wearables, no questions asked.
So what metrics are we talking about that we can get right now?
Yeah, so I actually gonna use the help of some slides just to highlight. So this is an office at MIT that we have, and this is my student. And you see a big arrow. So the wireless device is not even in the same room because we know we can reach to wireless signals through walls. So as he walks, look at the red dot on the floor plan. This is where the device thinks that he is walking now or where he's standing. As you can see, it can track him. No wearables, no cell phones, no wristband, nothing.
He's not wearing a little tracker device or anything. This is just
no.
walking.
I mean, for all practical reasons, he might not even know that we are tracking him from the adjacent office behind the wall.
Tricky.
We'll come back to this privacy issue, I think, later. So, but movements is not the only thing that we can track. So when we go to sleep, our brain waves change and we enter different sleep stages. So here, for example, you see our device and transmit very low power wireless signal, analyzes the reflection using AI, and spits out something called a sleep hypnogram. So it will tell you as your brain is moving between different sleep stages, light sleep, deep sleep, REM, and so on, and without putting any electrodes or wearables on the person. And actually, it is very highly accurate. We compare it to the gold standard. More accurate than the wearables who publish their results.
Yeah, and I've had to do one of those sleep studies before. That is not fun.
Oh, yeah, it's not fun. Yeah, and here you see someone sitting like you guys, and we are extracting their inhales, exhales. He was asked to hold his breath, so you see now his chest is stable, and you see the signal also stays at a steady level because he inhaled, he did not exhale. Everything that we do, we compare to the gold standard. And this respiratory signal actually has more than respiration that you can see here. So these are the inhales, these are the exhales, and these small blips that you see on the signal actually, they are his heartbeats. So even the pulsing of your blood leave signatures on the wireless signal.
So an incredible amount of data you can extract here. Can we see it operate in real time a little bit?
We can, but let me show you one thing that I'm very, very excited about, the last thing, because this is very new, the EEG. Your brain waves, EEG, during sleep. So here you see the first row is the EEG was putting electrode on someone's brain, and the second row is our Gen AI generating that same EEG signal, brain waves. And we tried it on many, many people, like thousands of people, and it works very well.
So it sounds like we're building successive layers of interpretation off of the core physiological metrics, but even those are based on interpretation of the Wi-Fi signals bouncing off a human being's
Yes,
body?
yes. And we can show you a demo, of course, like we can't show you sleep and like EEG during sleep and stuff like this, but we got the motion demo that hopefully we can show you. So maybe we can move to the demo. Let's see whether we can get it on the screen. Okay, so this is very similar to the demo, video demo that you showed. I showed at the beginning. Just the colors are different. Maybe I can ask Shichao to move around so that you can see that we can track him as he's moving.
So the white box over there is the actual device.
Yeah. So the white box is the device. We kind of emulating a home environment, just 2 chairs. This is a very small home. And now he's sitting on the chair. It knows that if he moves, you see the divider. So we try to like have something so that he can go behind occlusion and walls. So we cannot erect a wall here, but we have a divider. So you can see when he sits on the chair behind the occlusion, the device can still see him. But I wanted also to get you more than, this is an older result, as I said. I want to show you, Katie, the newer one. So Shichao, do you want to like show the new stuff that we can do? So not only we can get him as a dot on a floor map, we can now get a full skeleton with just wireless signal. So you can see when he moves and how like he's moving, now he's sitting. He's not just a dot on a floor map.
Can I give it a try?
Sure.
Right. I want to be a mind block person. Or Minecraft block person. There I am. So just to be totally clear, I'm not wearing anything on my body. He wasn't either. And can there be multiple people in a home?
Yeah, Shichao, maybe you can enter a bit again just to show multiple people at the same time. So
there we are.
Here he is. There is a bit of delay just because we are connecting everything here. So the changes in the the screen and the wires and all of that stuff. While in the home, we can do the stuff online.
So what kind of detail of movement is actually useful to like collect and interpret from the home?
So, I mean, think again of like diseases like Parkinson, ALS, and like many of the motor related diseases, motion conditions. So imagine being able to detect every single movement of your patient in their home and how it's changing across time longitudinally, whether based on the disease or based on a new treatment. And that is for motion, but you can say the same thing about everything that we talk. So sleep, for example, imagine like getting every night of sleep. And you said like you did a sleep study, so you are putting electrodes on people's head and body, but imagine if you don't have to do that every single night as a sleep test. Mm-hmm. Imagine like the new thing, EEG. In fact, one of the things that we are able to do is, so I think people think about AI as one model, but AI is a system. So we have wireless signal, then we have AI, and from that we get physiological signals. But after that, we have another AI model that can now ask questions like, does this person have Parkinson or Alzheimer? So these are questions that we can answer. In fact, the Parkinson paper we published already and we have very high accuracy. And in fact, we have some indication that it might be even possible to detect it before the motor symptoms, like before diagnosis. Yeah. But now we have even other things, like not just diseases. Like we can ask, does this person take antidepressant? And we have very high accuracy. I can tell from your sleep with wireless signal whether you take antidepressant or not.
I have a hard time believing that.
Believe me, this is actually like tested. In fact, not just antidepressant. Now we can see that we can have like a foundation model. Like if it's the same way as you think about text with ChatGPT. Like you have a foundation model that does multiple tasks. You can think about a model that analyzes physiological signals for multiple tasks. Like, does the person have Parkinson? Do they have Alzheimer? Do they have CHF? Do they have COPD? Like, are they taking antidepressant? Are they taking anticholinergic drugs? Are they taking insulin? And all of these things can be coming from the same model.
Mm-hmm. So I remember when this technology was first demonstrated in your papers. I was a hardcore science reporter at the time. It's a physics breakthrough. So can you tell me how you came to realize its potential applications in healthcare and start developing it indeed as a product in healthcare?
Yeah, so I'm a computer scientist. So my first interaction actually with medicine was when I come from Syria. So in Syria, in fact, I come from a family of all doctors, medical doctors. So in Syria, there is no pre-med. And if you are a topper in your class, you go to med school because this is where like all of the toppers go. So I went to med school, and then it's like, oh, I cannot just live without physics and math and like these kind of things. So I decided to switch, to the disappointment of my whole family, and switched to computer science and electrical engineering. In Syria at the time, it was electrical engineering. And, but I never really thought that it is different. It's really basically with science and math, and just like physiology is also like some type of science. Everything is connected. But so I started working on this at MIT as a faculty when at the beginning, we were like, oh, like X-ray vision, you can. And then it's like my grandfather, who lives in Belgium, at the time lived in Belgium alone, and the whole family does not even live in the same country, fell and went to the hospital. And after coming back home, I had like, okay, so how is he? Is he gonna fall again? Nobody is gonna know about it. In fact, when he fell, it was just by luck that the person who helps him, the helper, came within a few hours. But if she was not scheduled on that day to come, he would have been like on the floor for the next day, or who knows what's gonna happen. So that just connected me back to medicine, and made my family so happy that I'm coming back, seeing the light at the end of the tunnel.
So what do you see the value of this technology as compared to other ambient sensors that we can imagine in healthcare? You could have camera-based sensors, you could have wearable-based sensors.
Yeah, so I think, I mean, first of all, I think we should use as many sensors as we can, and information is always power, and it empowers both the patient and the doctor. But cameras are very unlikely to be used in people's homes unless there is really something that you cannot address otherwise, because the privacy level that you have to sacrifice with camera is huge. I mean, like people like move with like different types of clothing inside their home. You don't really want, if I try like with my grandfather saying like, I'm gonna put camera in your home, or my parents, it's just like, it's not just rejection, it's rejection of me and not just a camera. So that's one thing. So being able to get everything without cameras. And in our case, there is no camera, there is no microphone, there is nothing. And even the whole system, in fact, does not store any information about like the name or the address of the person. It's just like a random ID that is associated with the device, and everything is encrypted. Everything is like after like informed consent. We tell people what we can monitor, and even people can say, "Oh, I want you to monitor my sleep and my EEG, but I don't want you to monitor my movements," and that's fine. We can activate, deactivate certain things.
So where is this currently being deployed in healthcare or life sciences?
So where is it deployed? So we started working very early on with pharmaceutical companies. In fact, with Parkinson is one of the very early diseases that we worked on, and we showed that we can have statistically significant progression marker as well as like detection markers in Parkinson, and that they can reduce the cost and the duration of clinical trial and sample size a lot. But now, as we are speaking, like over the last few years, we have worked in almost like so many diseases, from Parkinson to Alzheimer to ALS, terrible, terrible disease. I just like really cannot tell you, like just watching the data, just you feel bad. To immune diseases, like we have been talking just like in the previous session about immune diseases like IBD and lupus, Sjogren, all of that stuff. And to kids who have eczema and like scratching, another thing that we can get is scratching. So it has been applied in many diseases, monitored many people. Like I think by now we have like over maybe like 30 different clinical trials across more than 10 different diseases.
In what phase?
So phase 1B, phase 2, and phase 4.
Wow.
And of course, natural history as well.
Do we have a question, or are we thinking about a question? If anybody has any questions, let us know. Yes? Okay, great.
Hello, Øystein Brekk, Eli Lilly. I was just wondering, how do you differentiate between patient and caregiver in the same home? I mean, the patient maybe never leaves, but if they do, how do you know it's them coming back?
Sure. So when we work in clinical trials, we have additionally a machine learning model that is trained on identifying the patient from every other person or even animal in the home. So it's patient, non-patients for that particular participant. As you would guess, many of these clinical trials, there are multiple people in the home. It's not actually just in the home. Many of these people have bed partners, and we can distinguish and zoom in on the participant and separate it from the bed partner. Some people have dog partners, and so it still works.
So the extension to that, I mean, you can still get a lot of information about a person, even though there are no cameras. So how do you deal with the privacy concerns that a patient might have or a trial participant?
Yeah, I think that's very important because every clinical trial, every study that we do, independent whether it's a clinical trial or just a research study, so it goes through there is a protocol and it goes through IRB approval and informed consent so that the patient or the participant knows exactly what is happening, knows exactly how the data is going to be used. Our kind of policy is that the person owns their data, and they can decide who they want to share it with. But even though they own their data, but also it is our responsibility to make sure that we use technology to protect privacy. So as I said, we never store any information in our system about the individuals themselves. Also, everything is securely encrypted, both in transit and in rest.
Mm-hmm. What do you think the next metric is going to be that you're able to build on top of all of these streams of data?
So I'm really very excited now that we have these collection of physiological metrics, that it opens up understanding diseases and drugs at a level that was not feasible before. Because basically, again, I was saying that we detect Parkinson. I'm sure most of you guys, or even all of you probably, thought that we are detecting the motion, the tremor, and from that we can detect Parkinson. That's not true. We detect Parkinson from the respiratory signal. And during sleep, and people when they sleep, Parkinson people when they sleep, they have no tremor. So it's really what we are able to detect are different pathway and changes, I mean, not pathway, but the changes how these different diseases and different pathways impact the physiological manifestation at the level that your best neurologist cannot look at the breathing signal and say, oh, this person has Parkinson's, this person has Alzheimer's. They cannot do this. So now is imagine if we can put these devices in the home of people, and then very early on being able to detect, oh, this person seems like his signal is moving toward something that looks like Parkinson. Can we react earlier? Maybe if we react earlier with exercise, we can change the trajectory of the disease altogether. Or even we can, there are now, people are working on vaccine for neurodegenerative diseases. Can we, analyze, people, analyze the physiological signal, understand them at, So one of the things that I found very exciting, for example, of course, most of you guys probably, or all of you know about the Genome Project. So imagine the Phenome Project. Imagine really understanding the different physiological manifestation and the phenome of the patients and even the healthy individual and how we can differentiate that. That's very interesting, can change the way we think about diseases.
I think we have another question in the audience.
Yeah.
Hi, my name's Emily Stein. I'm at an early stage biotech company called Gaia. First, congratulations, this is amazing.
Thank you.
My question was, so when you go through the process, you mentioned antidepressants, for example, or you're talking about Parkinson's and picking up on those respiratory signals. Do you have to train the model for each of those different things? So you have a data set that's like, all of these people we know are taking antidepressants, so you're picking up on something that's a biomarker no one even knew to look for, or what's the process like?
Yeah, so it's both. I mean, we have a bit of a hunch. So for example, let's take the antidepressant as an example. So this is now, we haven't published it yet, but the manuscript is like out, and we hopefully looking for good feedback. So, but basically what we do is first we know that we are getting physiological signals. We know from the literature that antidepressants change your sleep. It's just that basically the literature look at population changes, not as an equal one. So it's saying, oh yeah, so it's people who are taking antidepressant, they have delayed REM, for example. Okay, and you can separate the two populations. So we're saying, oh, AI can give us more power. Now let's go, and we have these physiological signals because we are able to get them very easily from thin air. We can augment them with data from sleep study, for example, so that we can get a huge population. And now we can train our model from wireless signal or from breathing, nocturnal breathing from wireless signal, or from breathing belt, it doesn't matter to us, to be able to detect that this person is taking antidepressant or not by using some labeled data. But then we can test it on new data from completely different environment, people that it never seen before. And actually, if you put someone on antidepressant, you see that first it's saying, no, he's not taking antidepressant. And then you put someone on antidepressant, and it goes up. And it doesn't go up immediately to the maximum value. It goes up to like a bit, and like then it gradually ramps up, which tells you that there is also the interaction. We know that the antidepressant is not immediately effective. So it detected as we put the person on the antidepressant, but it keeps increasing the response as more evidence are available.
I think this speaks to the challenge that you probably have as you're selling to pharma partners. Like, they need to have a certain level of evidence to believe that, you know, as you're building on top of these physiological metrics, that your interpretation of them is going to give them something true about the participants in their trials. So how do you deal with that challenge? How do you present that data and that evidence for them?
Yeah. So I mean, everything that we do. I mean, one thing is to, like, for our physiological metric is easy because you can like put someone in a sleep lab, and we have like study in a sleep lab with almost 1,000 individuals, and we can show that what we are getting with our sleep study from the wireless signal is very much what PSG, which is the gold standard, is getting. Or for movement, I mean, basically, you see the person moving, but for actual, like, exact measuring, you put, like, what's called Viken motion capture room. So you can compare, you can validate or against the metrics, like what was called analytical validation. Now, within a disease, now we are looking at, okay, so is this a progression marker, for example? Then we can compare to the gold standard in progression for that disease. So in Parkinson, it's a questionnaire called, unfortunately, it's a questionnaire called the MDS-UPDRS. So we show that we are highly correlated with that, but we can discover the signal much earlier. Same way with ALS, we look at, it's also another questionnaire, unfortunately. So we show that we are highly correlated with that, but we have the ability to discover the signal earlier or more reliably.
I think we have another question.
This is on? Yes. Good afternoon. Thank you so much for your presentation and data. Doris Brown Associates. My question is it possible that this technology could be used without the person knowing that it's going to be used, and where would that data then be stored?
Yeah, so this is, as I said, like privacy and security is like from the beginning, and one of the reasons why we chose to work with pharma, biotech as the beginning is because like it allows us to do everything under a protocol and like informed consent and all of that stuff. But from a technological point of view, because the device understands space, I mean, detect whether the person actually has access to the space. So let's say that I turn this, I mean, there is nothing outside, but like say somebody is like turning it and saying, okay, monitor the neighbor. Now the device is going to, okay, so show me that you have access to that space and start moving in the way that I'm telling you to show that it is you actually who is that person. So technologically, we have mechanisms to ensure also that you cannot use it to monitor people without their own consent.
Hi, I'm Donna Seibert, alongside Medical Ally. Thank you again. This has been great. And you may have answered this, but I was very interested in the sleep study. Have you validated your information against the gold standard sleep studies? And
yeah, so as I said, sleep, we have amazing sleep. We validated multiple times. Our most recent study include 840 individuals from the sleep lab, Mass General Hospital across, I'm not sure where the river is, but from Mass General Hospital. Not only we validated that it is highly accurate and achieves an accuracy that is comparable to the differences between two sleep technicians looking at the same data from the same person at the same time, they can have some disagreement, these small disagreements. Yeah, this is light, this is deep. So that is the level of differences is comparable. But also we show that it works across ages, across genders, different demographics, pre-existing condition, whether somebody has pulmonary condition, neurological condition, heart condition. So we have tested against the gold standard and in a variety of pre-existing conditions.
Yeah, still a lot more to learn. Thank you for the great questions, and thank you so much again for joining us, Dina. This is really awesome.
Thanks you very much. Thanks, everyone. Thanks, Kate.
Thank you, Dina and Katie. That was amazing. I didn't know that the Minecraft guy would end up in a healthcare application someday. Next, we're going to do another poll question. So before we jump into the rest of the jam-packed afternoon, we have our second poll of the day, sponsored by Zeno Group. We would like for everybody to please take out your phones and open the Bizzabo app. If you don't have it downloaded yet, you can use the QR code that should be up here on the screen from the App Store and quickly make a profile. You're going to open the app, and then on the bottom right of the screen, you will see 3 dots and the More button. From there, you'll select Polls and click on poll question number 2, which I'm doing just now. And for those of you who are turning in virtually with the browser version of Bizzabo, you should see the link to participate in the poll appear in the chat window to the right of your live stream. Our second poll question of the day is, what strategy do you use the most to build trust and transparency with healthcare providers? The options are written communications, emails or publications, in-person engagement, social media, or a prescriber website. Again, that is, what strategy do you use the most to build trust and transparency with healthcare providers? So everybody, please take a second to vote. I'm voting myself. All right, it looks like we have a pretty clear winner. I see 66% of you said in-person communications, and the follow-up at about 28% is written communications, emails, and publications. So it seems like most people value that in-person connection. That's great. Thank you all so much for participating. We will do one more poll question today, so everybody keep the Bizzabo app open and ready. Thanks so much. Bye.
For this sponsored conversation, please welcome to the stage Senior Vice President and Chief Technology and Digital Officer at the University of Texas, MD Anderson Cancer Center, David Jaffray, and to moderate, Stat Managing Editor, Gideon Gill.
Welcome, David, to the Stat Summit.
It's good to be here, Gideon.
Yeah, so at MD Anderson, your clinicians see roughly 187,000 patients with more than 400 types of cancer every year. Each individual's care generates more than 2 gigabytes of data annually. Imaging, test results, notes, treatment plans, you name it. And now you're using that data to build an Institute for Data Science and Oncology. Tell us about that.
Well, it's a really important mission we have, as you're all aware. Our objective is to make cancer history. If we could close our doors, we'd all be happy because we wouldn't need it anymore, and I'm sure you'd be happy about that too. But the reality is that we need to work faster to bring cancer under control. And as you all are aware, with the advances in AI and computing, data is a powerful tool to try to understand how we can better take care of our patients, improve their experience, but also drive towards new cures. And one of the things we've focused on substantially in the organization is how we manage that data. How do we steward that data? How do we capture as much as possible? You just saw in the previous demo the amount of data that's growing remarkably. How do we make sure we can use that to help answer important questions? So we built a new architecture that really flows the data within the organization and curates that and making sure it's the state of the art. The applications are numerous, of course. We can look at experience, we can look at efficiency, we can get more patients in and out of the OR, which is important because the demand is endless. But simultaneously, we can partner with pharmaceutical companies, for example. And we do that in a way that's quite rigorous. We like to make sure that we're tackling an important problem. Our clinicians get engaged. They understand the science issues, they understand the clinical issues. And then we think about how can we use those data assets to try to understand how we can move a cure forward faster. And this for us has been central. New organizations or organizations today that need to think about how they steward their data, how they build that trust. For us, patients and trust are together. They come to us for care. And we think about how we're going to manage their data and steward it as part of that trust architecture. And so building this capacity has been really key. And it's remarkable to see the engagement it drives. New young faculty realize they can come work with us using these data to extract insights and help answer questions that are important for cancer.
Can you give me an example of how you're using the data?
So our clinicians often are involved in clinical trials. They'll see a new therapeutic advance, and they see right in front of them patients that respond and don't respond. But the rules for the trial are set. The decision on go, no go, based on the outcome. But they see nuances. They see subtle differences. Some patients respond and some patients don't. So hypotheses are forming in their heads about why that is the case. Simultaneously, the pharmaceutical companies already put a ton of money into getting that drug into the clinic to a certain state. Maybe successful, maybe not. And so you take those insights and you make this understanding of the investment in data and how we collect it and curate it. And that in combination with state-of-the-art data science methods that let us simulate out possible outcomes for different trials, different cohorts of patients, puts together a very powerful combination where we can identify, for example, patients with specific mutations that should have responded but didn't respond. Why? What other biomarker could we identify? What other genetic characteristic or proteomic marker would tell us which patients would respond and which ones wouldn't? And this is not just like an activity we want to do helter-skelter. We really want to think about how we industrialize this activity in partnership with our patients because they're all compelled. They don't want someone else to go through the same treatment. So how do we learn from one patient so that we can treat the next ones better? I think the most interesting part of this is this mind shift around how we manage data. Today, healthcare data is used in a lot of different ways. We're very particular. We really think that the stewardship role is really important, even to the point where we're working with a little startup company that's looking at dynamic consent technology that will allow the patients to say, yes, please use our data in this way because we're compelled by the case, by the mission. And moving forward, I think organizations need to think about that, how they manage and steward data internally, and how do we engage with patients to make sure that they're okay with how we're using data to advance a cause like cancer.
Yeah. So when we talked a few weeks ago, you spoke about your hopes for creating digital twins for every patient. How would that work, and why are you so excited about their potential?
And we have about 1.7 million visits a year at MD Anderson, and every one of those visits is loaded with a decision, some kind of decision, either the patient's decision, the family sometimes involved in the decision, the provider involved in the decision. It's full of decisions. And so we ask ourselves, how can we use the data we have and the data we're going to collect next, but how do we use that data to help inform every decision we make? And we believe that computational approaches, advances in data science, including things like machine learning and AI, but other more mechanistic modeling approaches will give that decision-making an edge. It doesn't have to be 100%. It's like house odds. If you get 2% or 3% improvements in your decision, your predictability, that compounds. And so our vision through the Institute for Data Science and Oncology, which we've been developing over the past couple of years, is to bring data science to every single decision we make. And it's pervasive. It's everywhere from in the laboratory, understanding, do we advance this drug or this drug, to in a clinic, is this the right care for you or is this the right care for you? Or even at the societal level, how do we bring more data to prevention and other forms of approaches to reduce cancer? So let's go back to the digital twins. That was the question, right? Well, the digital twin paradigm is this idea that we can customize those models to each individual patient. We're all very similar. You can buy glasses off the rack, you can buy shirts off the rack. But there's enough variation from each of us that if we're going to inform those decisions, we need to capture data specific to individual patients. And then we can assemble models based on previous treatments for patients to try to understand what's a likely trajectory of this patient given this course of care. Is that likely to be successful, or should we consider an alternative? And increasingly, I think that this computational modeling paradigm is going to become expected. We have rich data sets. Like I saw in the previous session, we know so much more about patients, and we can inform these models. They may be very basic to start with. We have some great examples where we see prostate cancer patients, prostate-specific antigen, or PSA, climbing gradually through time. Wouldn't it be great to have a computational model that suggests, now you should go for another test because it's climbing, or based on that, we've seen a fall in your PSA, you're good. Rather than just have a series of tests, put together a computational model or a digital twin that could be used in consultation between the patient and the provider. Of course, there's uncertainty. We'll have to teach people about uncertainty in decision-making. But I think humans are more than capable of doing that.
Yeah. So your background is in physics and radiation oncology, where you worked with algorithms to guide the administration of therapy. How does that shape your thinking about the need for thoughtful adoption of AI technologies? And looking forward, what are you most excited about?
AI is pretty exciting. I've been working in basements of cancer centers coding since 1986. That's almost 40 years. This is the bucking bronco of technology, if there ever was one. It just comes in all different forms and shapes. It's quite disturbing and simultaneously very exciting. Healthcare organizations aren't built to manage computation. They're barely built to manage tech stacks. You're going to have thousands of AI algorithms running in your organization if you don't already today. Who's watching them? Who knows whether the data going in is the same as it was when it was trained? Who's understanding that if you had this algorithm running and you decided it was good and you put another algorithm in front of it, is this one still doing what it's supposed to be doing? We don't have that skill set. The companies won't bring you that skill set. The FDA is not going to bring you that skill set. Organizations have to prepare themselves to stewarding thousands of algorithms and thoughtful assembly of those algorithms so we can be confident and patients can be safe. That skill set is not in the IT teams. The physicians are very good at decision making. They're trained on it, but they're not necessarily trained at managing thousands of decision making or inference solutions within healthcare. So we're looking at how do we build a maturity in organizations to be able to actually capitalize on the substantial promise that's been presented to us from AI. And that's a lift. We'll have to get there, though. Have to invest a little bit to get the return of AI in healthcare. But the conversations are going in the right direction. People are realizing. And if you're with healthcare organizations, you should be asking, how do you govern your AI? We have human resources. Where is the Department of Robotic Resources that's going to help us understand how these work together? So what am I most excited about? I'm most excited about the fact that this technology is going to accelerate cures. Super exciting. I was just with a little startup, raised $300 million in Series A. I guess that's what I mean by little startup nowadays. They're bringing robots together with AI for generating hypotheses in collaboration with humans. And we'll see this combination of synthetic hypotheses by computers being shared with humans and thought through and executed with incredible efficiency through robotic methods. And that's going to just produce a huge opportunity for very important cures that will be able to allow us to close the doors at MD Anderson. That's exciting.
Well, thank you very much. Appreciate you being here.
It's great. Thanks, everybody.
everyone, up next, we are going to meet some members of this year's class of STAT Wonderkins. What is a Wonderkin, you ask? Well, each year, STAT selects a class of the best and brightest young minds in science who are shaping the future of medicine and health as we know it. These early career scientists have completed their degrees and are just starting their careers, but are not in a place where they're running their own research labs just yet. Our hope is that in recognizing these young scientists, we can help to pave their way to even bigger breakthroughs in the future. Today, we'll be joined by 3 members of the 2025 class of STAT Wonderkins. Please welcome to the stage Dr. Sydney Ramirez, Associate Physician at the University of California, San Diego, and instructor in the Crotty Laboratory at La Jolla Institute for Immunology. Connor Tu, Postdoctoral Fellow at MIT, and Dr. Tomer Yaron-Barir, MD, PhD, Pediatrics Resident at Boston Children's Hospital and Harvard Medical School, Research Scientist at Dana-Farber Cancer Institute, and co-founder and CSO of Mirandus Therapeutics. Thank you 3 for being here today. We're really excited about this. Just to start to hear more from you all about your research, Sydney, could you tell us about the research that you're currently doing to examine immune responses in the upper respiratory airway?
Okay, I'll give you a little bit of background first so it's not straight into the upper airway. I did my PhD in a molecular virology lab, so I worked with coronaviruses and other RNA viruses even before the pandemic. And then started examining host responses early in the pandemic. We looked at blood and immune memory, so B cells and T cells, different parts of the immune response that can fight off SARS-CoV-2 infection and protect us, and also the part of the immune response that responds to vaccines. And then we started asking, what about the upper airways? Like, we actually get infected here. Shouldn't we care about immune responses at the site of infection? And so pivoted to looking at immune responses in the upper airway using nasal swab samples to actually collect cells from people's noses and nasal cavity.
And what did that most recent paper that you released show?
So yeah, in 2024, we published in Nature, and we actually published a lot of amazing findings. So we published things that people had not previously known, but just assumed were present. So we found that there were a number of different types of B cells and T cells in the upper airway, and you can actually use a swab to remove enough of these cells to study them in depth using even single-cell transcriptomics and very advanced technologies, spectral flow cytometry. And we also showed things that were, again, kind of common sense in the medical world, but not really explored, where we showed that people have what we call the adenoids or the pharyngeal tonsils, so secondary lymphoid structures in the back of the nose. And these are where we have germinal centers. They're basically places where B cells and T cells work together, and the T cells educate the B cells to make the best antibody response possible. And it turns out, as adults, we do still have this tissue. They don't disappear when we're in our teens. And even though there's a decline with aging, it's still something that can provide protection even in people over age 60. So we found a number of different lines of evidence that point to the fact that we have immune responses in our upper airway. They are protective against respiratory viruses like SARS-CoV-2.
Thank you. Connor, will you tell us about the work that you're doing in gene editing, and specifically about the potential for non-viral large sequence integration in particular?
Yeah, absolutely. So, so I just completed my PhD this past spring, and so what I'll tell you about is a little bit of my PhD research. What it focused on was really developing new types of genome editing tools, and one of them really was around this prevailing problem in the genome editing field, which you heard a little bit about this morning, is that most genetic diseases are caused by dozens, hundreds, or thousands of distinct genetic variants that differ between different patients and patient populations. So this imposes a lot of economic and regulatory bottlenecks, which are being worked on, as you heard about. But an alternative that a lot of the people in the gene editing field have been working towards is, can we develop gene editing approaches that are more universal or off the shelf? Can we develop one editor that can then cure everybody for that all genetic variation for a given genetic disease? And so this is what I've been trying to work towards. And so one of these approaches is something called targeted DNA integration, and it's the ability to insert a healthy copy of a gene in a very precise location of the genome. And there's been a lot of work by various industrial labs and academic groups to come up with the molecular machines to do this type of insertion reaction, but they've all kind of been bottlenecked at one key stage non-virally, which is that most of these technologies have a component, namely the DNA message we're trying to get into the cells, that is extremely immunogenic if you deliver it non-virally. And so in work that we hope to report before the end of this year, fingers crossed, we describe a technology that we call install, which basically bypasses mammalian DNA innate immunity to, for the first time, be able to deliver and integrate kilobase-sized DNA sequences in the human genome. And hopefully, we're hopeful that this type of approach will then be able to benefit the largest number of patients possible, but with the fewest number of genome editing compositions. And so, yeah, hopefully, I'm excited for that space, and hopefully we start seeing some interesting results therapeutically.
It's very exciting. Tomer, can you tell us about Mirandus Therapeutics and how you're hoping with this startup you can tackle cancer resistance?
Yeah, so I think Mirandus Therapeutics is based on the research that we have done in the past 8 years, which is around a special group of proteins called protein kinases, which are 500 proteins that are governing the intracellular signaling in the cell. So I'm sure many of you are familiar with this concept with central dogma of biology, that DNA becomes RNA, becomes a protein. But then after a protein is being made, there's a whole set of chemical modifications that are being used in order to control the signaling in the cell. Because you don't have time to make a protein every time you need it. Your body needs to respond in a matter of seconds to external stimuli. And the most abundant chemical modification in the cell is something called phosphorylation, adding a phosphate group. And that modification can turn on and off proteins, can control cell proliferation. And when that communication going wrong, we get diseases. And at the top of the list will be cancer. So in our research, what we did is we built an atlas of the human kinome, of these 500 protein kinases, that what they do is really controlling that whole network of cell signaling, phosphorylation-based cell signaling, and using that atlas that we built experimentally and then build a computational system, we are able to really map the signaling of the cell, which is really right before the actual phenotype. It's very related to what we actually see, and also very prevalent in cancer. So protein kinases are the most targetable class of proteins in cancer, and using that, we are understanding the targets that we can then target using techniques such as Connor is developing. And so in Mirandus Therapeutics, what we do is we are trying to actually augment existing therapies in order to overcome resistance, which is the biggest problem in cancer. And the way that we do it is we study how the tumor, how the cancer evolves over time given a therapy. We try to find an orthogonal pathway that the tumor is exploiting to develop a therapeutic for that. And then when we combine it early on with the original one, same concept of broad-spectrum antibiotics, we are trying to prevent the cancer from even arising by looking at the pathway that it would exploit in order to overcome the existing therapy.
That's amazing. Thank you. Sydney, you mentioned that you were looking at coronaviruses before the COVID-19 pandemic hit, and you were an author on a very influential paper about the role of T cells in immune response that was actually held up by Fauci during a congressional testimony, and you can see your name up there. And in order to conduct that research, you had to go to hospitals and collect samples from patients and their family members. And I was wondering if you could tell us a little bit more about what happened and what that was like.
Sure. So when I was in graduate school and I was studying coronaviruses, I oftentimes when I was presenting was asked, why don't you study a more clinically relevant virus? Why don't you study HIV? Why don't you study influenza? Coronaviruses at that time were largely believed to only cause common colds. And so those of us who were in the field, and it was a very small field at that point, said, well, what about SARS, the original SARS? What about MERS? These are very dangerous viruses. They cause severe disease in humans. And so I didn't actually expect to come back to studying coronaviruses when I was doing my postdoctoral research because there was so little interest when I did my graduate school research. But unfortunately, when I was doing my first year of clinical fellowship training in infectious diseases, we saw the writing on the wall. We saw cases starting in China and then Italy and other parts of the world. And so I was supposed to start doing research in Shane's lab in July of 2020. And instead, the lockdown was coming. And I joined his lab in March of 2020, just before the lockdown in California. And because I was working on the clinical side and had access to patients who had SARS-CoV-2 infections, who had COVID-19, and I had colleagues who had IRB protocols that allow you to collect human samples like blood from patients, I asked, can I be a part of this work? And actually the initial response was, you're a clinical fellow, focus on your clinical training. So that was a little bit frustrating, but thankfully by joining the laboratory, we were able to make a concerted effort and really get things going between UC San Diego and the La Jolla Institute for Immunology. And I found that I was spending more time with patients as a researcher than I was as a clinician, because we basically didn't have anything to offer anyone at the time. And so more often than not, I was the only provider who would go into a patient's room that day wearing full protective gear. But it was to talk to them about research and to tell them, we want to find out, can the immune response actually be protective? There was some fear that maybe it was actually a bad player or a bad actor and making disease worse at that point in time. And so telling people, we need samples, we need your help to try and figure out what's going on, and if our body's actually able to fight off this virus, do we have a chance of developing vaccines against this virus? And thankfully, people were very, very understanding and wanted to support the research. And so I found that I was spending a lot of time with people, collecting their samples. We, because of the lockdown, didn't have clinical coordinators, didn't have other staff, so often it was me going to the patient's room, explaining the research, consenting them, collecting samples, taking it back to the lab, processing it sometimes even. And yeah, within 2 months of joining the lab, we had that first paper that Tony Fauci waved in front of Congress, and we were very, very glad to be able to tell people that the good news was average case of COVID-19, people recover, they make strong and durable immune responses, and they are protective responses. They're not something that's going to cause more severe disease. And so then going forward, it was much nicer to be able to talk to people and say, we're making progress, we're learning more, thank you for your help, you really are contributing to the science. And again, people were just incredibly generous with the support of the research.
Your Wonderkin recommender who wrote your letter said that you were able to collect the most samples, and he said that he thought that was because of your skill to be able to work really empathetically with patients and their families. And I was wondering if you could tell us about that part of it.
Yeah, again, it was a challenging time. I think it helped that in the field of infectious diseases, people had gone through the AIDS epidemic. And so unfortunately, for a lot of people who survived through that or who were familiar with that era of history, it was a similar time. There was a lot of fear, a lot of isolation for people. But for those of us who were aware of the fact that as long as we wore the right gear, we would be protected, I didn't feel afraid. And so I felt like I was comfortable going into patients' rooms, holding their hands, talking to them, hearing their stories. And I think that's really all that it takes, is just spending time with someone, getting to know them, understanding what's important to them. And even if you have nothing to offer other than the hope that most of us recover, that seemed to be enough to make people feel better and to just know that they weren't alone and to not feel isolated. So I like to try and practice empathetic patient care regardless of the situation. Sometimes we have more to offer patients, but just being there is oftentimes enough to at least let everyone know that we're on the same team.
Thank you, Sydney. Connor, I wanted to ask you, so my colleague Jason Mast has written about a lot of the regulatory and commercial challenges that are facing gene editing companies. The title of that story is "The CRISPR Companies Are Not Okay." I hope many of you in the audience have read it, and if not, you really should. But I was wondering, how are you thinking about those challenges as you strategize your own startup and the science that you're chasing?
Yeah, so I think the two strategies that I'm very optimistic about, and I think a lot of the field is now kind of approaching, is a little bit what you heard from Kieran this morning too, is either taking a mutation-specific approach, and the science and technologies are basically there, and how can we streamline and platformize the editor development and the regulatory and economic bottlenecks to maybe open up that type of therapeutic strategy. And like I described before, I think especially in industry, a lot of people are focused on these more mutation-agnostic approaches so that it's more feasibly economically and resource-wise, and just how much time and investment you're putting into each program. So I think there's merit to both, and I think the fact that we're exploring both options as a field is extremely promising. And as somebody who's hopefully going to translate and commercialize technologies, I think I'm looking at both sectors, but I'm really excited about the concepts of these more universal approaches that can maybe bypass some of the bottlenecks that we're seeing with mutation-specific therapies.
The other fun fact about Connor is that he's an internationally ranked ultramarathon runner, in addition to being a researcher. And he said that unfortunately, your brain does not turn off when you run. So I also had to ask what role running plays for you in your life as a scientist.
Yeah, big role. I'm not sure which one feeds what. Yeah, I mean, I think on all my long training runs, when I did my undergrad at Berkeley, they would have the Tamalpa Headlands there and all the Berkeley Hills there. We'd go out on like 6-hour runs just in my headspace or try to explain scientific concepts to the buddy running next to me. But he wouldn't be a biologist or a science person at all, but I think he learned a little bit. But it not only gives me kind of a headspace, but also, I think, room to troubleshoot all my experiments for one. That's what happens on a lot of my easier runs. And then to also come up with and try to invent these new technologies. So the two technologies that really came out of my PhD, one called click editing, and the other one is what I just described called install. Those both came out of runs that quickly jotted down and drew out the DNA strands and how they would work when I got home. And so that's been a productive way to not only escape, but become more innovative in terms of the ideas that I'm trying to pursue.
That's awesome. Maybe I'll have to try running now, but I'm not sure if an ultramarathon runner is in my future.
If anybody else is out there, I'm in Boston, so I can go on a group run.
Tomer, you are a computational biologist, and when I asked you what the greatest challenge in your field is, you said that it was AI and big data. Can you tell us why that is for you?
Yeah, I think that's a question that we've been dealing with for a long time now. And I think the biggest challenge is that, in my view, the research that we do now, this year, like our generation, is really different from the research that our PIs that trained us did. And I'll explain what I mean by that. Back in the 60s, 70s, 80s, the research was really hypothesis-driven. You were really trying to work really hard to come up with the right experiment that will give you the answer. And since then, around the 2000s, we had two big revolutions, or two new eras. We have the big data era, that we have high throughput, we can do a lot of data at once. And then we have the data accessibility that, together with the AI tools, allows you to get a lot of patterns / results. And I think the challenge is that the mindset has to shift from trying to really look at your hypothesis and come up with an experiment into how do I dig my results from the data. And the big challenge is, I feel that back in the days, you really were chasing your hypothesis and you tried to do more experiments, etc. And nowadays, you're just chasing data. And very often what happens is that when you have so much data and so much sophisticated tools, you will get something. You will get a nice figure, you'll make a nice result, you'll get a good target. But that's probably why most of the targets fail. That's why most of the, we keep saying like why trials fail in phase 1, phase 2, toxicity, patient population, you name it. But I think the number one reason is just because the mechanism is wrong. Right? Like the target is wrong. We are looking at the wrong thing. It's very tempting to follow up something that came up from big data. If you have big data, garbage in, garbage out, or not even garbage in, garbage out. Just a lot of data in, something will come out. So I think that the big challenge for me, or I think for us, all of us in this generation, is to shift from just chasing data and chasing results that we get one by one until we stumble upon something right, to go back, take a step back and say, how do I leverage this big data, this AI, these hidden patterns into hypothesis-driven research in a way that eventually would really allow us to get the most important thing, which is the mechanism of nature that we try to discover.
Thank you. In the few minutes we have left, I wanted to check if there's any audience questions in the room for our Wonderkins. All right, well, I have another question for you all. And that is, the theme of the STAT Summit is the next 10 years. And I was hoping to ask each of you to be so bold as to pull out your crystal ball and tell us your prediction for the future. Where do you see yourself in 10 years, and what do you think that your corner of science will look like in 10 years' time?
Okay. Well, immunology is a relatively new science, and it started predominantly looking at mice. And I think at this point in time, we've recognized that mice are great for doing mechanistic work, but they don't look like humans. So I think in the next 10 years, immunology really needs to pivot and focus more on looking directly at human samples and human immunology. And especially, like I was saying earlier, we need to look at mucosal sites and places where infection, where environmental allergens, where the disease process happens. And so if we want to study a disease that impacts, the upper airway, we need to look at the upper airway, not just in the blood. Because very often things are localized to the tissue, and so what we understand about tissue level responses in one site won't look like tissue responses in another site. So we can't just say blood looks like my nose, blood looks like my lungs, blood looks like my uterus. We have to look at different tissues across the human body, males, females, really tease out what's going on. And then we have to find a way to take that technology, all those advances that we made in mice, like reporter mouse models, lineage tracing models, how can we actually do the same sorts of studies in humans in a safe and ethical manner so that we can figure out mechanistically how human immune cells respond and not just base everything on mice.
And where do you see yourself?
And I see myself running my own laboratory focused on mucosal immunology, human mucosal immunology, and trying to answer some of these challenging questions and really push the field forward.
Thank you.
Yeah, on my end, I think we're already seeing the initial really promising results from various companies and labs for genome editing approaches. So I would hope in 10 years, all of those are hopefully on the market, and we'll see. And that there will be a lot more clinical trials in genome editing therapies. I think if you ask anybody in the field also, that delivery is obviously, as it is with most genetic medicines, one of the biggest bottlenecks as well. So can we deliver, especially perhaps non-virally, to extrahepatic tissues? I think there's some promising data starting to be developed, maybe in the lung in vivo HSCs, maybe, but hopefully there's novel and innovative solutions there. And then I think that hopefully as cases like more KJs are cured and more of these genome editing therapies are in the clinic, that those cases will really pave the way for public sentiment in terms of that these therapies can be developed safely and to be efficacious, and hopefully much more quickly as regulatory things are revised and reimbursement schemes are hopefully innovated. And then scientifically, again, I think that hopefully these universal approaches will also be much more validated and hopefully starting to have really therapeutic effects too. So I think among all those things, hopefully in 10 years, the genome editing field will be much more mature and we'll have things in the clinic and commercially successful as well. Oh, and then I guess where I'll be in the next 10, well, I'll be in 10 years, much older. Hopefully still running. We'll see with these knees now because I've been running way too much. But yeah, I'm kind of in an interim period. Hopefully I'm really interested in biotech entrepreneurship. So I would hope that there's some commercial interest or success there. And I'm also interested in staying at least connected to academia because I think certain indications are really well suited to maybe a company setting, but others, especially very rare diseases, are maybe more suited towards an academic type of route. And I think both types of avenues are really important to be connected with and involved in to see the technologies make the biggest difference. And so hopefully there's, among that kind of fuzzy answer, there's hopefully will be more panned out within 10 years, but yes.
I'll start from the personal aspects. Actually, I think the greatest project of my life started 3.5 years ago when I met this beautiful woman sitting in the audience that since then somehow agreed to be my wife. And we had our most important publication maybe a year ago when our daughter was born. So in 10 years from now, I really think that it gives you proportions, and I hopefully will have extended family even more and happy and healthy. Regarding the personal life, the professional aspect, I think the pace of things moving around is so fast that every time somebody asks me that, my answer is, I hope I'll do something that I can't even tell you about nowadays because it still doesn't exist. So I have no idea what I'm going to do in 10 years. I do think that more broadly, what interests me personally is the way that I think the scientific world with industry and pharma and biotech is headed towards personalized medicine, but I think maybe in a different way than we are thinking about right now. I think nowadays, I said that AI is a challenge, but also it's an opportunity for collaborations. Nowadays, I think that people try to use AI to create their own personalized medicine. It's a very competitive field. Like, how do we get medicine for one person? In my view, personalized medicine, it's impossible in the current regulatory system, regardless of the political environment, the system itself. And we talked about it a lot, about approving an individualized therapy. I think that the personalized medicine is going to be a puzzle where we have a lot of approved therapies from different entities, different companies. And the personalized thing will be the actual puzzle that we build from each other. And that's what we do with Marendis. The idea of Marendis is not to say we are trying to develop a better drug than others. It's to say we're taking something that exists and we know it fails eventually, because everything fails eventually. How can we solve it? How can we come back and to partner, to collaborate with these big companies, with these big pharmas that have the resources and the knowledge to be able to make the puzzle more complete? And I think that's where we're headed, to a place where we are leveraging these AI technologies in order to benefit the patient in a collaborative way rather than in a competitive way.
Thank you all so much, and congratulations on joining the 2025 class of Stat Wonderkins. With that, we are at time. Thank you.
Please welcome to the stage Genentech Senior Vice President, External Affairs, Joy Russell, and to moderate this sponsored conversation, Stat co-founder and executive editor, Rick Burke.
Hi, everyone. Hi, Joy. Aren't those Wonderkins inspiring every year? If you should look at the full list of them, but like, that's our future, right? And we're going to talk about your future in Genentech's 50th year. But first, Joy, can you talk a little about your past and how you're kind of not a lifer, but you've been at Genentech for a long time, which says something about how much you like it or they like you or whatever. So talk about your role there and your history.
I'm hoping it's a mix of both. They like me and I like them. Well, thank you, and happy birthday to Stat. 10 years.
10 years.
So as Rick said, my name is Joy Russell, and I've had the pleasure to work at Genentech for 23 years. I started as a teenager, overused joke. But I've seen Genentech evolve in so many different ways. But what I started in was state government in Harrisburg, Pennsylvania. I had a wonderful opportunity to work for Governor Tom Ridge, then a moderate, and a wonderful guy. And really got bit by the intersection of policy and healthcare, and really thinking about how we can change what people think healthcare should be. And so one of our clients at the time was Roche and Genentech, and I had the pleasure of working for Roche. And then soon we became one company.
This is a crazy thing to throw in here, but wasn't Tom Ridge talked about as a vice presidential candidate?
Yes.
That's how, for Bush or something?
Yes, for Bush.
That was like a whole different
for 43,
era.
Yes.
Yeah.
He was the first Homeland Security.
Yeah, apologies for the sort of going back. So you've been doing policy and sort of legislative affairs and all those things for many years. Obviously, things are different now in our nation's capital. How are you coping with it? How are you equipped for it? Is it anything you've seen before?
I'm not sure we've used the word coping before, Rick, right? Yeah. So I think one of the things we're noticing at Genentech is in external affairs, you have to think about the whole patient and the whole experience in your community. And so we're really, I think, at an intersection of policy, of access, of considering the right to operate in this Congress and in this administration. So as a major employer across this country, as a US manufacturer, we really believe in thinking about what access is now and in the future. So when you show us the Wonderkins, we're thinking about that innovation pipeline, not just in our workforce development, but in how we deliver medicine to patients.
What's your strategy or approach in getting innovation and pushing through progress in science, given all the government bureaucracy and politics and so forth?
Well, I would be lying if I didn't say that this year was a dynamic year.
Is dynamic really the right word?
Again, It's also like when we use unprecedented. How many more times can all of you reporters use unprecedented times? But it has been an unprecedented time. And I think what we all have to do is sort of ride the wave of what is going to really serve us well to have a healthier America. We're going into midterm elections, as we all know, in this room for sure. We have 36 governors up. We have a real important time at both as an innovator, as a science company, and as a company that wants the decisions to be made more locally for patients.
When I think about innovation, I think nationally, but you all really focus a lot on, as you said, state and local. Why is that, and how can you have innovation at the local level? How does that work?
Well, we're in a state right now of innovation. And my California friends, where we're US located, would debate it where, but isn't it great to say that we are in a state, and the Commonwealth is a major employer of pharmaceuticals? 1 in 3 or 4, I know one of you will keep me honest, in healthcare, whether you work at a hospital, whether you work anywhere in the healthcare ecosystem, it's happening here in Boston. But in a blink of an eye, it can also go away. And so I know that we have elected officials, not just in this state, in Speaker Mariano and Governor Healy and so many that believe in the state of innovation here. But you have to foster that through your academic institution relationships, private partnerships, and public partnerships, and understanding that these are decisions that are being made locally, but yet do we serve everyone in our locality? And that's something that Genentech is very committed to.
What are some of the issues you're most concerned about on the local level?
Access to care. And I think, this is a time when this is about underserved, as well as those that may be going without. And that might be because their insurance coverage was lost. This has been a turbulent year, but it's been a turbulent couple years, not just for patients, but the communities that serve them, their families going back and being needed in the home to take care. We have a crisis of a time. And I think that it was coming no matter what administration, but we have to face it with smart and responsible solutions.
When I think about the Genentech 50th anniversary, I think about the origin story. It's like 2 men walk in a bar or something. Like, can you talk about that and sort of what Genentech was and what the possibilities are for the future?
Sure. In 2026, next year, we'll turn 50. And for those that have turned 50, you know that it can be a bit of an identity crisis of who we are and where we've been, or where we want to go. And I think like you and Stat, we're thinking of innovation. Science is going to tell the story ultimately. That in our last 50 years, we're about a banker and a scientist walking into a bar or in their garage and truly making miracles happen. And so if you haven't heard about 2 scientists walk into a bar, Google it. It's a pretty cool story about the people that still really believe in innovation and research, and how do you get that to patients. And so just recently I was on campus and I saw our founder, Herb Boyer, who's still walking around and on campus, and I think it's an incredible inspiration of the commitment that he had to the unmet need, to science and development.
Was he one of the 2 that walked in the bar?
Yes.
Wow. So what's in store for the, what are you all doing for your 50th anniversary?
We're gonna party. We're gonna hopefully be still around. We are going to continue to look at neurological diseases, oncology. Genentech is not a household name. And that's not what we're striving to be. But our pipeline necessitates us to be a more consumer-looking company, because decisions are made by patients and their communities. And so alongside, obviously, prescribers and thinking about what kind of care and they're going to need for the future for their families and access to it. Because just because it's available doesn't mean it's accessible.
When you say Genentech's not a household name, certainly everyone here knows Genentech. What's a takeaway that you want people here who know of Genentech but don't really, what do you want them to know that they might not know right now about
Yeah, let me clarify. We're not a household name because we make really, really products for really sick people, and we have for 50 years. If you were on a Genentech medicine, you were probably really sick, chronically ill. And so I used to laugh when I was one of those consultants, and when the client would say, we're going to cure cancer. And we have sustained lives for many, many, many years. And I'm really proud to have worked here and dedicate the time of that. But I think whether it's in the unmet need, in making cures more accessible, that's what we're in the business for life for.
So when I ask like what people might not know about Genentech, it's just how successful you have been in extending lives, or is it?
I think it's the commitment to science in the future. It's building and looking at social determinants of health, thinking about the access barriers that we have now. What will that look like in 3 years? What will that look like in 7 years? And so that's where the intersection of policy has to come in. People are advocating for patients in ways now that are from NIH funding to be thinking about that outcome that we may not achieve. And so that's the role that Genentech needs to play. Can we look around the corner?
One last question, just looking around the corner. We've heard on the stage today, some people are optimistic, some people are dealing with a lot of questions about public communication about health and science and where we're going. What's your optimism level on a scale of 1 to 10?
Rick, when you have a name like Joy, you have to be optimistic. I mean, I have to be optimistic. I wake up every day thinking about how policy affects people. That's kind of boring, but it's true. But I have to be optimistic. I have boys, I have kids, and whether you have children or not, I think this is that unprecedented existential crisis time. And whether you're turning 50 or 10, we have to meet our moment.
So it sounds like a 10 on the optimism scale.
Yeah, I have to be.
Thank you, Joy.
You're welcome.
Appreciate it.
Our next session, we are excited to be joined by a speaker who took a pretty long flight to get here and will be able to tell us why China is becoming biotech's newest global hotspot. Please help me welcome to the stage Michelle Jia, founder, chairwoman, president, and CEO of Akeso Biopharma, and to moderate, stat biotech startups and venture capital reporter Allison DeAngelis.
Well, Catherine mentioned that it took you a long flight to get here today, but it's also taken quite a bit of time for Akeso to get to where it is today. The company was founded back, I think, in 2012, well over a decade ago. And I think I want to start off, for those in the audience who don't know, about a year ago, one of a drug that had been developed by Akeso called ivonescimab. There was clinical trial data that really kind of grabbed the industry's attention that showed that ivonescimab had bested Keytruda, the leader in cancer treatment these days. And then in the wake of this, there was a lot of attention on the US company that Akeso had partnered with, had licensed the drug to. You at Akeso, your team was getting a lot of attention, and you told my colleagues at the time about your ambitions, all these things that you wanted to do. How has the last year been for you? Have you been able to meet those ambitions? Have you had to kind of change them or alter them at all?
Thank you, Allison. It's my pleasure to be here. So it's worth of traveling. Talking about last year, that's amazing happening. We ran this head-to-head phase 3 trial for first-line PD-L1 positive non-small cell lung cancer and against the Keytruda. And we got positive results. And that, you know, in a way, because we were being developed ivonescimab since year 2015, and we started our clinical studies year 2019. And so all the way to that point, you know, we got the phase 1 data, phase 2 data, and then phase 3. To us, it's like kind of like a naturally happening. But of course, the results is very, very exciting and draw the many, many attentions from public, from you know, the peers, industry peers, everything. That's absolutely amazing and exciting. But on the other hand, I think, to be honest, like, to me, it's like nothing changed, because our ultimate goal is to, you know, have a develop good product to meet a medical need, and for the world, for China, for US, and everywhere else. So to us, to be honest, I think we do the same thing. I don't know if I answered your question, but I think we do the same thing still.
What about like moving forward and building off of that success? Akeso, I think at the time, or last year you had, I mean, somewhere along the lines of like 50 drugs in development. Yes. Like Akeso is actually a much bigger company than I would have anticipated. So what's next for you guys after, you know, kind of besting PD-1s?
Yeah, I guess to answer your question, I would, you know, like review a little bit back about our background, right? So we started year 2012. Like, give an example of myself. I was trained in UK and US, and I joined a biotech company in US and joined a pharma company in US as well. And with many years of training, back to, actually that was year 2008. There was an opportunity that my friends told me, okay, look, pharma companies, we are working on the drug development, the cost is so expensive. And if we do some, you know, like a CRO company to help out, and that's a good business. And talk about CRO, you want to have a cost cut, expense cut. And maybe we should utilize Chinese talent pool. So we should do it in China. That was, you know, like original thought to go back to China to do something. So I started to work on this company called Crown Bioscience. Maybe many of you know about that. Very pioneer CRO company for animal studies. And so I started from, you know, together with my colleagues, build the lab and train the talents from graduate school. And then we did I think we did such a good job to recruit many farmer customers from outside of China. All of a sudden, I realized, okay, look, we don't have any customers from China at the moment. So I started to realize, you know, after review the background about the new drug in China, how long does it take to be able to launch in the market from US? And I was shocked. Okay, this is a good opportunity. We have a good talent pool. And we have the people like myself trained in US and gained a lot of experience. And now it's time to do something in China. So that was very original thought. And obviously, as you can see today, how many biotech companies in China, I think that's a lot of them. It's the same kind of like same background, same education, same original thought, go back to China to do something. So then, you know, I just follow my kind of thought to see more about this. And nowadays, people all by the sudden say, wow, this is so many good results come from China. And, you know, like last year, people talk about 40% of the deals actually made between Chinese biotech and the global farmers. How come that happened? And I think this is not like happened overnight. It's really like we started year 2012. From there, when we started, the funding for biotech industry in China is not much. Actually, it's very little. But since year 2015, the funding to biotech in China dramatically increased until year 2021. As we know, we can see the rate between Chinese funding, US funding for biotech companies, Chinese increase. But to be honest, if we see this year 2024, the data, actually Chinese funding dropped a lot. US dropped up. But no matter what, it's like, I want to say that those results from China is not like happened within one day. It's actually happened with many years' work there.
So wait, then Michelle, I'm curious, when you look forward at what Akeso is going to be doing in the future, so much of the reason why we're talking about Akeso and ivonescimab has been the work that you've done with Summit. And how does that factor into the future? How much of Akeso's business plans is going out and partnering with a pharma company or with a venture capital firm to build a new startup?
Yeah, very good question. So then this is back to the original thought when we build Akeso. So we have an idea that the product comes from science, and the science need technology to support, to facilitate. So when we started our company, Akeso, we determined to build the platform to improve the technology, to help the science transform to product. I think up until now, we still have this idea. We believe we need to use technology to facilitate science transform to product. Therefore, you can see our ivonescimab, and we have another beautiful molecule, cadonilimab, and many others. We all kind of follow this direction. We need to utilize technology, good technology, improve the technology. And then talking about the future of Akeso development, I think we want to still follow that route, meaning that, for example, ivonescimab, we have a very deep belief that potentially it could change the industry standard of immuno-oncology field. And then is that it? I don't think so. For example, right now, ADC is another hot topic, and a lot of companies working on that. And to us, we think the combination of bispecific with ADC absolutely is going to be a good future. However, you have to have a good bispecific, good ADC. So what is the good ADC? We believe then we need to use good technology to improve the safety profile of the ADC. So that's the Akeso is doing right now. We have our first two ADC molecules already in the clinical trial. We use the good technology. We think it's a potentially good technology, and we try to make it better ADC. And from that point of view, for the future, combo of the ADC plus the bispecific antibodies, I think will give us a better results.
I want to talk a little bit about results. There was, on top of the clinical trial data that we've already referenced from last year, both Akeso and Summit Therapeutics, the US company that licensed ivonescimab, have continued to release data over the last couple of years and over the last year. And there was some data that came out last month that seemed to show, some people looked at the data and said, well, it looks like there's a difference between the Chinese patients and the Western patients, which is a big question mark that I think that a lot of people in the US and European pharmaceutical industry have about taking drugs from China and licensing them. They're like, well, how are they going to perform in Western patients? When you saw that data last month, what did you think?
First of all, I think that fits to our expectation. That trial, actually, if I'm going to talk about it, it takes another an hour to explain why we came to that trial, why we speed up our process. We got a deal with Summit earlier year 2023. At that moment, ivonescimab was tested in Caucasian patients in Australia, but not in US. So then think about it. We managed, Akeso and Summit, especially Summit, managed to have the US physicians, clinicians, use that drugs in the short period of time without phase 1 confidence, without phase 2 confidence. I think that's a big success already. And then after all, because of the complication of the trial design, so then first of all, I want to emphasize the consistency, the clinical meaningful benefit, the trend is there. People cannot just superficially say, oh, okay, it's superficial, my opinion.
Yeah.
So then, but takes time to explain, maybe it's not a good time today. However, I think that fulfilled our purpose. Within a short period of time, we see a safe efficacy molecule in US patients.
I wonder if there's anything that you take away from the experience over the last year or two that anybody here in the audience who works in the pharmaceutical industry can kind of use, or any tips they can use about how to translate data from China, data from partners that are in other countries, and bring it into more Caucasian patients, more Western patients.
Yeah, Alison, I thought about this question for many times already because I kind of constantly heard this question. So I feel like, okay, let's step back a little bit. Let's think about any product these days, global product. A pharma company run a phase 3 registration trial. Do they run it's called MRCT, multiple regional clinical studies, right?
Oh,
they do, right?
Multiple regions, yeah.
Yeah, multiple regions. Absolutely include the Chinese clinical centers. And we see so many trials, we see the consistency between the Chinese data and the US data for different molecules. So the question is, to me, is it's not about the quality issue, it's about this science of this product, if it's really behave the same for Caucasian patients or for Asian patients. That's the question we need to answer. We are on the road to answer it. But in terms of the quality, think about it. ICH, International Council for Harmonization, for the requirements of technical.
Yeah, this is a big international kind of coalition that sets standards for clinical trials.
Chinese become standing members since year 2017 and become management members since year 2018. And the founding member is the US, Japan, and the EU. And now we have a Singapore, we have a Brazil, we have a South Korea, we have a Chinese, right? So from Chinese join this so-called council organization a long time ago. And so in terms of follow the good guideline from ICH, Chinese is doing it, right? Not just a GCP, good clinical practice, but also good manufacturing practice guideline. And like a company like us, we follow it. And so from the quality point of view, I think in the past 10 years, the quality in China is getting better and better as somewhere else. So now come back to okay, if ivonescimab would behave same for Caucasian patients or Asian patients, that's a scientific question. And we do have a belief, though, because ivonescimab is a function for both PD-1 and bivalent anti-VEGF. And we all know Keytruda, Nivo, and other PD-1 molecules, they work very well for Caucasian patients and also for Asian patients. Ivonescimab is the same. So we have a big belief, and we are on the road to generate more and more data to showing that the consistency.
I want to talk about the data that's coming, but I believe that we have an audience question. Catherine, where'd you go?
That's good. We have a question already.
Hi there. Josiah Seal. I work with Clarivate. I was wondering, based on your experience, could you compare and contrast your experience with the regulatory environment in China versus the EU/Europe, and also maybe the way it's evolved since you got going in 2013?
When
you began, sort of how has it changed over time?
Okay.
Also.
Okay, I think my opinion in terms of the quality expectation, it should be always the same. Then you talk about, let's say, year 2013, that was Chinese regulatory revolution, if I call policy change, started from year 2015. And before that, that's also much more before we joined ICH, right? To be honest, I cannot comment on that. Sorry, I cannot totally comment on that because at that moment, the drug in my company not in the clinical studies yet. But since year 2015 and since year 2017, China joined the ICH, and also I think Chinese regulatory authority. They also, invite FDA personnel or previous FDA personnel join the regulatory agency, and they did a fantastic job to really, follow the international standards and getting better and better, and absolutely now, follow the ICH. The quality, I think, expectation should be the same.
I wonder, Michelle, in a little bit of a similar vein, looking back at your career, starting off training in the UK and then going back to China, that's a very classic story. There's so many of your colleagues in the Chinese biopharmaceutical industry did the same. And as you were just expressing, the Chinese biotech industry has really been evolving and growing and changing during this last decade. And I'm curious, just with the way that kind of different international governments are approaching, this area. I know that the US, there's a lot of policy makers who are, kind of concerned about that issue, about people coming from China to train and then going back to China. Do you think if you had, a child who is interested in getting into the drug industry, or if you yourself were restarting, would you take the same path, studying in the UK, studying in the US, and then going back to China? Or do you think that actually the pros and cons in the different regions have changed at this point?
That's a tough one. First of all, I have a daughter who is 23 years old, and she works for Wall Street in New York.
Okay.
I don't know. I think these days, plus AI, everything changed so fast. Follow your heart with the purpose, and any choice is okay to me. And you talk about the policy and environment. Somehow, I think healthcare industry is a so beautiful industry. It's mankind, humanity industry. Really, to me, it's we shouldn't have a borderline, that kind of thing. And think about it. The other day, actually, I was thinking about people talking about the deal between Chinese biotech and U.S. pharma or European pharma. And I was thinking, hey, come on, the biotech industry and pharma industry, the collaboration is always there, right? You don't expect every single biotech company, grow up, become a big pharma. Some of them would, right? But then collaboration is always there. Deal is always there. And then you think about, like if company is in Boston and farmer is in, let's say, Boston as well, maybe the only difference to a Boston biotech and a Chinese biotech is just the distance. One is maybe 200 kilometers away. The other one maybe 5,000 kilometers away. But then the consequence is all the same. Biotech give a good product to farmer. Farmer utilize their power, their capability, scale up capability, commercial capability to make the compound benefit globally. As actually I answer that gentleman's question again, you talk about the quality of the quality of the clinical study. I actually touched base a little bit, saying that pharma company nowadays all run clinical studies in China as well. They use the same clinical centers as us. If they want to have their drug, the pharma's drug, approved, launched in China, they need a Chinese authority approval as well. So from this point of view, it's all harmonized to me.
I wanted to really quickly, we have another audience question, really fast, Michelle. This coming weekend, Akeso is going to be presenting new data at ESMO. You are getting back on a plane and flying out for ESMO. What can you tell us about what you've learned from that data?
Back to the first question you're asking, what's about last year? I kind of feel like, even not just in US, but in China as well, the investors are so fascinating about our results. They go so much detail about any single number, any single of our explanation about our trial. So we become I'm proud of that. We become so much kind of put education effort to the audience of the investors, telling them, what's the hazard ratio, what's the PFS, what's the OS, any single small numbers, what does that mean? So it's funny. Back to the ESMO. So we are going to have a very big presentation about the first line squamous non-small cell lung cancer. Again, it's our ivonescimab combo with chemo against the PD-1 plus chemo. And obviously, we announced already it's a positive results for PFS. So I'm sure it's going to be a lot of question comes out again, but it's the honor, I think. That means people are interested in that, right?
Yeah. Well, and you had mentioned offline that you feel like you've had to build a lot of trust. That's part of what you're doing right now is building trust, and is that part of why you feel like you have to explain the results so much and everybody's examining them very closely?
Yeah. So explain the data is one way of build the trust. I think the most important way is really you do the good job, you do a good quality job, and you got a good product. Time will tell.
Okay. All right, let's go to that last audience question before we run out the clock.
Hi, Michelle. Thank you so much. My name is Tomer. Yeah. I wanted to ask, you spoke about the difference between the pharma industry in China and the US, and that's obviously being affected by the regulation and the system. But when it comes to innovation, the very just small group that try to think about a problem, what do you think? I think it's rooted in the cultural differences as well. What do you think is one thing that we can learn in the US, maybe Western, innovative biotech field from you guys?
No pressure, Michelle.
One thing maybe everyone knows about it is Chinese people are the people working very hard. We don't have a weekend. That's the learning. You don't take a rest.
Okay, wait, I have to ask because John Maraganore was on stage earlier today, and he talked about the work that we're seeing come out of China and some of this licensing data that you had mentioned as being a Sputnik moment for the U.S. In reference to during the Cold War and the space race, the Russians, kind of sending the first object into space.
Sorry, your question again? Sorry.
Oh, do you agree that this is not only an important moment for China, but for the U.S. drug industry?
You mean the biotech company?
Do you think that, kind of going off of Tomer's question, that the U.S. biotech industry might need to kind of take a look and say, there are some things that we might need to change right now?
I do have a belief. A lot of things when you get into you need to wait until the turning point, and then things will change. So it's like a Chinese sentence about people talk, we talk about when a boat reaches to the bridge, it will turn. So it will naturally happen if give you an example. The funding I'm talking about between year 2015 to year 2021, the funding to biotech in China is dramatically increased, and then now it's dropped significantly. That's why a lot of deals happening. To be honest, the people need. Biotech in China needs money, right? And then when it's get into the bottom, I believe the fund will raise again. And U.S. is the same. If people start to, I think U.S. Famous in innovation and still the most powerful innovation harbor in the world. So I'm not worried about that. I think things will come and will come.
Okay. Well, everyone, please thank me in joining and in thanking Michelle for taking the time to talk to us.
Thank you.
I told you this would go fast.
That's very fast.
That was such a great conversation. Thank you, Michelle and Allison. And with that, we are at our second coffee break of the day. Upstairs in the mezzanine, you will find snacks, drinks, and our sponsor booths. Please go check them out, and we will resume programming here at 4:20.
Please take your seats. Our presentation will begin in 5 minutes. Thank you. Please take your seats. Our presentation is about to begin. Thank you. Please take your seats. Our presentation is about to begin. Thank you.
All right, we're going to get started in a minute. If anybody is out there come in, come sit, come stand on the mezzanine, although if you're up there, I can hear you. The bar is being set up, so it's not long now, but we have 2 amazing sessions coming up, and this one I'm really looking forward to. For our next session, please welcome to the stage Matt Might, professor in the Department of Medicine and director of the Hugh Call Precision Medicine Institute at University of Alabama at Birmingham, and Casey Ross, chief investigative reporter, Data and Technology, STAT. It's going to be a great one.
So we're here sort of celebrating STAT's 10-year anniversary. This is a meaningful session for me, especially because I've been at STAT for 9.5 years, and getting to tell Matt's story and that of his family was probably one of the most meaningful experiences of my time here. So I'm very excited for the conversation. We're going to be talking about artificial intelligence and its use in drug development, discovery, repurposing of drugs. We're going to be talking about its use in diagnostics. And we're going to be talking about that through Matt's story. And Matt is the only person I've ever met that had to learn about the application of those things in order to help his son. When did you realize something was wrong with your son?
I think when Bertrand was 6 months old, I was actually at my first faculty retreat out at the University of Utah as a professor in computer science, and I got a call at the end from his mom, and she said, the pediatrician thinks something is wrong. And it was the start of a 4-year diagnostic odyssey at that point. And at the time, she just said, we don't know what it is. It looks like brain damage, we're not really sure, but something is just not right. He is not in line with his developmental milestones.
And how was the health system sort of prepared to figure that out, figure out what was going on with him and treat him?
I mean, at the time, I'm not really sure it was prepared. This was back in 2007, 2008. We didn't have ready access to things like genomic sequencing. It was a lot of, well, it looks genetic, but all we can do is run a lot of spot genetic tests. So there was a lot of what we called hot potatoing, where you're just kind of thrown from one specialist to another who would say, well, I'm not really sure what this is either.
And so what did you do to help him to figure this out?
So, I spent a lot of time learning genetics because I said, well, if this is a genetic disorder, I need to understand what's going on in the broader space of possibilities. And actually started trying to get him enrolled in clinical trials to do genome sequencing of some kind, and got very lucky that in 2010, Duke opened a clinical trial of exome sequencing, I think one of the first of its kind for kids just like him on these intractable diagnostic odysseys, and that's what it took.
And so how long did it take before you began to get some answers about what was going on and then what you could do? The answers were not great.
Yeah, so at 4 years old, we finally had an answer. It was a long diagnostic odyssey. And it turns out Bertrand was the first case ever discovered of a brand new ultra-rare genetic disease called NGLI1 deficiency. Now, in a weird way, I was actually excited about that because I had an answer finally. But of course, the first thing all the clinicians said was, but this is not actionable. I mean, there's not even a second patient to compare him to. We don't even have a prognosis for you. But yeah, I saw it a little bit differently, I guess.
Well, what is NGLI1? What does it do? What was it? What symptoms were manifesting as a result of this issue?
So in Bertrand's case, severe developmental delay, a movement disorder, epilepsy, and probably on a day-to-day basis, the most annoying and frustrating one was an inability to make tears, or a lacrima. So you wouldn't see him, he would never cry physical tears, and so he would have all sorts of recurrent eye infections from the lack of eye moisture, which was a real struggle to deal with.
And so they tell you your son is an N of 1. You know, there's nobody else like him. He's got this deficiency. There's no way to effectively treat him, handle this. So what do you do now?
My first step was to say, well, I doubt he's really the only one. In fact, I looked in some of the population genetics databases at the time, and I looked at the frequency of what should be these disease-causing alleles like his. I said, well, there's got to be at least a few hundred kids like him out there somewhere in the world. I just need to find them, to network us together, to build a community and make some forward progress in the science. So I actually turned to social media. I wrote a blog post that was very explicitly designed to go viral and rank high in search engine optimization results so that other patients like him, particularly ones that couldn't cry tears, would find this page and then find me. And that's actually what happened. So within a few weeks, I'd found, I think, 2 more patients in Israel. In 1 year, there was 14 patients from around the world that we'd stitched together as a fledgling patient community.
What were those conversations like, meeting these people that didn't know about this illness, didn't know anything about this deficiency, didn't know that there were other people like them? I mean, tell us about the sort of formation of this community.
It was kind of like, meeting family you never knew you had. To this day, it's a very close, this is true for a lot of rare disease communities, I think. It's very close-knit. It really feels like a family when you all get together and you all recognize you've had the same struggles, the same challenges, and really the same goals at that point. Like, everyone in these communities is very aligned on finding treatments and cures.
And so what did you begin to do yourself to sort of help, leverage at this point, given your background, the ability, the skills that you have, you essentially make a career change.
Yeah, for me, it's really the beginning of a pivot. So, right after I got tenure in computer science, I did the last thing I ever thought I would do, which is quit. I literally quit my job and changed professions entirely. I pivoted into academic medicine, and in that role, I've been trying to bring a computational mindset to everything I do in the context of precision medicine, trying to really accelerate and individualize the process of identifying treatments for patients like Bertrand.
And what were some of the successes that you began to have in looking for things that might help him, that may improve his quality of life?
Yeah, so for Bertrand, what's always striking to me is that by understanding the basic biology of the disease, by being able to model it computationally, I could start to make predictions about the other things that could be going on inside of him. And so about 2 years post-diagnosis, I was able to predict that he was likely deficient in a particular metabolite called N-acetylglucosamine. And lo and behold, you can buy this on Amazon. Literally, so like a bag of it shows up on my doorstep. And I think I actually took the entire first bag myself, called that FDA phase 1 safety testing. Because hey, in my household, I am the FDA. That's how this works. Ended up giving it to him, after a lot of ethical wrangling over, is this the right thing to do? Not knowing what it may or may not do, but in his case, the thing that changed most dramatically was his ability to make tears. So he could literally start to cry once he received this. And we ended up doing a lot of follow-on science to this. So one of my collaborators at Utah, Clement Chia, he's a fly geneticist. We built the fly version of the disease. And I don't know if the flies cry when you give them this, but they go from about a 13% survival rate to an 87% survival rate. So we'd really latched onto something fundamental about the biology of the disease with this. But it was all born of the ability to do some very basic, well, I think nowadays we'd call it basic computational modeling of what's happening with the disease.
And so, you're making these discoveries with your son. Your career has changed and done a complete sort of 180. You're leveraging your background in computation and artificial intelligence to do these things. What are you thinking at this point, sort of more broadly about what that journey is making possible? What you're realizing at that point about what you may be able to do, not just for your son, but for other people?
Yeah, so I think really for the past 8 years since I've been running the Institute, I've bet really big on artificial intelligence and precision medicine. I really see artificial intelligence as the democratizing vehicle that allows precision medicine to happen for everybody. Because if you look at what I had to do early on for Bertrand, it was pretty mentally labor intensive to learn all this stuff, to figure out how to do this stuff. And I think now, we can do what took me years, in some cases in days, in terms of the kinds of analyses we can do for individual patients, thanks to advances in AI. And so I really think AI is a democratizing force in precision medicine.
And I mean, you were doing all this, for your son back in, the very modest deep learning era, which we're now through and entirely onto, an entirely new sort of technology, the advances in AI and now generative models. How is that, changing things for the work that you're doing on behalf of, rare and undiagnosed patients?
Yeah, so I think the shift to full generative AI really led to an inflection point in our capabilities. So early on, like 8 years ago, we were using the technology available at the time to mine out the literature so that when we wanted to ask a question, we could try to ask it against the entire published corpus of literature, which at the time was a pretty advanced thing to do. And now, not only is it more or less trivial, but we can do it with a much higher precision so that when we're dealing with a patient, we can say, well, we think that because of this rare variant, we think this is the mechanism to go after. And by the way, there's 6 or 7 published papers or 8 or 9 biomedical data sets that tell us how to go after that mechanism. And allows us to form hypotheses much faster than ever before that are grounded in evidence that we can cite for why we might want to take one particular strategy or another.
And tell us more about how this works at the Precision Medicine Institute. I mean, you're dealing with patients and trying to help them out, people who are coming to you without answers, who are frustrated, who have gone on all these medical odysseys and just don't know where to go, where to turn, what to do. So how do you go about helping them? Maybe you can walk us through, an example of that.
Sure. So we actually have a patient-facing research program. We call it Consultations in Precision Medicine. When patients consent in, we basically say, look, we can't promise we'll be able to do anything, but what we're going to try to do is really understand the biology of this disorder so that hopefully at the end of this, we can generate a research report that will go back to your treating physician, and that may give them new options or new ideas for how to influence care. The thing we try to do most often is identify existing drugs that can be repurposed for that particular condition. And part of that is because, on a practical level, it's probably the only thing right now that scales economically to essentially any case. It's let's find something else that's out there that could work for you. And build up some kind of scientific, mechanistic rationale for why this is a reasonable thing to do. So as an example of one such case, I can mention her name because she's given me permission to multiple times, Sandra Sermoni. She is the mother, I think, the first diagnosed patient of ADMP-driven autism here in the US. And she reached out real early on after my pivot to UAB and said, is there anything you can do for ADMP-driven autism? And here, the question we had to ask of our AI at the time was, is there any way to to increase the expression of ADMP? Because in this case, it's a haploinsufficiency. You've got roughly 50% of your functional ADMP. It's still there, it's just the thermostat's turned down. So our AI tools at the time actually stitched together a path through the cancer literature and said it looks like low-dose ketamine should actually nudge up the expression of ADMP. So fast forward, I think 2 or 3 years later, the ADMP Foundation has actually marshaled the resources to do a clinical trial in New York, and it actually works. So I think 2022, we published the results of that clinical trial and showed really dramatic improvements for all the ADMP patients that went in. I think 13 out of 14 of the things they were looking for showed statistically significant improvements. And also there's a story in STAT that talks about what did this mean for the families. That's one example of the kinds of things we try to do for patients when they reach out.
I wonder, as you're doing this work and you're trying to help these patients and you're producing these recommendations through AI, how you're interfacing with doctors on that who are treating these patients, what that is like, because, I know from talking to you about your journey that when you would kind of suggest things to physicians about the treatment of your own son, it wasn't always easy.
Right. And this is where I think, an important but very real buzzword applies called explainable AI. So from day one, I've never invested any research, anything that wasn't fundamentally explainable AI. And what we've learned is that if you give a physician a recommendation, unless you can trace through an argument that grounds out in the literature, they're not going to take it. And honestly, I don't blame them. I mean, why would they take it if there wasn't some, valid technical reason or scientific reason for why this is a good thing to do? So I think explainability is really the bottleneck in a lot of what we do when it comes to bringing AI into the clinic. Because without explainability, in terms that a frontline physician understands, it's never going to cross that barrier and make a difference. So that's something we always keep front and center, is how do we explain whatever predictions we make to a physician?
Well, your work, like many of the people in this room, your work is largely supported by government funding through ARPA-H, other sources. You're working through the Precision Medicine Institute, trying to advance your work. Tell us what this environment is like for you as you try to navigate and build on what you've.
Sure. I'll try to say nothing that won't get any more funding cut.
If you want to lay down on the couch for this answer,
yeah.
You go ahead.
Yeah, I'll start with the high level. Just say this is a really challenging environment in which to do scientific research as an academic. There's so much uncertainty and volatility injected into the system right now that it's actually crowding out time to do meaningful science. With grants getting canceled, then uncanceled, and funding calls disappearing and then reappearing. I mean, it's really become such a moving target day to day that as a leader in academia right now, I have a hard time steering that ship, not knowing, how much funding are we going to have, in a year? I've never had a harder time predicting what's going to go on with the institute than now. So I think that uncertainty, that volatility, is really creating serious headwinds for scientific research in the U.S. right now.
Mm. What do you think about that when you think about what the consequences are for the patients you're trying to help, for the advance of AI, for the ability to take this advancing technology and begin to apply it in ways that really help people?
Yeah, I worry in some sense we're fumbling the future. I mean, we're right there on the precipice with this powerful transformational technology, and we may be dropping the ball right as we're about to deliver it to patients. And I worry in particular about rare disease patients because for so many of them, they run out of standard care almost by default or immediately. The only care that you get in rare disease is science. And so it's hard not to have an immediate ramification on the rare disease patient community with so much volatility in science right now.
Tell us about, your son passed away in 2020. Tell us what happened and as you reflect on it, what you take away from his life and for what it meant for the people who learned about NGLY1 and their lives improved as a result.
Yeah, so he passed away. It was about 5 years ago this month that he passed away. And as I put it, it was literally the shortest hospitalization of his entire life. I think in less than 48 hours from the time I took him in, he was gone. And I wasn't used to the speed with which something like that could progress in him. It turned out to be sepsis, then septic shock, and then he was just gone. And I think I remarked to somebody at the time, a close friend, I just don't have the time to do the science fast enough this time around. Because I'd had other instances where he was in the ICU where we were literally doing metagenomics in real time, trying to figure out what was going on, and that didn't happen that time. So, one way I put it is, some people wonder, what their calling in life is. I literally never wonder. I know what my calling in life is. I know it's to do the kinds of things I was able to do for him for as many others as I can. And that's really how I think about my day job at this point, is living out his legacy, trying to scale up precision medicine for as many others as I possibly can. And I'm encouraged because, there's a growing number of success stories that we've had where we have been able to make a really big difference in the lives of patients. And I think that the rate at which we're doing it is actually accelerating. And AI is obviously a critical factor in this. So I think I'm grateful for what Bertrand taught me how to do in life and for the opportunity to bring that to so many others.
And how many people do we know about now in the NGLI-1 community?
So I think the known NGLI-1 community is in excess of 100 patients now. And the other really remarkable thing about this community is how fast it has pushed the research forward. So, multiple treatments arose during Bertrand's lifetime that made a difference for him. Multiple more have arisen since. In fact, there was a gene therapy trial for NGLI-1 launched about a year and a half ago. So it would have been before Bertrand's 18th birthday. If you think about that, that's pretty remarkable that you could go from the discovery of a disease to a gene therapy in the span of less than a generation. I mean, that's really impressive when you think about it.
Well, thank you so much, Matt, for being here, for telling your story. It's an inspiring one, and just want to thank you for that.
All right, thank you.
Well, thank you again, Matt and Casey, for sharing the story of Bertrand. That was a really, really moving discussion. We are back with the final poll question of today, sponsored by Zeno Group. So please take this moment to take out your phones and open the Bizibo app that you should have downloaded. If you have not downloaded it yet, you can use this QR code over here to get it from the App Store. Once you have the app open, you'll go to the bottom right-hand corner and press More, and then press Polls. And then you will select poll question number 3. You should see this question over here populate on your screen. And if you're tuning in from home, you should see a link to participate in the poll appear in the chat window to the right of your live stream. But our third question today is, how do you promote patient affordability support programs to healthcare providers? And the options are through sales and or field team communication, with social media content targeted to healthcare providers, through emails and publications directed to healthcare providers as part of a product website, or communicating patient affordability to healthcare providers is not a priority for me. So please take a moment, reflect, and select which one makes most sense for you. That is, how do you promote patient affordability support programs to healthcare providers? We have some fun music playing while you all ponder this question. I'm opening my own app now to see where things are headed. Oh, I need to select an answer. Okay. All right. Right now, it looks like the most popular answer is as part of a product's website. So I think most folks are getting their information directly from the source. Well, thank you all so much for playing the poll questions today. We'll be back tomorrow with even more, but we appreciate you all sharing your insights. And now, for the next session today, for our final session of the day, we are going to talk about a very big topic, which is what it is like to navigate the FDA's drug approval process in this moment. So please help me welcome to the stage Sushil Patel, Chief Executive Officer of Replimune Group, Inc., Linda Marbon, Chief Executive Officer and Director of Capricor Therapeutics, Inc., and to moderate, Stats Senior Writer for Biotech and author of the Biotech Scorecard newsletter, Adam Feuerstein.
I think we're in the way of, we're the last thing before drinks, cocktails, so.
Don't you think that's appropriate?
It is appropriate. And I got a message from my dog, Bo, just a little while ago. He's really mad. I haven't made him as famous as Dr. Mike's dog. So he wants me to work on that. So I have to start doing some selfies with him. I appreciate you guys, Linda, Sushil, both for being here. Under maybe not the happiest of circumstances, we might have been here celebrating and talking about the approval of a therapy for Duchenne muscular dystrophy and a treatment for advanced melanoma. But unfortunately, both of the medicines were rejected by the FDA. Both, they told you that they could not be approved at this time. And we want to talk about that because in this environment where obviously the FDA is under a bit of a microscope from everybody in terms of the leadership changes and the turmoil, the staff upheavals, and what impact that has had, what role that may have played in the decisions that went against both of your medicines. Particularly, I think what's interesting in the commonality here is that you seem to both be sort of cruising towards approval, and very late in the review process, you obviously received some bad news. So maybe, Sushil, maybe you can start and just tell us what, we'll get into some of the details, but like from a high level, what the last few months have been like for you and Replimune.
Yeah, I mean, it's been incredibly difficult, but interestingly enough, you said that in a sort of touch of irony, this was the room that we were looking at for one of our launch meetings. So, that's how sad it is. But no, it's been difficult. It's been, we've gone through sort of denial, shock, anger, and I think now we're really focused on figuring out a path forward. There's a lot of patients waiting for this treatment. I think we have strong data and we really believe in what we're doing, and so that's kind of where we're at right now. It's obviously been challenging for not only the patients, many of our employees with the uncertainty right now, but we're very much focused on figuring out a path forward and getting this drug to patients.
What about you, Linda, for the last few months?
So it's been, a situation of real whiplash. We applied for the BLA that we did based on FDA's actual recommendations. So we went in for a pre-BLA meeting for our HOPE-3 registrational trial. In that meeting, we showed data from existing data sets and natural history data, and they said, well, you have such powerful data for the cardiomyopathy, and there's no treatments for the cardiomyopathy. Why don't you apply on that? And then you can use, HOPE-3 for expansion worldwide or whatever you want to use it for. So we went home and wrote the BLA in record time from August into December. We got ready, submitted it. We responded to 50 information requests between January and June, passed our pre-licensing inspection of our brand new manufacturing plant, told we were having an AdCom, prepared for an AdCom, and we're cruising towards our late stage meeting, sort of backslapping and getting ready for launch when we got the CRL. And it was a shock because we don't really feel that the CRL reflected the ethos in which the application was filed and by which the FDA had originally suggested we file. So thank goodness in our published CRL, which happened when I was in the middle of a banking conference, it said that they wanted another clinical study. And so our stock took a hit that day, and it was all of this scrambling until I reminded people that we have another clinical study. We have HOPE-3. We're luckier than many of our other colleagues because we have this trial. It's going to be unblinded before the end of this quarter. The primary efficacy endpoint, we went back and forth with the agency on switching that to support our cardiomyopathy indication. We believe that they will support that indication based on our Type A meeting, although they won't let us change the endpoint. So if we hit our endpoints, then we have the opportunity for a full label for DMD. So
let me ask you, because both of you guys, you filed under, essentially you've had reviews sort of that have kind of bridged two different FDAs. Going from Rob Califf to Marty Makary, and obviously other changes in the leadership. Both products reviewed at CBER, would it be the biologics division? Used to be run by Peter Marks, now run by Vinay Prasad. What in your interactions, and maybe we'll start with Sushil, in your interactions with the agency during the review, what kind of change, if any, did you notice? In your meetings, your interactions, whether it be, however those interactions occurred.
So, just like Linda, we had our lead asset, RP1, is in, as you mentioned, advanced melanoma. These are patients who have definitively failed checkpoint inhibition or PD-1 therapy. And we had almost a third of those patients with durable responses with a very nice safety profile. We took that to the agency. We got breakthrough designation, priority review, and we, just like Linda, had more than 50 information requests. A very thorough process, as you would expect, as it's a single-arm trial, we'd expect that, and I think the review team did an excellent job and a very thorough job. So, to your point, we then went through mid-cycle, late cycle, we had inspections at our manufacturing here in Framingham. During mid-cycle and late cycle, we didn't get any adcoms or any mention of that, no major clinical issues that we had to address. We were dealing with some CMC requirements as we think about post-marketing commitments. So we really had no indication. Literally until the day before our PDUFA date, we got this letter, and I mentioned shock. That's, I remember my regulatory lead knocking on my door and sort of saying, she looked sort of sullen and just was just beyond.
And you were told, because you filed for accelerated approval.
We did.
And again, it's a while it's a biologic, it's in CBER, it is it's a treatment for melanoma, like you said, and which is an area where obviously accelerated approval is a well-established pathway for, particularly in advanced patients, which are the patients that you're targeting. So I mean, and that has come up now as an issue, right, for you. Like, what can you tell us, if anything, about why it seemed like all of a sudden, the FDA was saying yes, file for accelerated approval, and now no, it's the data that you have is insufficient for approval?
I mean that's very hard to actually understand and get behind, and we've been obviously doing sort of investigation on that. At the end of the day, there was leadership changes. I think we've known there's been various staff changes at the FDA recently, but what we're really focused on is figuring out a path forward, because quite frankly, at this point, that doesn't help us. We are where we are. I think we stand by the data, and we're working, I think, constructively with the agency to find a path forward. The reason behind that is just very hard to actually determine, Adam. I think it's just different leadership. We got no indication that the bar had shifted as we were having these discussions. As I mentioned, we went through very, very collaborative discussions. We had a lot of FDA interactions. It wasn't a one and done. We had multiple we had type B, multiple type Cs, pre-BLA, mid-cycle, late cycle. So, it's really hard to understand.
Do you think that the change, the change in particularly in the offices, change in personnel, there were certain people who left the agency, did that play a role, do you think, in what happened?
Again, I think it's hard for us to speculate what happened. Clearly, there was some discussion and some different opinions coming in late to the process, and who and how exactly those decisions were made is hard for us to actually sort of determine at this stage.
So then I want to ask you about, because obviously Duchenne has been a controversial, it's been a disease that's obviously got a lot of high profile and a bit controversial in terms of the way the FDA has approved drugs. The FDA has used, pretty maximal flexibility when it comes to Duchenne treatments. Peter Marks, the former head of CBER, played a pivotal role in prior decisions, particularly with Sarepta. Tell me about your relationship with Peter Marks, and how is it the same or different with Vinay Prasad, who now is in his role?
So, it's an interesting question as to whether the change in administration should change approval for drugs or biologics, as is in our case. And so one of the things that our entire ecosystem is dependent upon is reliability of the FDA, right? So if you run a public company, for instance, which I think we both do, I know we both do, you are dealing with so many factors. And in this world in which biotech is built from academia to founded companies, we've seen them all, so many of them here today. KJ is a perfect example in sort of what are they going to do with gene editing. And then we heard so many people, the running theme is that we need to support academia to small biotech that feeds into large pharma and ultimately, large drug development. And I think that we have to ask the agency, whoever is running it, to be able to to recognize those challenges, recognize the situation, and then help us work towards approval in a collaborative fashion. And I'm not going to take on personal relationships or professional relationships with the leadership at FDA, but we would ask them to maintain that collaborative nature that we need in order to drive the whole field forward.
But is it fair to say that they've been less collaborative?
It's fair
than they were in the past.
And
I understand it's, and I think people need to understand that this is, for these guys, it's sensitive, right? Because they both have drugs. You want to get your drugs approved, and there's an agency standing in the way of that. And if you piss them off, that could be a problem. But I still want to sort of press you on changes that you've seen between Peter Marks, because Peter was known as somebody who was very communicative and very outgoing and did a lot of outreach with, particularly again in the Duchenne community. it seems like maybe that's not the same now, and I want to get that.
So the whole ethos of the agency appears to have shifted. So under Marty Makary, they have been discouraged from communicating directly with sponsors, is what I've been told, and that's been our direct experience. Peter had, I think, a different kind of style. His style was more, let's talk and sort of figure it out, and then we can sort of make a plan to move forward. It was wonderful for biotech, for us, because we could then decide what we needed to do and then do it. So I think the fact, Adam, that you even just had to allude to pissing somebody off in order to potentially impact the approval of your drug is a little scary, right? Like, it should be based on the data. It should be based on whether it makes human beings better, based on whether, you know, the statistics and the clinical relevance is all there.
And maybe just to add to that, I mean, in my experience, the leadership role at the FDA is like, you really should take a back seat. It's really the review teams that do the work. They're spending hundreds, thousands of hours reviewing your data. They know the data intimately. I think it's really important that the FDA moving forward empowers those teams to make the decisions. I think when you have top-down decisions, that's challenging for companies, especially last minute.
And what we've heard, you know, based on some of the reporting that I've done, it seemed like for you, Sushil, in terms of RP1 and your product, that there was support within CBER. You know, there was support for approval, you know, in that division. But then there was, you know, whether it was Rick Pazder at OCE, you know, or anyone in leadership, where it was overruled sort of at the last minute.
Yeah. Again, that seems to be the case, and we don't know why. And I think what we're focused on is, I think we have really strong data. We've got a hugely supportive melanoma community that wants this drug approved. It's safe, it's effective, and I think in many ways it sort of checks the boxes of what an accelerated approval should be. It should be in an area of high unmet need where you really can't easily do a randomized controlled trial, because further checkpoint inhibition in these patients wouldn't work. They definitively failed. So this should have been a setting where we really would look at accelerated approval as an option, and given the safety and risk benefit, hopefully be something that we would bring forward for patients.
And when we think about Sarepta, and again, you know, for those who don't know, both have drugs and a gene therapy approved for Duchenne, there's one line of thought that says that their experience in the way that the FDA has handled them is sort of beneficial for other companies pursuing treatments for gene therapy for Duchenne, because they have set this precedent. They've kind of the bar, whether you want to say the bar is low or whatever, but then you might also argue that there is there's almost like a pushback against that, and that now it's, you know, it's almost like it, because of like there's sort of the flexibility, now that the FDA is sort of, you know, pulling back from that. I mean, I wonder how you feel. Has Sarepta been sort of helpful to you or hurtful for your cause in trying to get Demericil approved for Duchenne?
You know, it's interesting. I'm not really sure. In some ways helpful, some ways hurtful. I think a lot of the upheaval at the agency can be, especially on the CBER side, of course, would be attributed to what happened with Sarepta. You know, a lot of the reviewers and even mid-level leadership did not want to approve, you know, Elevidys, and they were overruled. And I think they felt, you know, sort of somewhat not betrayed, but maybe their role wasn't considered to be important. And so I think they kind of developed a little bit of a, you know, we want to have our voices heard, and perhaps that's had an interesting reverberatory effect on all of us.
I think we have a question from the audience. Catherine?
Sure. Hi, my name is Debbie. I'm from a biotech here in Cambridge. You mentioned the patient community. I'm kind of curious, what was the response of the patient community in both of your cases?
It's a good question, and I was going to ask that. Thank you. Yeah.
So our patients have gone really quite viral in their response. We actually took a patient and his mother to our Type A meeting with FDA, and he made an incredibly impassioned plea for approval. The reviewers were crying. He has since posted his story on YouTube. His name is Aiden Leffler, so if you get a chance, take a peek at that. Elijah Stacy is another one of our patients and a patient advocate who heartbreakingly posted recently that he's 24 and the average lifespan of a Duchenne patient is 25. And his heart is running out. They know that we have a treatment for the cardiomyopathy, and we've seen stabilization of cardiac function for years in the late stage non-ambulance. This is their lives that are at stake. So they're loud and sending letters to the agency in support.
Yeah, we've had more than 300 patient letters sent to the agency. We've had unanimous support from all the patient advocacy groups, so AIM at Melanoma Foundation, the Melanoma Research Foundation, Melanoma Research Alliance have all banded together, wrote a letter to the agency, again, explaining why there's such an unmet need for patients. Some of those patient stories were very harrowing. I think we think about, immunotherapy and melanoma as kind of we're curing everyone. The sad reality is 50% of patients will progress, and there really aren't great options, particularly when they've had definitive progression. And so that's why we were filling, I think, a very important need for those patients. And again, not just the efficacy, but the safety we bring is one of the real sort of benefits. A lot of the treatments they're using today in that space are very toxic. High grade 3-4 toxicity, and we had a very tolerable profile. So again, another option that was desperately needed by patients, and they've certainly expressed that. We also had a huge number of physicians, top treatment centers who treat thousands of melanoma patients right in, and it wasn't just a, hey, FDA, this is an important treatment, we need it. It was really going through the CRL points around heterogeneity, contribution of components, and kind of explaining why, actually Replimune did a really thorough job and really is trying to establish a bar and bring the science forward. If you think about checkpoint failed disease, this is a challenge across many, many tumor types, not just melanoma. And the fact that we haven't got a lot of approvals explains why it is a difficult setting. It's why we try to set a high bar. We try to use very strict criteria so that we know that these patients have definitely failed and that the contribution is really due to RP1, not some sort of magic happening to these patients. And the fact that we're getting into long-term benefit, I think, is really important.
So this is not about patients or families' responses, but I think listening to Sushil and sort of the panels today, I think the overarching question with the FDA right now is why? What is the value of turning down a therapeutic that is safe? Understanding, you asked about Sarepta, like there's a lot of safety overhang on Sarepta's, drug, and that can raise a lot of concerns perhaps. But if you have something that's safe and the data suggests that it works, we have to ask ourselves, what is the real value of the statistics? And what are the statistics telling us? The statistics tell us how likely is your data or what you're seeing in human beings due to chance. And if you have any chance that you're making humans better, why not grant accelerated approval or some opportunity to move something forward? And then you always can pull it off if it doesn't, meet that standard at some point in a confirmatory trial.
Do you think it's because in your situation, and just to sort of quickly summarize it, you filed on a clinical trial that had, let's call it mixed results. I mean, there was some issues with the study. It was a relatively small amount of data. And so there were going to be questions about whether or not the data were going to be sufficient, strong enough for approval. But at the same time, sort of wisely on your part, you had started a study called HOPE-3. It's a randomized controlled study, phase 3 study that was going to be kind of the next study. You have that almost completed. So, I mean, is it unreasonable for the FDA to say, hey, you know what, these data that you submitted are not, we can't render a judgment on these, but let's just wait a few months for the HOPE-3 data to come in. It's more definitive. We'll have a better idea of the efficacy and safety, the profile of your treatment.
Absolutely, and we understood that, and we had that trial ready to go. We filed the BLA based on the fact that they had seen the data, all the things that you raised and wrote about, and all of that. In terms of the HOPE-2 data and whether it would be enough, too small sample size, whatever. We knew about that. They knew about that. They knew about it when we filed the BLA. The issue is, as I started this panel, was the whiplash, right? That we were dragged back and forth. If they told us on January 10th, look, you have another trial that's going to read out in 11 months or 10 months or something like that, let's wait. Fine, we would have then focused on getting there and conserved capital and managed the company in a different way. So I think that's really the issue. But yes, absolutely, I do see the value in HOPE-3.
And that's just to continue that thread. The study that we're talking about, HOPE-3, it is completed. I mean, you're basically at the final stages analysis, and tell us a little bit about when we're going to see data from that study.
Yeah, it's a 105-patient randomized double-blind, placebo-controlled trial that's due to read out before the end of this quarter. The primary efficacy endpoint is the performance of the upper limb, which is arm, hands, and shoulder movement with a key secondary, which allows labeling of cardiomyopathy measured by ejection fraction.
Yeah, we also had the same question, hear the same thing again and again from physicians, why not? They're really scratching their heads and trying to understand why is this the drug and the situation the FDA really wants to dig into, because as I explained, the unmet need's tremendous. The product's safe. It has a really, I think, compelling safety profile. And so the why not questions come up again. We also understand it's a single-arm trial. We actually had started our randomized confirmed trial, understanding that this is a commitment we have. We know there's been some bad actors in the past and haven't sort of delivered on that. And I think the FDA has appropriately come down quite hard and saying to sponsors, you need to get these trials done. We were providing regular enrollment updates to them. The trial, I think, is very attractive and is enrolling well. And so it's an overall survival trial. That would have been the definitive evidence. And by the way, if the drug hadn't panned out and didn't deliver overall survival benefit, I think no one wants a treatment out there that's not going to help patients. So that was a commitment we made and understood. And so we're really hoping that we can have that discussion with the agency.
So is for Replimune, is the accelerated approval pathway, do you feel like that door is shut and it's not going to be reopened? And now the discussion moves towards the phase 3 study, which would have been the confirmatory study, but now may serve as the study that you need to get approval. Where are you in that kind of conversation?
I think we're really sort of assessing all options and pathways and having a number of discussions with the agency. Of course, yes, I think the phase 3 is a path forward. What's not appropriate for us is to wait 2 or 3 years to get to the end of that study. And obviously, from a funding situation, we've made a lot of investment decisions, won't allow us to do that. However, we're certainly open to working with the agency. That being said, I think we're also looking at more near-term opportunities and sort of additional data we provided the agency to maybe help them get over the sort of line in terms of what they're looking for based on some of the feedback we've had at these meetings. So right now, I think everything's still on the table, but we're very much focused on trying to get this drug approved as fast as possible.
Is there a timeline that you can offer in terms of, ultimately you're going to have to make a decision, right? Whether, whether accelerated approval is something that's possible or not, and then what to do. Because I know you've talked in the past about whether, you've said in the past that if accelerated approval can't happen, you may just drop RP1 and move on to there's another product, RP2, which is a similar product, but another product in your pipeline. I mean, is that
I think there's two different things. There's accelerated approval based on the phase 3, because, again, FDA guidance has allowed that, where you could look at potentially an interim readout. There's also accelerated approval using IGNITE and maybe a combination of IGNITE and phase 3. So I think there's different permutations where we're having discussions with the agency around what would sort of meet their criteria and satisfy some of the questions they raised at the Type A meeting. So right now, I can't give a definitive timeline on that, but it's sort of in their court right now, and we're hoping to have a sort of a response to that very soon.
So if you were to speak directly to FDA leadership right now, maybe they'll watch the video of this.
Hopefully.
What's the message that you would want, based on kind of what's happened to you? And obviously you don't want, other companies don't want this sort of things to happen to them. You want consistent guidance, obviously, and that, like your point was that, if we had known this, we would have done things differently, and I'm sure you would have done the same, right?
Yeah, again, I think we would have welcomed these conversations much earlier in the process. It's not like there wasn't multiple opportunities to have these discussions. I think these things like heterogeneity and contribution effect are kind of known issues and that you get with all single-arm trials. And so I think having those conversations early, really working collaboratively with the agency, what else they would wanted at an early stage, we could have course-corrected. What's difficult for us now is we made decisions such as manufacturing. We do in-house manufacturing of our own product here in Framingham, Massachusetts. So, it's not easy to turn that around on a dime. We're doing commercial fills. We're making decisions and spending tens of millions of dollars based on prior discussions we've had with the agency. So to suddenly get something at the end, that's not easy for us to easily address. And obviously, we'll do what we can to address the funding situation and continue our trials, but that isn't something we can do indefinitely.
Yeah, exactly, the same type of situation. So, we are working closely with the agency right now to submit the HOPE-3 data. We have another meeting scheduled with them coming right up to make sure that we give it to them in the form of a response to the CRL that they find appropriate. And we hope that we'll be able to get relatively quickly to PDUFA should the trial meet its primary efficacy.
Yeah, I would just say in terms of drug development, look, it's hard enough as it is.
Yeah.
We've been at this for 10 years. I mean, even, the amount of work and effort we put into our clinical trial to collect the data and make sure we have this robust criteria it's difficult enough. So I think whatever we do, I think we need to have a discussion with the agency and sort of appreciation of, I think this is going to kill drug development. If you don't have consistency and transparency, it's really going to make it very difficult for us to bring, I think, innovative treatments to patients faster. I think the US is an innovation hub. The founders of Replimune have brought several companies over to the US based on sort of the investment environment and being able to raise capital here in an effective way, and the regulatory environment. And I think in the past we've worked with the agency to come up with solutions on how do we bring these treatments, particularly in high unmet need areas, forward. So I would hope that we could go back to that discussion, that partnership. And again, this is not about being in industry's pocket. This is about doing the right thing for patients and coming up with, in some cases, innovative trial designs so this kind of ecosystem here we have in Boston can continue to thrive.
Catherine, was there a question? Oh.
Jonathan Rosen, I'm a family physician. So I know it's hard for you to speculate about internal communications in an opaque organization like you're dealing with, but is it possible that there's a mandate to make decisions not based on efficacy, but more based on financial considerations, and that they don't want to pay for these expensive drugs?
So we're definitely hearing that that's something that's going on within the leadership, that the concept of, what are these things going to cost, and is it going to, have a marginal impact in efficacy, and is it sort of worth it? And we know, that's been, one of the issues that Dr. Prasad has published a lot of op-eds on. So we are aware of that. We haven't seen evidence of that as yet, but we have heard that that's one of the issues that could be driving some of these negative opinions.
Yeah, that hasn't actually come up in any shape or form, but certainly there could be discussions happening at leadership level on that.
Hi, I'm Tim Leshin with the Association of Schools and Programs of Public Health. I've been listening to this podcast, Why Should I Trust You?, where they have folks from MAHA on there. I'm not a MAHA person myself, but they would say that they're very worried about the conflict of interest of industry when it comes to drugs. And I think, you ask the why, I think it's because they're listening to MAHA, they're not listening to science. And I think we have to think differently about how we communicate the great breakthroughs that you're talking about, so that folks who are very skeptical about the inconsistencies in industry aren't just listening to the other side, that they understand the implications of their opposition.
So I find that absolutely fascinating, and one of the points I was actually going to make here today is that one of the mandates of the current administration and sort of the MAHA movement is this movement away from conflict of interest and big pharma, and yet a lot of what's happening is driving back towards that old model of big pharma, right? Because if Pfizer got a CRL like I did, and they had to go back and do another phase 3 trial in an indication like Duchenne muscular dystrophy, and they had to spend, I don't know, $20, $30 million. I don't know if their stock would go down 5 cents, right? But for me, it could drive us out of business if we have to do another clinical trial. So if something goes sideways with HOPE-3, which I don't expect to happen, everybody knows I'm a super optimistic person, or I wouldn't be doing this for 20 years. But the concept is that it drives our companies out of business. And so the way that they can get around that conflict of interest is to actually promote small companies and let them get to where they're earning revenue and building new products and then feeding sort of the not big pharma model.
I think interestingly on the MAHA thing, that's kind of the right to try model, right, as well. So on the one hand, there's no, and on the other hand, the right to try should be something that particularly, again, situation like we're in a high unmet need, safe and tolerable drug. So there's sort of two sides of that equation. And again, in terms of transparency, I think we're fully open to whatever the FDA wants to do about that to ensure there's appropriate company involvement. And how do you have a collaboration if you don't actually engage with each other? So I think we've got to get to a point of trust and respect and figure out how we actually move forward in that environment. I think just saying that we're in FDA's pockets is just, or I don't think it's helpful, and it's really not moving the needle forward.
We are out of time. I will be following both stories closely as you.
Oh, you will.
But you'll be hearing from me. I want to know when those results are ready. You Absolutely. Please call me.
I will.
Sushil, Linda, thanks so much for your time.
Thank you, Adam. Always a pleasure.
Oh, great. Well, that's it for today. There's a lot more tomorrow. Thank you all for being here. It's lovely to see such a packed room. The bar is opening. I wanted to say again to thank all of our sponsors. Just up on the mezzanine, we'll be hosting a networking reception brought to you by the Stat Wonderkind sponsor, the University of Texas MD Anderson Cancer Center. There's food, drinks, please enjoy, and there are these. This is the Rising Star. It is the Wonderkind's cocktail, and you are all invited to go upstairs and get one. We'll see you back here tomorrow. More great programming. We'll hear from the president of the American Medical Association. We'll have some questions for him based on today, you might imagine. We'll hear from Lily's Dan Skavronsky, Nubar Afay and Ari Beldegrin, and Rick Burke's interview with Joe Kennedy, which I think will be great. We'll close the day. So thank you all. Hope to see you tomorrow. Cheers, Catherine.
Cheers.
Some margarita.